All right. Hey, everybody. We are out here just off Brazil, looking at what we see in the midwater. All midwater, all the time. We're looking at a large mucus structure, wondering if at the bottom there is an organism hanging that secreted this structure. Could either be for parachuting or it could be, oh my goodness, it's a larvacean. Okay. They're going to want to scan this.
They want to scan it?
But they're not here. So, I don't think we can do it without them here to start this, but we could collect the animal, which is this little guy at the bottom, arrow-shaped thing with yellow head and gray tail. And I think a D-sampler would be the best way to sample it. If we can just hang here for a second and give them a chance to come in. Hopefully they're watching the screen.
Connie was just finishing up. There she is.
Good. Large larvacean. Biggest larvacean house we have found so far.
Oh,
Yellow head, clear tail, hanging at the bottom, shaped like a big parachute, basically.
Oh.
So, can we scan?
We have
No?
one shot.
Yeah, we have one shot.
Looks really
It's super delicate, super diffuse. All right, so it'll take them a second to get set up. I think we have enough that we know there's an animal there, so now we'll just kind of back off a little bit and wait until they're ready, till they give us the go-ahead to, Slipo. DeepPIV.
Yeah, so I guess we'll probably want to center up the animal as much as possible. The challenge is it's hard to see the animal from far away.
So right, you can leave like nuts on. That should give us some.
I'm gonna turn the numbers off yet. I come real quick just to make sure you can still see the animal.
Okay, try
We got
meats.
mids and uppers right now.
Shut off meats first.
Okay.
I still can see something. See that? Then when I get
Who
really
is supposed to
super
be on
close, then
sea
we
log
go
right
for
now?
They've been leaving the center and the lights on. So I'm gonna turn off the lower capex.
Okay.
So the center and lights and uppers are still out.
Okay, I think I still see it.
Too
I'm not
much?
sure.
Is it
No, no, it's
still
okay.
out there?
Okay.
Yeah.
So I had to try to do the sea lock.
I'm getting closer now. You get ready?
You ready, Kaikani?
Yeah, we're ready. We're recording. We're ready to
Okay.
take the uppers off.
I'll let you know when. Once we get
Super,
it in December.
super
I'll wait for
delicate.
you. Oh, you have the proper feeds?
Come on, closer now. All right, get ready. Okay.
Pick them up?
Yeah, I think so.
I can't see it at all. It's so diffuse.
No, I'm not sure.
It's in there.
Got it?
Keep going for it.
Keep going. I can't even tell what's house and what's just marine snow. Such a diffuse thing, but this is probably the best view.
So you see there is structure there.
There is some structure.
There you go. Something there.
Oh my god.
Or something.
That is
Okay.
crazy. You can't even see.
Reverse.
Super slow.
Bring the light up and do it again.
Let's try it one more
Try-
time.
try it
Looks
live.
like it's still intact.
Mostly still intact. Kind of in two parts. It's this super diffuse cloud.
To the right more.
How's that?
That I think is good.
Going forward. Right.
More to the right, did you say?
So,
A little more to the right.
all right. Now,
Because I think this part is the one she wants. So move that over just a little bit before you go forward.
straight as possible.
As of
Very gentle forward. Oh, see, there's the central part of the house is just up.
okay, coming higher.
Higher and then reverse. A little bit higher. Okay, now reverse.
Oh, got it
Yeah.
that time.
Yeah, so it's in.
It's in this top upright quadrant.
So I think this
Totally
bright
can't see it.
spot that's almost in the center, I think that might be the animal.
All right. That one to the right, right?
Yeah, so I think you now have it centered up.
Okay.
Now it's all high.
Go forward.
I think we have one more shot in it.
That one there in the center.
So you want it a little lower. And a little bit more to the right.
It's up in the neck.
It's a little lower. Okay, now reverse.
So there's one part that's a little bit denser, and that's the one we really.
A little bit higher.
need.
Okay, try there. Okay, and then reverse. So now it's to the right. Okay. I think we might have gotten something there. The signal is really low.
Super diffuse.
It's possible that it just.
I mean,
Okay,
I think
well,
we've
let's,
got it a
um,
couple of times, but still there's this dense bit there that you can't see at all until it hits the DeepPIV, the laser.
So go, try and do forward one more time. Oh, go straight. Straight, straight, straight. Straight. All right, now reverse. Okay. All
That's
right,
good.
if we can collect that, Karen.
Yes, that is the objective.
Okay.
I
D-sample?
don't see him. D-sample would be great.
Okay, so what is
So I think he's one or the other of those bright dots next to this lower batch.
As long as he stays in the house, this is going to be easier.
It would be better. I think it's this one or this
You
one.
zoom in.
Better look. I think it's this one. May not be. No, I don't see the tail. The tail was pretty easy to see. The head was yellow and the tail was transparent.
there?
don't think it's either of those, unfortunately. Can you look at, there was another bunch of it up to the right slightly. Yep. I think it ran away. I have no idea what animal that was. I think he got away. What about this?
Some structure.
Some structure, but super, super diffuse.
Yeah.
Whatever it was, like I couldn't visually see any inner house until we scanned it, and then you could see there was density there.
Yeah. Yeah. Can we zoom in just on like that big dot? Because it looks like there's a lot more there.
It's in the camera. In the camera. Okay, so there are some big houses here somewhere. 600-700 meters. That's pretty deep for a. Oh, yeah. Hi.
Thank you.
Thank you for coming in. I was like trying to do it and trying to keep an eye on it, and I was like, ah.
That one there.
Yeah, I don't think it's, the tail was pretty visible. It should be distinctive. Russ may be able to give us a genus, at least, from the photos.
Yeah.
Yeah,
All
all right,
Well,
pretty sure it's gone.
a mystery mucus house.
That's fun.
Cool, let's find some more of those, because that was so fun, right?
Yes, of course. That was not bad.
All right, that was super cool. This is the first larvacean house we've seen that was not just walnut sized. And so we want to scan those with one of the cool pieces of technology that we brought along with us called the DeepPIV. It's a laser sheet. That's the red that you see that goes out, and then we drive very slowly and steadily that laser sheet through the animal, and if it's transparent or it's mucus, then we get the 3D slices of what that animal looks like, and we can reconstruct we get 2D slices of what that looks like, and we can reconstruct those to make 3D structure. For these really delicate mucus houses or really delicate transparent animals, it's one of the only ways to get their 3D structure, and awesome to do it in situ where we don't hurt them, it doesn't disturb them, and we get information that we can't get by collecting the animal and bringing it up to the surface because they're just too delicate or they're too large, or they won't make their houses in the lab. And so it gives us an idea of what they're doing. Larvaceans are really cool animals. They look like a little flattened tadpole. They have a body on the front and a tail in the back. They use that tail to pump water through this filter house that they make. Essentially, they sneeze out a bunch of mucus. That mucus is actually really delicately structured. You can put it under a scanning electron microscope. Oh, off to the far right, there was a lobate, yeah. So if you put that mucus under an SEM, a super high powered microscope, you can look at the filter size of it. And it's very specifically sized for the type of particles that animal wants to eat. And now with this DeepPIV, we can look at the structure of the houses and how they push water through it to concentrate the particles of the size that they want. So we got a nice scan of the house potentially. Unfortunately, the animals have a little trap door in the bottom of the mucus house that they can just zip out of when they feel threatened. Their house gets disturbed. And that is probably what they did while we were scanning it. Animal, that one was really cool. It had a yellow head and transparent whitish tail. But we're not sure which one it is.
So the one we were looking at, that's the one you were searching for?
Yeah, we
First.
would love one of those.
That's the one you mentioned earlier? Super.
Oh,
strange
no.
name.
No, Tomopris, gossamer worms. No, these are larvaceans. These are something that Russ really wants, and also Kakani is really interested in the house structure, and Russ is one of the world experts on that group as well. So we have somebody who can actually identify them and somebody who can look at the structure of the house. So, a great combo. No, the gossamer worms are, well, we'll see one. We've been seeing one or two per dive at least. Oh, what is that? Okay, so good. Yeah, no, that's great. So we're just working our way down and forward. Is that an aberration on the screen? That black spot that's not missing, not moving. It seems very dark, but only on the right side of the screen, Sarah. The science cam seems really dark on the right side for some reason. Are we pointed over a lot?
Yeah, that came up yesterday too.
Can we look at this?
Turn the mid side.
Let me look at this guy up there.
You want to?
Yeah. Sorry, I can talk louder. Don't talk that loud.
That one.
No, I think I'll up this way a little bit more. That. It's like a calicopheran of some sort, but it's pretty beefy.
That in
Yeah, this guy. Nice big copepod there. So our objectives on this cruise, we're working about 360 miles off the coast of Brazil,
that thing.
just below the bump.
I'm gonna bring it to the middle.
And our objectives are to sample midwater diversity quite broadly. We want to build reference libraries for the animals that are out here so that when people come out and try to use eDNA or environmental samples to tell what's here, they will actually be able to identify things that are here because we have come first and identified the, oh, look at that.
Oh, sorry.
Look at this blackfish. If it decides, nope, it's not going to stick around. Yeah, we'll stay on the Siph. Yep. It's just Brilligama. I'm going to guess Brilligama. They're like little baby arenas in my thinking, but I could also be completely off because I am not a Nidarian person. No interest in scanning this guy though?
That's pretty small.
It's fairly small.
No.
Okay, all right, we can leave it. It does glow beautifully. If you poke it, it will shimmer blue all around all those nice bracts that it has. The bracts are the gelatinous leaf-like structures that protect the body. So that transparent part that you see below the swimming bits. onward and downward.
Okay. Okay.
Yep. So we are at 733 meters. We're headed down and forward, hunting our way down to 1200. Trying to find a nice cross section of the animals that are down here. There's a question in the chat. Are there animals in the trawl that we are not really seeing in the ROV? Yes, definitely. Here's the little jelly. Let's just get a quick look at who that is. Just get a quick look at this guy. Sorry. We tend to get smaller, halotrophes. We tend to get smaller things in the net than we can really work with easily.
Sure.
Halotrophes. It's a Tracheomedusae. We've seen several of these, so we can probably let it go. Although it was beautiful. But I think we've seen at least one a day.
Yeah.
You get the location?
No.
Hmm?
Really cool one. I don't know what it was. Yeah. Yeah. And its house was so diffuse, it was really hard to, you couldn't even tell where the inner house was until you scanned it. And you're like, oh, it's right there. But
Small
like,
house.
couldn't tell it otherwise. Yeah, too small. I mean, we could try.
Only way you want.
We can try, but things that look a lot like marine snow and are very tiny can be very difficult to collect. Because everything else is super easy, you know.
Sure.
What is this guy? Looks like a mysid or a shrimp. Yeah, you can go by. What? We get those in the trawl?
Most should do.
Not really.
That's how they see.
Yeah. Almost all the shrimp spend most of their time vertically. Fish over there. Right, so back to our objectives. We want to build the reference libraries for a broad swath of the animals that are out here, and for that we need good identifications. We need a voucher to go in a museum. All the vouchers for this cruise are going in the museum in the University of São Paulo here in Brazil. And we need sequence. We are sequencing on board, which means we will have the data before we get off of the ship to have the genetic data for most of these specimens. And that will take care of one of the objectives of the cruise. The other objective is to image a lot of these animals that are really super delicate and can't be imaged any other way except doing it down where they live. So those are kind of the most basic objectives of the cruise. Another objective is to bring together a lot of the Midwater community. And so we've put together a team here that includes researchers from 13 different institutions on 4 different continents. We have researchers from University of Western Australia, from Tohoku University in Japan, from Harvard University, from University of Rhode Island, from the Smithsonian, from Monterey Bay Aquarium Research Institute, from University of Alaska, from Stanford University, from University of São Paulo, from Federal University of Rio Grande. So it's a big team of 25 scientists out here looking at all these amazing, cool animals, sometimes puzzling over them, sometimes knowing what they are right away. For example, yesterday we collected a dark red gutted Tomopteris worm, or a gossamer worm, and just so happens that we are just about finished with a description of 2 other species that look very much like it. And because we have already done that species work to describe those 2 other species when we got this one, it was very quick to tell whether it's different from them or not. We'll confirm it with genetics, but there are several morphological characters that definitely place it in that group, but distinguish it from the 2 other species that are about to be described. So that's an exciting first confirmation of definitely a brand new species that has not been seen before. And we'll get that description added to that paper and that submitted in the next month or so. Should be exciting. Cyclothone, reportedly the most abundant vertebrate on the planet. There's a Ketognath off to the left. Ah, there's a radiolarian. What thing that looked like a puffball. So on our dive today and all of the dives this coming week, we will spend all of our time up in the midwater. We don't have any plans to go down to the bottom, which is at about 5,000 meters where we're working. We will be exploring the mesopelagic from 200 meters down to about 1,200 meters, which is just into the bathypelagic. We just want to get an idea of what's here. Cover as many groups as we can, get some beautiful footage to show you guys what is out there. We're headed to 1,200 meters today. We may get there, we may not. But we're gonna try. If we get distracted by too many amazing animals before we get there, we may not make it.
Buenas tardes, mi nombre es Silvina. Y les voy a traducir algunas cosas que estuvo hablando recién la jefa de esta investigación, que su nombre es Karen Osborne, que es de la institución del Smithsonian de Estados Unidos. Vamos a tratar de ir hasta los 1.200 metros, pero esta expedición no va hasta el fondo del mar, que acá el fondo es más o menos a 5.000 metros de profundidad. Esta expedición se trata de investigar organismos del zooplancton de media agua, o sea, organismos que están entre los 200 y los 1.200 metros de profundidad.
Marine snow is a really important source of carbon moving from shallow water into deeper water. Many of the animals here eat marine snow. On marine snow, bacteria grows. Excuse me. Oh, what's this? Oh, it's probably one of the radiolarians who's just making a funny hand signal. Not a rude one at all. Just
Es materia orgánica que va a decantar hasta el fondo, que son restos de organismos muertos,
I think it's
restos
up to the right a little bit.
Entonces todos esos puntitos blancos que vemos en pantalla son llamados de nieve marina.
I would like to look at it. We may collect it.
Fernando, no vamos a ir a Fernando de Noronha. No. Vamos a mantenernos en la zona de aguas internacionales afuera de la zona exclusiva de Brasil.
Which means it was probably really close to the camera, so we can leave.
Económica exclusiva de Brasil, solamente en aguas internacionales.
Can you just zoom really quick on that jelly out there? Looks a lot like Halicreus. Too far away.
Y Karen, nuestra jefe de expedición, estaba explicando que hay bastantes especies, probablemente van a estar describiendo nuevas especies en esta expedición. Ya encontraron algunas que son morfológicamente diferentes de otras que ellos ya conocen. Y están haciendo también, además de describirlas morfológicamente, obviamente les van a hacer un mapeo genético para poder diferenciarlas de otras ya descriptas. Entonces en breve, en los próximos meses y años por ahí, van a empezar a salir artículos científicos donde van a estar describiendo estas nuevas especies para la ciencia que fueron descubiertas durante este crucero.
The way to do it, right? Because I think it's hard to ask people to
Not the jelly, I thought.
call
We can
in
leave it.
and watch these dives. It's like you fly, if you don't have space on the boat, you should just fly them to a location so that they're extricated from their normal lives to focus on the stream. I think that would work.
I went to Silver Spring for the first ever Gather's Midwater dive, and that was partly because of that. Because couldn't get out on the ship, but they wanted something.
It is good.
Completely focused on it, so they shut me in a room every time they were diving.
Yeah,
That kind of a model worked pretty well.
right.
Well,
So you can have like three
Yeah.
safe houses.
If it's a shrimp.
Safe houses. I mean, because otherwise, like, there's no way to do this from home. Like, I would have other meetings and other things and commitments to be able to get away from family and life. So
It is. Get a little video of this guy. It's awfully pretty. Oh. Also a little shy, it turns out.
DSER, so that's a shrimp.
We can leave him. We can leave him. Like a jelly type shape. It's a larvacean house.
Oh, that's a Larvacean.
It's another one of those bumpy ones.
Esas que estamos
Are
viendo
these
es
too
una
small for you
casita
guys?
de mucus de un larváceo. El organismo construye esa casa.
Pretty small.
See it.
It's pretty small because we can't see him in the sit cam. He's pretty close since he's out of our focus.
super
Okay,
close.
too small. Yep, let's leave him. It's a little jelly here.
Yeah.
Small but mighty.
Did you see the big one, Russ?
Hmm?
Did you see the big one?
No, I just.
It had a yellow
heard
head
it.
and a transparent
With the
tail.
clubby fingers.
Clubby fingers.
You mean the
Clubby.
filter? Yeah.
Oh, this one was. No, it had this huge parachute-shaped house, and it was down at the bottom. And it had a very distinct yellow head and a whitish, almost diamond-shaped tail. The part you could see.
I'll have
was
to
kind
look
of
at a,
milky
I'll have to
whitey.
look
Yeah.
at a replay.
Oh, that is a pretty little guy. Halicera. This looks like. Those are gonads, correct?
Yes.
Big,
H-A-L-I.
big eggs. Look at the size of those eggs. They're huge.
Oh, it's got a slight.
It's something else.
Yeah, it's okay. We'll stick on this guy. Any interest in this Dougal?
If you'll suck it up, we'll take it.
Do we want
So family I'm working on, it's really small.
But with those big eggs, it's easy to see. Let's suck it up.
There's only one.
I would like some genetics on this guy.
Okay.
bring out around?
They're on number 5.
So this is about its top speed and its most evasive maneuvers. So hopefully it's small enough. That means we can get it in.
Oh, I can see it. Very small.
Ahora vamos a
It's very
intentar
small.
succionar esa medusa que está en pantalla. Para eso contamos con la habilidad de los pilotos ROV para que lleguen cerca del organismo y lo puedan succionar. De esa manera muestrearlo y poder estudiarlo mejor de más cerca.
Straight up. Straight up front. Perfect. Almost. Almost. Yeah. All right, now just drive forward, turn off the suction.
Ahí
And
en
we
la
have the
imagen vemos
buckets up already.
el container, un frasco, que es donde,
We're starting on
se
bucket
guarda el
5.
animal una vez que es succionado. Entonces, una vez que en la imagen aparece como que se succionó, hay que prestar atención al frasco para ver si realmente entró hasta
So
ese
just
container
drive forward
puede
slowly
ser
until we
traído
see him come into the bucket.
a
It'll take a
superficie para
while.
estudiarlo.
The shadow cam is on there today.
Hola Silvia.
So rigged into the
Silvia
middle
Romero
of the
fue
suction hose. The animal goes
figura
down the
principal
tube.
de
We
un
pump
uno de los
water at the
proyectos
back
que
of the
fueron
hose into these
desarrollados en
canisters
Argentina
at the back
el
of
año
the
pasado.
RV. And the animal travels through a hose. In the middle of the hose, there's a little chamber. And on either end of the chamber is pollinated light. It's another Frilligama. As it passes through this chamber, it gets imaged by a camera that basically takes a picture of its shadow. And then it carries on out of that chamber, hopefully back into the bucket. I have not seen it come into the bucket yet. We have lights and a camera back on the bucket so we can see when it makes it in there. Still no sign of it. There it is. All right, we'll let it settle a little bit before we index.
Go to 4 or to 6.
Yeah. Okay, I think you can index.
6?
Yeah. 6 would be great. Super. Awesome. All right.
What do you wanna? Lara, you wanna do one hour, or
Bueno, voy a tratar de hablar más alto
John,
igual.
can I have a screen with show
Yo
feed?
trato
'Cause I
también
like
de
the
dejar
time code on the
a la
bottom of the
investigadora
feed
principal
camera somewhere. Can I have
explicar también, porque
feed
obviamente ella
somewhere
tiene mucho
so
más
I can see the
conocimiento
time
que
code
yo.
on the
Yo
bottom of
trato de
that
ayudar nada más.
display?
No soy
So,
especialista en
20
zooplancton.
Maybe this one.
we just put next up.
Something giant.
Yeah, that the one was scanned.
Oh.
But probably
Okay.
strawberry trust enough. Sorry. There's one.
There's one. Still pretty. It's not terribly small. Either that or it's right in the camera. That is a different looking house. Yep. So we're going to want to scan this with DeepPIV.
Okay.
Give a time code, so just go with this.
Yeah. Here is
You want me to leave off or
no, I'll let you know once
It's a larvacean.
Unless
It's
Joel,
kind
you're
of
okay with me
appendicularian,
zooming in?
so it builds this house.
I
Yeah, it builds a house.
not
that
yet.
acts as
Let
a
me
filter.
get
Off
it
the filter
closer
goes
and
out. And then every day or so it leaves the house because it gets clogged and it builds a new one.
And going.
And it's an important form of carbon transport to the bottom of the ocean. You see these sinkers going to the bottom of the ocean.
If we collect data from it, write it down here, so like this one, I don't know. Oh, here's the
Can I zoom in?
Yes.
You just put the time code.
Bueno, lo que están tratando de escanear con el láser
What is
es
that?
I already wrote this one.
okay.
Oh, no, we're doing this one.
ellos construyen esa casita de mucus. Esa es la que
Um,
vemos
so
por
we're
fuera, es
going
una
to
casa de
carry
mucus
these,
que la usan para atrapar partículas y de esa forma alimentarse. Y esa casita la construyen y la abandonan todos los días y cuando la construyen otra, porque la casa se llena de materia orgánica y se tapa, entonces, hablando medio de forma informal, y esa casita es abandonada. Y esa casa va a decantar hasta el fondo y de esa forma contribuye con llevar carbono al fondo de los océanos.
So if we collect data, like we scan it, then we write it down, we collect it, then we write it down here, that's what it goes in the I can do it at the time.
Nah, almost. It's a little outside
Take a little
to
break
the
in
right.
between.
Yeah, no breaks. We don't take any breaks. All right, it's back in.
We actually tried a reconstruction of one going back and forth recently.
Yeah.
We tried really hard.
And
So far we have not been successful.
oh no, keep going. All right, we're almost there. Yeah, the pauses kill us. Like that's, I mean, that was pretty good. A little bit slower is ideal. It's back in.
Hay que tener bastante paciencia para poder escanearlos porque imagínense en un mundo
That was pretty good.
donde no hay
have another
superficies
shot?
es bastante difícil que se quede quieto el organismo para poder
Right.
escanearlo.
Ah,
Entonces depende mucho de la destreza de los pilotos del robot.
reverse. Ah. Oh, it's maybe too high. Ooh, that was pretty good. Is Russ around? Does he want us to collect this?
We would definitely like to collect it in a D if it's big enough
Okay.
and it's still there.
Yeah, we'll see.
Ooh, okay, well, I think we got a couple good scans, so if we want to collect
Okay.
it.
Let's put this in a D.
Thank you, Joelle. Well done.
Yeah, the part that tripped us up is like whenever we have anywhere where we go over the same position, even if the speed varies, like we're better at
D
direction
number
for
4.
that now. But like as soon as we start going back, it's very hard to stitch that right. I think we're better off in that case accelerating
That's the
through.
inside one.
If we have to choose between going too fast or too slow.
Gracias, Silvia. Sí, somos dos argentinas a bordo. Julieta Ceboar, que está ahora haciendo el Sea Log, que es como un registro de todo lo que
I
hace
think it was in.
el robot.
Probably went through.
Y,
I thought it went in, but I don't know if it went
I
y
in.
yo
thought it
estoy
went
acá
in, so I
tratando
think
de
it might have gone
ayudarlos
down.
con el chat.
Couldn't tell if the animal was still there.
I don't
Can we
see
look
it.
down?
Let's confirm.
Who's that? Yeah, let's look down in the bottle and see.
I think it's right there. Top.
You see it?
Right there. Yep. Right here.
Oh, yeah.
Oh, yeah.
The house is there.
Yay.
Woohoo! Yeah. It's so
It's so faint.
It's so faint. It just floated past the top.
Okay. Hello.
Which number?
D1.
D1.
G.
Hey.
Yeah, it's probably down between here now.
I'm not seeing anything.
I definitely saw it.
There's always this
I don't know if the
suspicious
animal
D.
is there, but I definitely saw the house.
Okay.
Did we figure out what
They were good last night, both Ds.
was it?
There, over there.
All right.
And the animal is there.
Right.
Awesome. That is great catch.
Well done.
You collected a tiny thing we can't actually
There you
see.
go to Larvacean. Yeah, I think we have the Larvacean. There you go.
Yeah, in there with its house still.
Where they-
Awesome.
They won't stay in
Right.
there for long. They usually escape. There's a little escape hatch. They
let's
swim
see.
out of their house.
12:45.
Acá están festejando porque pudieron capturar en uno de los contenedores el
Is it
larvaceo
Joel or
con la
Joel?
casa. y todo.
Spoil.
Well,
Well, then that's not
it's kind of an unusual name in Mexico, no?
That's how it sounds
spelled
to me.
well.
Yeah, but it's not a typical name, I don't think. It's easy for me to remember. It's my biological clock.
Just the name.
We just call it MBARI, but it
It's a-
stands for the
oh,
Monterey Bay
it's a name.
Aquarium Research Institute.
Research Institute. Try to
Oh.
translate.
Yeah.
I can't see it in this thing.
Where's Russ? I was showing him. We wanted to see the pre-filters on the.
He's probably getting some lunch.
Oh, okay. He backed down. He missed that whole thing.
Okay, so continue that.
Yeah. Yep. Down and forward.
This is much better for your neck.
Cyclophony all over the place.
what else
And
can we
then
work
something
on?
else there.
Right now, there's plenty to do.
Estamos en el norte de Brasil, enfrente a la costa nordeste de Brasil, pero en aguas internacionales, o sea, en aguas bastante profundas, más o menos de 5.000 metros de profundidad. Pero vamos a bajar nada más que hasta 1.200 porque el objetivo de este proyecto es trabajar con esos animales que viven en la columna del agua desde los 200 metros hasta los 1.200 metros, principalmente invertebrados. Tenemos a bordo también dos investigadores de Brasil que trabajan con peces de profundidades. Son investigadores de la Universidad de San Pablo.
That's super tiny.
Pero los peces
Not
generalmente
super
los
tiny.
vamos a poder ver acá
It's about the same size as
Eh,
the last.
That's okay.
But it's
pasando por las imágenes, pero no pueden ser capturados porque son muy veloces
Look at him
como para
go.
And
capturarlos.
swimming to the top left.
It's a
There it
fritillaria.
goes. And it's gone. Left its
Time
house.
to make a new house. And that house will become a sinker in a nice little carbon packet. Will sink quickly to the deep floor.
Is the iris X-ray view doing
Oh,
alright?
stop. Tomoptris.
Yeah, or it was.
Tiny,
Oh, it might just be
fast,
zoomed in on that.
highly maneuverable.
I still haven't
No, that's a
been
Keating now.
successful at fixing on it.
No, not that.
No, but I had to,
It probably is.
Move the thing around.
Move it,
Go on.
apply.
Oh, we can look at this
Move it
jelly.
again, apply, and
Yeah.
then it's gone.
Yeah. Tomoptrids are
Between that and
too
having to
fast. If they
merge with
get past
HDMI
us, we
on
will
that computer,
probably not find them
somebody's
again
not-
unless they really want us to. This looks like another
It's kind of hard
House
to test the
Araconica.
roles using different monitors.
No, it is not.
But,
It is something else.
Medusa.
And it is super spazzy.
it's a halotria.
Looks more like, I don't know, Botronina. It's a weird shaped bell.
Bottomina?
a tracking medusa, though?
Una linda medusa.
But I don't know which one.
It's an interesting bell shape.
It's an odd bell shape. If it wasn't for that bell shape, I would say it was Halicera. I mean, it probably is Halicera, but different species than the last one.
Botronema has a bulge like that too, right? But
Usually
it's
more of a
narrower.
knob. This could be Bigelowi, Halicera bigelowi. I don't know.
From the can.
It's definitely not Conica.
What do we think it is?
It's not Halicreus. It's definitely,
What do you think it is?
it's definitely a Halicera. Possibly Bigelowi.
Let's see what the deep sea guy.
And that is only because the last one we got was Conica, and it didn't look like this.
Oh, Bigelowi has a similar, or
Pretty close, huh?
dome
Están
shape.
discutiendo cuál podría ser la especie, el género. Al género llegaron, pero a la especie no.
Yeah?
Halicera bigelowi.
Woohoo! Every once in a while, I can ID a jelly. Not often. I think we don't want to
Oh, you know,
scan
okay.
this. We could suck
Oh,
it up.
okay.
Running?
Yeah.
Okay.
No, not scanning.
Okay.
You guys, we don't want to scan this, do we? I
I
think
don't
we
have
will.
one of these.
No?
Can I do it real quick?
Yeah, sure.
Essentially put it.
We do want to scan it.
Okay.
Just kidding.
Thought you were going to get off
I'm
easy.
sure you are loving all of this right now.
We'll do a quick scan
Oh, yeah.
and then we'll suck it up. We would like to include it in the reference library.
Van a escanearla primero y después van a
Alright.
tratar de succionarla para poder colectar ese organismo. Porque es una especie que es conocida
Ah,
para
next.
ellos,
Oh.
ellos la conocen, pero no es muy
I'm
común.
zooming in. Darn it. Don't move. To the right. And probably high.
Times it to 0.
Oh no, so I didn't get time code. Good eye.
Welcome through. That's too far.
You got it. It's pretty small. Oh, that was nice.
One more time, one more time. It's in the DeepPIV camera.
You notice how she said it twice? That means 2 more times.
It's in the DeepPIV camera.
Nice. And then back.
First, and let me get one more, and then we can move on.
Yeah. Oh, that was
I
beautiful.
think we got it.
All right.
Okay.
Cool.
Now, can we suck it up?
Thank you.
Bueno, ya fue escaneada. Los investigadores que desarrollaron del grupo que desarrolló esos escáneres
I'm
están
gonna
acá
write
a
it down
bordo.
so it's in the
Son
pocket.
del
That's that
Monterey
off.
Bay Aquarium de Estados Unidos.
Look at that. Perfect scan of
Son
that
unos
novel.
equipos que no estaban antes puestos en este robot y los están testeando durante esta
You know what?
expedición
You should
y
leave that
producen unas
laser
imágenes
on.
perfectas de
Really
los
helps.
animales.
It's super helpful for
Además de
this.
también
Yes. Come back on
obtener
the,
algunos
a
otros
little bit back on the,
parámetros como los
And also
movimientos y
retractable.
flujos de agua alrededor de
You want to line
los
that laser up
organismos.
right in front of it?
a little bit more.
Yeah. So one of the most challenging things for the pilots when they're flying is looking at a bunch of 2D pictures, trying to figure out in 3D where things
La
are.
medusa es chiquitita
And so the
y
more
vamos a
cameras
tratar
they
de
have looking in different
succionarla
directions, the
con
better.
ese cono que es un aspirador.
You want me to leave it on when you do corrections?
Yeah.
Okay.
Oh, we have a very nice room for you. You can just relax. Sit back. Oh, that is super helpful to have the laser. Comes outside. All right, it's getting tired now. Oh, just inside. Oh.
Hello.
How's it going, team?
Well, we did not see vampire squid yet.
Neither did I.
But we have been told by our chief scientist that we can stop on them on our radar.
I appreciate that. I think that's
You know.
smart.
Looking out for you, just
Thank you. Good stuff.
Of course, we were speeding through water. It's much too shallow for them, so the likelihood of
Yeah,
seeing
we're
it was
good.
even smaller.
We're
but
good now.
Now we're
Under
good.
600, we're good.
Yeah,
Did you get lunch, my dear?
I did not.
Want me to cover? I got you. My meeting is done.
I mean, I might as well wait my half an hour.
Are you sure?
Yeah. Thank you, though.
Make sure. I'll be back in half an hour.
Alrighty.
nice.
Do my
See you in a bit.
student grades. Grading.
Come on, little buddy. I thought he was getting tired. Clearly, it's not. It's interesting how it still has a few tentacles that it's trailing. Most of them are tucked up nice and tight. Yeah. Go. Go. Nice.
Get going forward.
Excellent catch. Okay.
We collected that.
Bueno, la cuestión es tener paciencia. Ahora parece que entró.
All right.
I missed all of that. Did that get collected
I
in
think
the
that's
deep?
what I like.
No.
I think
In section 6.
But we have already something.
We can pass this guy up. That is the same that we just did.
Okay, Sarah. Your turn.
So this one was.
already have something.
Oh. No, we had info.
Ah,
There is.
entró. La pueden ver adentro del frasco número 6.
Nice. Oh, it's bigger than it looks.
Wow,
It's good.
Look at you guys. Killing it.
Some downstream.
Una vez que entra el organismo adentro del frasco, se gira para que se tape. Y ahora el próximo frasco que está abierto para recibir animales es el número 7.
I can't spell in my head. That probably doesn't help. Wait, you got me great. Just spell it. Take the whole space.
Just realized we're on cruise, so that's fine.
Great.
Focus.
Sorry, I can't spell in my head.
Should I turn on laser
This is one of those dyslexia things where it just doesn't. So if you tell me, oh, but you get that, and you spell it J-U-L-A, I'm like, like just like visualizing the letters in my head. Yeah. which is also why it's easier for me to read Spanish than to listen to Spanish, because I see it and it makes sense. And,
Seems fuzzy.
this one. Those two streaky bits totally drive me crazy in there. But now it's very start to the right here. Do you guys want water today? You want water? Same as yesterday? Same as yesterday. Okay. So, we're gonna say 400.
Where would we from? Cafe? 200?
Yeah. And so then this would be surface.
We didn't take so much time to look at it. That's a guess.
Almost like it's
50, 50, and
Well, I could be
like
wrong.
650. Yeah,
That's what I think
like this.
Mm-hmm.
Yep. Okay. This is what we want. Look at what we get. Okay. Hopefully, no matter who's here, they will see that and get us what we need.
We're gonna leave it there.
Alrighty, let's find something. One of those red gut democracy would be awesome, because it is definitely a new species. So far, the 2 specimens we've gotten have 3 characters the same that are unique from the other 2 species that we've described, or are in the process of describing.
Go more forward, or how's this?
This is good. This is good, because we also want to be, we do want to work our way down to 1200. We're not terribly particular whether we get there or not, but just want to make sure we hunt for deep animals and we hunt for shallow animals.
I like that. I like that.
So they know.
So let's look at this guy. Odd little jelly. Might be Paraphyllina again.
I have no idea what that was.
We'll see when we get a close up.
Yep.
It's like Paraphyllina to me. We got one of these the other day.
What probability would you get?
Very low.
Paraphyllina is a
It's
skyphil.
embarrassing because I love these andrews.
Skyphil medusa. Yeah, and then para is P-E-R. I-P-H. Y-L-I-N-A. Might be 2 L's.
It's pretty.
Probably 2 L's.
What is it?
Paraphyllina.
Bring it on.
What is it?
Atorella.
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 Totally wrong.
Okay.
Yeah.
I think the tips of the tentacles are these.
The capitate tips, yeah. Do they have a red fluorescence or something on them? Are we able to? No, it's just the bleeding of the video. Oh. those gonads not being circular, but being bean shaped.
Yeah. Do you want to scan this, Kaikani? We definitely want to sequence it, so
Yeah.
we'll suck it up.
It's super. Is it really small though?
It's not terribly small.
Let's do it, Sarah.
Let's do it.
Oh, we want to scan some tag. Yeah, we do. I'm gonna start by zooming in. If you're
Bueno,
okay
parece
with
que
that.
esta es una
When
agua
they're
viva,
stationary,
una medusa
those
del género
6 tentacles
Atorella.
are out horizontally around the animal.
Yep, they
As you
were.
slowly
Oh my god, what is?
relaxes again. You might do that.
Alright, so zooming in. It's a good size. It's gonna be fun. I'm recording, so
Tenemos acá a bordo y está acá en la sala el profesor Dougal Lindsay,
It's frost.
que es
to
especialista
the right.
en gelatinosos. Él es uno de los principales acá a bordo y que trabaja con este grupo y es taxónomo, entonces él sabe reconocer prácticamente todos los organismos gelatinosos que vemos en pantalla.
pull down. Pull down. To the right. Pull up. Pull it down. Pull to the right. Pull up. It's brought in its tentacles. That would have been nice to get in the model. Might be a little high. Oh, there it is.
Right.
To the right, a little low. It's back in. Oop, a little low. Back in. A little low, a little high.
Right.
Okay, okay. Okay,
That was good.
that's not bad. Yeah, I think if we could do one more series and then we're probably
So
fine.
locate down below.
It's so small. Ooh. One more time, one more time. Little high. Little high. There you go. A little far. A little to the right. Oh, a little high.
Refreshed.
You had a shower.
And shower.
Yeah, 'cause I couldn't tell it was
Clean
you.
line,
A
song
little
in
bit
my heart.
slower.
It's a t-shirt today.
New t-shirt. Yeah.
Yeah, algebra.
And a larvation in the G sampler.
Coming back into Ascot.
Oh, that's a new shirt.
Oh, just outside, Sarah. To the right. Oh, go straight. Looking good. A little high. Oh. Still a little high. Still a little high.
Oh, that was actually pretty good. I think might have just missed the top of his bell, but all the important bits were there.
Well, we might have enough to do the scan. So if you want to collect,
Okay,
I'd rather we do that than leave.
let's suck them up. We can do one, maybe one more.
One.
if you want, and then we'll suck them
Good measure.
There it is.
Yeah.
There it is. It's looking good.
It's going to go right.
It's going up. Oh no. There it is.
Things are okay here.
up. Okay, there it is. Oh, nice. All right. Call that one good.
I think we got a couple good scans.
All right, so let's suck them up.
All right.
Thank you.
It's just out.
Do you want me to leave
past
the
the
laser
end
on
of the
for you?
boom.
Yeah, leave the laser
Center
on.
mid-left.
Okay.
Yeah.
Is that a
We can
glare or
extend
is that an
this
alpha?
port valve 7.
No, not the little light one, farther. Yeah, right here. It's like a chill.
How do I spell this one? La 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 la.
Oh, it's a little red side. Dip it up to our left.
There we go.
There we go.
Species.
Nice.
That was really good.
Yep.
Nice,
Put it a
Sarah.
little bit.
I'm getting better.
Nice scan.
Oh.
It's a pretty little red side. Dip it.
There's a red.
Yep. I have to get this guy in the bucket, get him some shadowgraph pictures on his way in.
Nice.
Good.
7. Atorvella.
Right, just going for it.
Okay.
Haven't seen it hit the jar yet.
No, he's got a ways to go.
Yeah, that was
No.
Oh.
He'll make it. Still coming. Still coming.
Russ, I need a photo of your shirt.
I'm sure it went down.
It definitely went down. It'll come. Unless the hose is disconnected again, but it shouldn't be. It's on the shadow
Oh, no,
cam.
it went in like there was suction. I know.
It definitely
That
went
will be a
in.
fear.
100%. There it is. All
All
right.
right, section's off.
That
Let's
took a
settle.
long time.
Yeah.
Where are we going? Number 8.
Yep, to 8.
All right. It's settled.
Perfect.
Yay.
Woohoo!
Good job. Good job.
Excellent. Excellent scans. Action link collection.
Bueno, una
straight
medusa
ahead.
más
What is that?
colectada.
Straight
Ahora
ahead. The
pasamos,
mung?
entonces cerraron la
No.
botella
Fun
número
looking
7 y ahora
thing?
estamos
Yeah, the
con la
one
número
looking
8,
thing, it's definitely
esperando
moving.
It
al
definitely
próximo
moved.
ejemplar que quieran colectar.
Oh,
That is a squid.
it is a squid.
Straight ahead of me?
Heather, you're welcome.
Looks a lot like Octopus or it's eating something.
Oh,
Hmm.
awesome.
Atola?
Oh
No, it's a
no.
squid.
It's a squid, yeah.
Wow. Let's get some really good shots.
Really nice.
Oh, it's a squid. We can go on.
I think that
Where's Heather?
Heather. Can we see the
Look at that
eyes?
posture.
Might want to iris down just a little bit. These guys are pretty reflective.
Oh, yeah, you heard my feet. Say you heard my feet, my friend.
Estaban todos esperando a que llegue Heather, que es una investigadora de Estados Unidos, que es especialista en cefalópodos,
Really
o sea, el
big
grupo al
fins
que
as long as
pertenecen estos calamares.
the body, I think. Yeah,
Los
like
calamares
an
son
octopus
muy
is.
difíciles de capturar porque son muy rápidos, pero la idea es sacar bastantes buenas fotos e imágenes de ese ejemplar.
The Stigoteuthis.
Yep. I think.
And it does not like our bright lights.
No, he's not gonna like
It's tiny. Look at it
blinking.
relative to.
Changing color.
Is that a typical thing to be able to iris? Scan it over their eye?
They don't have it over there.
Because it looked like it went. No,
No, he's open.
He just
See
moved.
his eyeballs open.
He just moved.
There should be a
things
photo
around.
for. I would love to see the bottom side of him, but I don't think he's gonna
Just
agree
like
with
when
me.
somebody sees a
Probably
bird
not.
nest.
Yay! How deep are we? 800.
800.
Everybody shows up.
The Stigoteuthis.
I think my tentative. I could, if there's a photophore on the bottom side of his eye, that would help my cause. They're pretty close to chiroteuthis in the
Really?
families. Yeah, those families are close.
No one ever shows up when I say there's a nice
Mhm.
Russ, we showed up last night.
Oh.
I just don't hear you inside the car.
I guess I just
Yeah, this is a
don't
mess.
advertise it enough, yeah.
There's a couple of species options.
Totally you, Russ. Totally you.
It's not. What was that? Mastigo?
It seems pretty.
It's not the subject matter.
Yeah, it's-
It's all me, I know. Larvacean house is kind of snappy.
Larvacean house.
Oh, I might be
Yeah.
wrong.
So.
Everybody has some time.
Yeah, there's photophores under there. The mastigos don't have it on the mantle like this guy does.
Oh, look at those.
Isn't it okay, wait. No, mastigo's out the door. Bye-bye.
This one's like, that's my better
Not Mastigoteuthis.
Probably
Acá tenemos a la investigadora
A
be a long time
non-toothed
before it can
family.
see anything.
Heather
It's a toothus.
It's
de la Universidad de South Florida y está haciendo
That was
la identificación del
Can you
organismo.
tell me if those tentacles have hooks
Enseguida les
or suckers
contamos.
on them?
They look
can
like
zoom
they have
in.
There
hooks.
was
The
this really
tips.
funny exchange where there was a
When we had more of a
squid
profile of
deploying
the
its
tentacles, they have
tentacles.
hooks.
and
They
then
look like hooks?
Yeah,
Because
when we had
most
more
of us
of a
were
profile.
engineers on that.
Okay.
None of us knew what we were looking at. And so I think it was Yost who yells, "It's a
So
toothus!"
Okay, I understand that is not an easy thing to do.
Did we rotate the container ultimately before we got to start?
What's that? They're very far apart, those
Yeah, that's
dots,
what I'm
but very
saying. They're
regular.
not- And there's photophores around his eye.
Yeah.
It's not a strawberry squid.
Can we back off of it a little
It
bit?
is not
There we go. No, no,
an
not the vehicle.
anoploteuthid, but
Sorry,
those
zoom out
fins
is
are
what I meant.
absolutely wrong. Siphonophore doesn't have it on the mantle like that. The firefly squid do, but the fins are wrong.
What does the salinitude look like?
Not that.
Not
The,
like
the fins are little at the top, and there's a photophore at the top of the tip of the mantle in the Siphonophore, and his eyes photophores only have
Oh, now you're
5.
serious. Zoom back.
Yeah, we're not stopping on that.
Been there, done that.
I didn't guess.
Ancistro? What about Ancistro cheerus?
Which one?
Can somebody look up that one real quick? Ancistro cheerus.
Just curious. That's a
Ancestrochurus.
Ancestro.
A-N-C.
H. Yep. C H.
Oh, zoom out. Zoom out.
Yep. CHERIUS. Something like that.
At some point, he's going to be tired of us.
No, it's not a histio. The mantle's too big and the fins are wrong. I have Ancestrochurus.
Yeah,
I forgot my
you
phone
missed
in my mad dash. I'm trying to play with her eye.
He's pretty fast.
There we go.
No longer.
He has photophores like that.
Explaining the right stuff.
A-N-C-I-S-T-R-O-C-H.
I can see him down there.
E-I-R-U-S.
Angel squid, angel club
Coming up.
squid.
You'll have to
Doesn't look like
Bring the
it's
camera
like-
down.
No.
Yeah.
Not enough.
trying to
And zoom out.
I know it's a bit
Not
big,
an
but it
Onycho
does feel
toothpick.
like you should try.
Come down.
Oh, look at that.
What about a
Wishful thinking.
disco toothpaste? Cyclo toothpaste? They don't have photophores. Nope.
I like that name,
Nope,
Disco
no, no.
Toothpaste.
I don't, can we suck them up? I don't think we're gonna get them.
Probably not.
Probably not, but you get one shot at it.
Or the jar.
Oh, he's got a wrist lock behavior there.
Is it
If he will
Some
hold
kind of
still, you
pirate
could
ship.
D him.
If we could desample him, but I don't know if he's
No,
gonna.
you don't want
You'd
to
have
try.
one shot. If you touch him, he's
Yeah, no,
gone.
I understand. I think we've got good
But it's
video.
worth a try.
I would love the
Good try.
ventral side, but I don't think he's gonna give us more.
I mean,
We can follow it for another minute or so, if possible.
Try the
Oh, you're
DL.
like, I am right over
Oh,
the
yeah, I was.
sampler.
Heather está diciendo que necesita verle la parte ventral para tener más seguridad de la identificación del calamar.
And when it does,
He's like, no way. I don't like that talking about putting me in these.
Pero el organismo no está colaborando, no se está dando vuelta para que le pueda ver la parte de abajo, la parte ventral del.
he's out.
Okay, that's okay. We got video footage. I'm gonna go get an idea.
That was a huge.
That was fun. Thank you guys. Let me go figure out what
you
his
can
next
pack up
round.
and go
I'm gonna
home
to
now.
my
You're
key.
done for the cruise.
I'm done. No, I'm done by any means. I'm gonna go get. I know exactly. I see the picture in my head.
Amazing
photos. Yeah, of course.
The photophores on
They're
the
beautiful.
front of the mantle.
Very nice time. Beautiful
Right.
Going back to
video.
that, really.
Beautiful animal.
And now everyone leaves.
Thankfully.
They don't have a computer system inside the cold room. That's why I don't hear you.
I try to be more, charismatic and likable. It just does not translate to my animal. I'm not sure I'm supposed to take that personally.
I don't know. Every time I see you, Russ, I think of a Larvacean. It's just something about you.
Right association, all right.
At least he didn't say it. He thinks of mucus.
You're not the one sharing a room with him.
Wait, what? A sinker? No, it's a good. Yeah.
Come
Sinker.
No, that's a.
Gotta be some other big things around
Was
if
it?
there's
It looked
a
like a
sinker like that.
Hmm?
That looked like a sinker to me.
It's a house. It's an abandoned house. Yeah, maybe.
Bottom left, Tomopterid maybe? Coming up to the center, it is a Tomopterid.
Well, you think you could call it a tomopter, but we'll have a look.
Anything you call a tomopter.
He's not wrong.
He's not.
Little blue one?
This is the one with little baubles on the tips of its acicular series.
Oh yeah, you can see that. Oh, that's you're right.
Oh wow, look at those baby jellyfish inside its body cavity.
They do have parasitic pagantha in there sometimes.
You are trying to get. Wow. Tomopteris. T-O-M-O-P-T.
Alright. You're definitely going to want to collect.
T-O-M-O.
Yes,
And then
Tomopteris.
Suction, most likely.
T-O-M-O-P.
They are pretty wily.
T-E. I know. N-A.
He's up. Yep, there we go.
E-S-A. A worm.
I'm coming out.
It reminds me of my physics professor. He used to say,
Put your hand around the fire. Got it. The good news is I was correct. If only we could spell to find it correctly on the internet.
I don't know where the.
What is it?
What was it?
It's Ancestrocheirus. It's this guy.
Really?
All right.
This guy right here. Look at him. He has those fins. He has that mantle photophore. He has hooks on his tentacles.
Is your camera
Wouldn't be
way
the
over
first
to the
time
right?
Jill was wrong.
Yeah. Okay.
Bill's got control.
Okay.
Yeah.
Zoom way out.
Lesser eye.
That's what I have.
Yeah.
None of the photos.
It's not.
It's just
Well, we have better
random.
photos.
That's
probably
not
random.
unusual.
Right. It fits the description. So
Random.
yeah,
I've never
it's,
even heard of that guy. So
It is this species, Ancestrocheirus.
Let's see what the deep sea is.
So with the laser.
Ancestro.
Pretty sure I've never seen one before.
No, wait.
Had you seen
That was
one
the first
before?
time
No,
I
I've
seen
not.
it.
Do it, buddy. Don't do
Not
it.
bad means.
Hey, Kakani, can we have the laser?
CIS.
You want the laser
I
off?
may have seen
Yeah.
it on Rice once, but I
What are
don't
we
think I've seen
collecting
it a lot.
now?
Yeah.
The worm?
Come on.
E-I.
It's running.
Nope. No. E-I-U-R-S. E-I-U-R-S.
I have it. But I don't have any photos.
There's no photos. We don't collect them often. They're pretty rare.
I don't have any.
I have
deep sea
probably
guys.
two specimens from all the DeepPIV cruises.
Wow.
Yeah. And this was a what, 800-ish?
Yeah.
Come
Outside.
I can do
You probably want pretty strong suction.
Yeah, Ancestrocheirus lesseri. Yay, good one, you guys. Thanks.
What did they come on?
That's a great question.
And Ancestrocheirus.
The sharper Enoch. Sharpier Enoch.
Just rolls right off the tongue.
I like Ancestrocheirus.
We should ask the chat what common name they think we should.
Right? Come up with a common name, that would be good.
Yay, Sarah. Midwater worms do rock.
I'm just gonna go put my computer up and I'll be back.
All right.
The Judd Squid.
Nope. That Ancestrocheirus is good.
Come on, buddy. You gotta be getting tired.
Bueno, ahí les pusieron el nombre científico del calamar que estábamos viendo en pantalla.
It's flying.
La especialista
They're not
Heather
easy to keep
Judkins lo identificó.
Actually, the reason we started studying their swimming is because we were embarrassed that they could outrun us. They shouldn't really be able to swim very well, but they do.
That was a really beautiful scene. It's even more beautiful than it is to see.
Alright. Slowing suction down a little. And
Excellent.
just moving forward, but I'm still leaving
Yeah,
some
I would leave it
suction
on a bit because he will swim out. Super, thank you. You can turn the laser off.
Well done, well done.
Very, very nice catch. Looking at the buckets.
I'd like to think I contributed in a small way.
There.
There he is.
Shut the pump
Nice.
down.
Okay, off. Yeah. Oh,
Bring on screen at the top.
he is on the screen, yeah. That'll be okay. Just want him to come down.
Suction off, but still pushing forward.
Actually, if you go backwards a little bit, well, I don't know
Or
if
if we
this
just
sampler, if it, yeah, just hold still. Just want to get him. There he comes.
There he is, coming
There he
off the
comes.
screen.
Coming off slowly. Still trying to get away. Let him figure out a
There he
different
goes,
direction.
he's down.
All right,
Yeah,
go ahead
rotate
and
it.
index to 1.
Yay.
Woohoo! Awesome.
Well done, well done.
Thank you so much. Super.
The old ship, that was the other.
Bring all the worms. Squid.
Bring on the worms.
And houses.
That's what we're doing today. All the funny things. Okay, and we're doing good. We have 4 buckets filled and 1 desampler. I'm gonna go eat some lunch.
Sí, Silvia tiene un nombre de ese tipo. El nombre común Más o menos eso.
Look at this variation. There's one of those in the D for you already, Russ. Did you hear that? I got one of these in the D for you. The animal is there.
You
The
do?
house is still intact. Yeah. It was a beautiful collection. Oh, not this one. It was a different one. No, it was the one that. Yeah, yeah. We want to look at this. Sorry,
Regular
I was talking to
Hunter
Russ.
with the clubby, clubby wings.
The one we got was not this species. It was a different species, like a fritillarid with the pretty things. Gekani, you want another scan of this? I don't know how
Is this clubby or
big
no?
it is. It's bumpy, yeah.
Oh, maybe.
Just a nice
Clubby.
bumpy
Yes,
one.
oh my god, yes. Let's do this.
Okay. Let's scan it.
All right. I am ready. I might zoom in a little when you're set up. Just to the right. Here. I think it's scratched. Do you see it right in the center here? Terrible.
Things with carrots. Jellies we haven't yet sampled. No, we're still headed down. We're hunting
Oh, between the 7 and 12.
7 and 12.
And then, do these guys want any water? Yeah, that's what, do you guys want any water?
They do. It's written.
It's already written
down.
down.
Well, it's written down where we want it. There's a long dot.
Oh, I think
I like that.
it's way low, yeah.
That'll
So-
work.
I thought that you were worried about my
I am worried.
presentation.
I am worried. It's all important.
Yeah, it's nice for
Warm.
It's really
Bird
good.
in
Get
the
in there.
deep with
inside.
Yeah,
Alright.
it's the only really great to sample
was
these.
way too
I
fast.
know, there's no point in section.
Yeah.
I like that.
Do you see it in my
Well,
camera?
It'll be easier, but you won't get an animal.
Well, or your big little bail, and then you can't get the
I'm
animal.
gonna zoom in
Should be there.
whenever you're comfortable.
By chance.
Hello. Oops. A little fast, but that motion outside of that was great. I'm gonna zoom in.
Yep.
So. All right.
Can you scroll this down? Scroll that down.
To the right.
Oh, sorry.
That's got our.
Did you add the name?
Yeah.
It is
Just to
17.
save you.
17.
A little bit high.
Thank you.
Ew. So weird. Stubby toes. Hello. Coming in hot. But it got in there. Just a little high.
looking good. It's inside the laser. Sorry, don't want to reverse the vehicle. 'Cause it's getting really close. Uh-oh. Okay.
I was disintegrating.
Not an easy target.
Not at
For
all.
sure. I may have gotten a scan that I can use. So that was, do we know what that is? Do we know what that is? Microflora.
It's a Nicoplura day. Didn't really, the host didn't look completely smooth, but I didn't really get a good zoom in on it, so I have to see the reconstruction.
Yeah.
And we've used half the buckets and one DCM. Back in action.
Back in there.
All right. Hey everybody, this is Heather Judkins. I have taken over for Karen Osborne for the next couple hours. We are going to continue moving down towards our 1,200 meter mark, looking for targets that we want to investigate. I think that was a cyclotony. Yep. Heating nav. Kakani, you want the lasers on still? DeepPIV?
I'll turn it off.
Okay, it's up to you.
But I know it helps, I guess, with the collection. So just
Yeah,
let
it's
me
up to
know.
you, whatever you want. Okay.
It does seem to help him.
Does it?
get that
I mean,
lower.
the-
Trying to suction, yeah.
That's good.
Does it give you a field of light? Nice. I didn't know how far I went over. I think that was a little copepod. Yeah, we're good. Yeah. He was already on the camera pretty much. Oh yeah, middle. Oh, is that a glare? That's a glare or something.
Yeah, I'll give that a go. Does look like a bullseye.
It does. That's why I was like, wait, that doesn't look right.
Super bullseye now.
That's the most stealthy organism out there. It's not moving at all.
No, this is on that last, yeah, and on that last one, center here, I don't know if it's, it just needs to be cleaned or scuffed. I think it's just got a little inch,
Middle. Thanks. Google, anything in your world? What is this? Is this a piece of a tentacle or something? Oh, Google's not in here. I think we just get a
That
good
looks like-
video on this and then we can move on. What do you think?
That looks like mung.
Yeah, that's what I'm thinking too. Okay, great. Thank you guys. We can move on.
Like just
Yeah.
mucus.
Sinking
that's
down.
sinking.
Mm-hmm.
It could have been an old house, a mucus house.
And it just got in concrete at the end.
Yeah.
Yep. Great. Thanks, you guys.
A mung.
One word for it. Copepod.
Oi pessoal do Brasil, da Argentina, sou o Marcelo, sou da USP e agora cheguei agora aqui, começou meu turno, estou ajudando aqui tanto na transmissão quanto a registrar
Middle?
os dados.
It's a fish or Ketignath. It's a Ketignath.
Okay. You going?
A gente
Yeah,
está
we're
vendo
good.
agora um ketognato aqui,
Doubling as a fish today.
parecia um peixinho a princípio, mas é um
Yep.
ketognato.
Stand on this. Stay on this. This is Medusa.
Agora apareceu uma medusa bem bonita.
If we can get some zoom and some good video.
Fazer um zoom nela.
Kakani, any ideas on this Medusa?
No.
Yep, it's about where I'm at. I know Dougal always wants to get the top of the bell if possible, the connection points where the tentacles meet. Helps him get the IDs. I think we've seen this before. Yeah, this is the one that has the ends of those tentacles have those white tips. He mentioned that the other day. But I don't remember what group it went into. Yeah, he's
É
close.
medusa ou Siphonophore?
Just give him a
I
minute.
think
Yeah.
it's a medusa, but it is weird. Like we're getting some refractive effect.
That's that
to it.
refracting light.
Flashing.
That's why I'd really love to get a nice zoom on
Yeah.
the bell. I haven't
Yeah,
quite.
we're working on it. He
Okay.
was super close to the camera. Yeah. Bell is the goal. Super zoom if possible. Yeah. Oh, stop moving, little Medusa. He's down going. Almost, almost. Oh, come back, you little thing. Oh, no, a little bit. There he is. Yeah. He's going off my screen.
It's like coming straight at the
Oh, I
screen.
see him.
Holy
Yeah.
moly.
Don't move. Don't move. Oh, he's still in there.
He's down below.
Yep.
He was right on the D jar.
Oh, yeah.
A gente estava tentando entender melhor, chegar um pouco mais
Here's come
próximo
back in.
para conseguir identificação melhor. Aparentemente é um estilo medusa.
Yeah. Yep.
And the camera.
Oh, that was a squid, I think, that just went by. But we're sticking with the jelly. Oh, I just lost him. No, no, that was something else. That was a lube 18-4.
Work. Yeah.
Okay.
Essa saiu do nosso campo de visão agora. Vamos continuar a busca para as
Oh,
outras
yeah,
coisas.
there he is.
Slide the
Nice.
arrow.
Ela tá voltando ali. É realmente difícil conseguir localizar essa espécie.
Like I don't even know where the camera is looking
Yeah.
right now.
Well, we had it a second ago. Yeah, there we go. He was still going off that way. I think he went over to the right. And down. I think we're okay. I mean, we gave it a go, and he had a couple in the very beginning, so we'll continue on. Oh, wait, nope. That's a little. That little bait. Okay, cool. Let's keep going. Thanks.
Agora a gente vai continuar aqui a jornada. Né? Nesse momento estamos o ROV Subastion está a 810 metros de profundidade.
Can we get the light a little lower on the science camera? A little bit? Iris close or something? Seems pretty bright. Something needs to go down. Middle, going up to the top. Yeah, thanks. So we want the middle. It's another medusa.
Agora tem mais uma medusinha ali no meio. Logo atrás tem outro, parece um ketognato.
Same as before, if possible, if we could get the gonads at the top of the bell, and if we could get the tentacles. It's pretty small, so.
It's got the little process on the head.
What it looks like. Yeah.
It's a siphon medusa as well.
That's a great question. It's a medusa.
Okay. We are fine.
We are fine. We've gotten it. Somebody's on Squid, right? Yeah.
Hmm.
Do you know which kind, Russ?
No.
Me either. So I was like, okay. See if we can zoom in on this little guy. He seems pretty active. You're not using autofocus. Never seems to work. Right, autofocus never seems to work. Yeah, to get it close, yeah. Yep. I don't have it in my screen anymore. Maybe? There he is.
There he is.
Yep.
I'm gonna try to bring it in. Go. Like that.
Yeah. Yep. Down off my screen. Now he's back in.
Oh,
Back
was
there.
Doesn't look great.
almost in focus. That's different. Very short.
It's like, nope.
Yeah, I think I saw the process on the head.
On the head, I think so too.
It's like a little like glantha.
Yeah. Apparently not. he was listening. Yeah. Almost. Oh, so close.
A equipe
So
agora tá
close.
tentando colocar a medusa
Oh,
em foco
maybe?
pra achar alguma característica que seja
Let's just try
diagnosticável.
for one more try kind of thing, see how it goes. He is small. No, I know, there's swirls. Got him there. There is a process on the head. On the bell. He's got 1, 2, 3, 4. It's like 8.
Good. Okay. No.
Keep coming down.
No.
It's like, yeah, right. Nice try. This little guy's like, don't worry. Okay. Mm-hmm. It's all right. We're giving you guys a challenge today. You're like, you're doing that every day. Okay. I think we're good. I think we've got enough that Dougal could probably tell by when it was a white dot in the background. So I'm going to go, we're good. So we can back zoom and then head down again, please. Thanks, you guys.
A gente vai seguir a nossa jornada agora.
Uh-huh. It's a lot.
They're probably like how you put
You look
mascara
focused.
on, you're like
No, it does not look like that. You look focused, but you do not look. I'll take a picture the next time, so you won't know.
oh no, I never wanna
It's not bad, I promise.
get problem if I'm like
Nope, didn't hear anything. I think you're good.
there's something.
Mm-hmm. It was one of those. It was a rhizaria.
So that little one was attracted to Medusa. That we were looking
Oh.
at a few minutes
This
ago.
guy right here, he might be super close though. Sorry, you guys. It looks like it's just a sinking house. I don't see an animal in it. But you want a laser.
Well, just in the future, because they look. I don't know. Sarah, were you there earlier this morning? They are very transparent. So the next time we see one of these
From
right
far
now,
away.
yeah, it's
Yep,
just
perfect.
Very slow.
Yep, okay, good call.
Sorry, it's my fault.
No, that's
Looking
okay.
away.
So far, we haven't seen any of them like this, when they're diffuse like this at abandoned structure.
Yeah, this
Yeah.
is the second one we've seen this morning.
Okay, thanks you guys. Sorry about that.
No worries.
Então isso a gente não sabe exatamente o que é. Pode ser algum organismo já morto,
Yeah, I saw it. I was like, oh.
né, que estava
Like, he's
afundando
close already.
e se aproximou muito rápido aqui do hog e acabou
Yeah, that'd be
se
great. As
desmantelando.
far as possible. But as
Alguma outra coisa que é mais frágil. Então a gente está agora
The house is around the animal, but last time when we did the scan, right, we couldn't see anything until the laser was on it.
What do you got here, my friends?
I'll see open maybe, or I'll see. Can you zoom in on it?
A tentacle end? Or a jelly tentacle end?
Moving like it garage.
Well, it's in the wash a
I
little
think it's
bit.
an alciopid.
That's what I was
Alciopid.
wondering, if it was an LCO.
Oh, the little guy. So that white at the top is his head?
Might be.
Yep. If we can zoom in. Yep, exactly.
Oh, it's not moving like I would expect an alciopid.
How do you spell it?
Okay, I don't think
No,
it
this
is.
looks like a lure of something.
Yeah,
Looks like a bioluminescent lure at the bottom.
because the alciopid you would see like little legs all along
Yeah.
the length. I'm not seeing.
I don't agree.
É, a gente
Yeah, I
acha
don't
que
think
pode
it's...
ser um tentáculo abandonado, uma coisa assim.
So let's just get a good
Yeah, I don't
couple
know
of
what
frame
that
grabs on it, and then we'll keep going.
It's not the lens, okay. I don't think it is. No. unidentified object.
Uh-huh. Other? We have an other
Táxi não
category.
identificado.
Super long.
I have something there.
Não, realmente não dá pra gente identificar o que que é. Deve ser algum pedaço de organismo que abandonado, como um
Crustacean.
tentáculo, alguma coisa assim. Pequeno
not
crustáceozinho ali.
Okay. Okay, I think we're good. We can keep going. Oh, there you go. Yep. Uh-huh. Rusty. No. Okay. I think we're good. Yeah, we can keep going. We've got some good images of this. We'll see. Unidentified underwater organism. That's what I'm going with. Heather's official. This is what you get when I get
Tem
up
mais um
here.
camarãozinho
It's your
aqui.
I think it's very clear.
Thanks. Thanks, you guys. Pilot changeover.
E agora tá na tropa aqui da equipe.
Nice job, team. Nice job, team. We'll see how these two do.
They're gonna
I'm only kidding. You guys have been amazing.
You expect to get invited back.
Don't cry inside. Oh, I'll get it right back, I'm sure. I have no doubt.
Yeah, he did.
Oh. You know you're
Proud
more than
of
welcome to
something
turn it
there.
right back around.
Yeah, all of them are moving. What is that?
That's just a
It's
glare.
a crowd of something.
Yeah.
Yeah,
So we're
it's,
moving forward and down
reflection on the
towards
glass.
1200. Would be great. If we go a little forward, that way we're going into it a little bit.
O objetivo de hoje é chegar até 1.200 metros de profundidade. Nesse momento o ROV está a 820 metros de profundidade. Um pouquinho mais fundo.
Stop. Sky. Yep. This is a Fisonect. I can give you that much. Yep. This is a Siphonophore. We're going to be zooming in on.
Siphonophore. Esse é um organismo colonial, dá para ver
Sí,
vários
es
membros
un
da colônia.
sifónóforo y ahora le van a dar un
Apolimia,
zoom
maybe?
para ver si lo pueden
It looks like one of
identificar.
these
É uma
saw the other
apolínea.
day. Let's get some zoom and let's take a look. Yep.
Oh.
Oh yeah, that looks like bulimia.
Oh yeah.
El género es apolimia. Ya lo habían identificado en otras de las bajadas del
Are you doing it?
Apolínea.
I was gonna say,
Do a quick scan.
yeah, of course. DeepPIV, yeah,
Yes, please.
of course.
Although, I would be curious about iris.
Whichever you want, my friend. We could do both.
Wow.
You tell us what you need.
Polônia. Is that right? Spread?
Polima.
M-A, okay. Apolima. M?
Usual for the lights there for you?
Yeah, I'm sorry.
Yep. Remember, you guys turn off the lights right as it gets to the sheets, and if we could get them somewhere in this little box is better, so we get it full screen for them and super slow, and you can go back and forth in the same.
Call it when you're ready for light source.
And then I'm gonna zoom in. Cause you know, that's more fun.
We like a challenge.
What or what?
Yeah. A little, right, and he moved, it wasn't you guys, but if she can get a solid plane of the animal not moving direction, but that's gonna be near
You might
impossible.
want your legs on the
He's right there, we've got him. Oh, this could be lovely. Nice and slow, if possible.
And he's out.
Yep, he's not in.
Autobranchia is.
Oh, he's back in.
There he goes.
Yeah, the autobronchial type, the branchia talks about.
Still too fast?
Gills,
Okay.
so.
He's moving off my screen, as long as you've got him.
Their type of bones.
If you go to a little to the left.
Their type of sea squirt. Or not sea squirt, sorry,
Yeah,
sea
yeah.
slug.
Oh, but now he's moving so fast.
Some of them are pelagic.
No, not really. There are other gastropods that are pelagic, but none of the nudibranchs.
Just this one is the only pelagic.
These are this is not strictly planktonic. It's newstonic. It lives right in the ocean surface, and it feeds on floating jellyfish, like Portuguese man of war or the
He's toying with you now.
Jelly, animals that live in that. And one of the reasons that these are so heavily pigmentous is like sunscreen.
Come on, buddy.
Right? So
Come on,
they
buddy.
have a very, very high ultraviolet environment.
Oh.
And these colors are like a sunscreen. They're designed to block the light and break the light down, reflect the light. Very iridescent. They're such an odd exception. I think they call them sea dragons. I think is the
Yes. Yes, correct. I want to see. Blue dragon.
when they harvest the skin themselves.
So that they
Yeah, so like the front one
Lower?
is to try to get
A little bit lower. Grab and chase this guy.
Uh-huh. He's definitely moving.
Oh, just a little high. A little high.
Oh,
Right.
maybe.
To the left. Oh.
He's toying with you now.
I don't know who's toying with who. Almost. Let's get one.
Por lo menos tenemos la imagen de ellos en 3D y con otras informaciones como el flujo de agua alrededor de ellos, que los ayuda a entender también cómo son los movimientos de los organismos. Por más que se los capture, esto ofrece una imagen del organismo en su medio natural. Entonces tiene bastantes ventajas contar con esta tecnología a bordo del submarino.
Please choose turn.
And high. Not too high. Ever so slightly too high. So close.
Want to try one more, or want to try Iris?
Let's do one more.
Okay.
Dá pra ter ideia aqui da dificuldade de fazer isso, e da paciência do pessoal operador de ROV, né, em seguir as instruções que a gente passa aqui para eles.
That was looking good.
I know, I didn't want to say anything. I'm not saying a word.
Ele precisa entrar exatamente no foco do equipamento.
No! That was just to the right.
E se ele estiver exatamente no foco, não adianta.
So high. To the right, I'm not sure. It wasn't in the camera.
Right. It's looking beautiful way out on the right.
No.
He knows. He knows he doesn't want to be on camera. Oh, maybe.
Right.
No, I think he's coming that way. Oh, I thought he was.
No.
Still to the right.
Just teasing you.
All right. Should we try Iris?
Yeah, let's try Iris.
Can we just
Yet
put
another
a box,
challenge.
A tape native box around where we.
We've tried all sorts of things. Do you want to do Iris or?
Sure. These guys are excited. I'm excited. Let's give it a shot.
All right.
A gente vai tentar todo o equipamento agora, que é o Iris. Está realmente muito difícil colocar o bicho no foco, mas...
All right, we're going to try one other imaging technique with this Siphonophore, and then we'll move ahead, right? We don't need to collect this one? I don't think so, right?
I don't think so.
It doesn't, I don't think it's new. So.
Yeah, he hasn't left me with the impression that this is something new. Well, in fact, aside from the fact, this group has probably got a lot of undescribed stuff in it.
Right.
He's not been excited about it the same way as Smoke does.
Right.
Hey, Kakani, are we leaving the arm right there or are we moving it?
Wait, we haven't put it in position yet, so
Okay.
my suggestion is to keep
You
it
can
on
take this side.
the port side, but position it.
Take this side.
It's kind of up to you too, you know, where it's too fast, right?
I think it needs to be straight, at least straight. The
Straight and probably
DeepPIV.
in line.
Five?
Yeah. Like, yeah. Right, Kakani?
Yeah.
Perfect.
Just change some
So something like that?
Yeah, that'd be great. Thanks.
Locked.
looking good.
All right, let's get him in those crosshairs of all those beams. That's the goal.
That's the aiming camera. Where's their camera?
This one,
Oh,
right?
you guys have to send me that, right? 'Cause that's-
Esse é um outro equipamento chamado Iris, também equipamento de escaneamento.
That's DeepPIV on the
Só que ele usa vários
Yeah, we
peixes
used to
para
have
fazer
iris.
isso.
What's the number, Kakani? We could. Okay, wall C.
Esse é um dos equipamentos que trazidos pela equipe do Embari, do Monterey Bay Aquarium, para
These were
escanear
the
os
questions I had
organismos.
last night too, Kakani, because we tried this at like 3 a.m.
That should be the mud camera.
Someone's changed
the
it
skid
since last
camera.
night, I can tell you that.
Right,
So y'all
something.
were fussing with
I don't know who was, but it didn't. We couldn't make it look right back here, as far as I was concerned, yesterday. Last night, the early one this morning.
I just remember it, no,
Yesterday, I had Kakani's here so we could see the color, and then we had something like that.
do you want me to do it? I think I can do it.
Well, yeah, but if we wanted to change every time we push that button, I'll have to do that. But yeah, you can do it now, and then I'll catch up to it on my commands. But yeah, someone removed camera last night. It looks like.
That's maybe why we couldn't get it. I was like, I know it's here. I saw it. But yeah, that's it, my friend. You're getting them in there. Almost. Yeah. He's pretty close to line up, Kakani, when you're ready. Okay. Oh yeah, see it. That's the key. Yep.
It should be the skid cameras.
This camera on the right is gonna, when he comes into view,
This one.
yeah, you're gonna want, right. And when he gets into frame on this one, you want him towards the bottom right-hand corner, which puts him in the middle of this one.
There he is.
Yeah.
Right. So he's got to get it. He's in there, but he's got to get a little far away. Coming back towards him.
Can you bring me the little closer to us?
Sure. Bridge RV.
Bridge, go ahead.
Eric, you mind speeding up toward this a little bit, just to close that distance by about half?
Yeah, speeding up.
Thanks, sir.
And now if he goes over to the right corner. Why isn't he in the frame? Does he not show up? Is he invisible?
You showed up a minute ago.
Yeah, he did. Sonny had him in the right spot, I think.
It's a little thin. If he's big in this one, you're probably too close. I'll tell you that.
Uh-uh.
Is it like that?
No, he's fine there.
I think it's to the,
Down a little bit.
I don't have a good orientation from where I'm sitting.
Now he's in the camera. Okay, now he's back. Maybe he does need to back up a little. Oh, there he just came in. He just went through, but he's close. That's him. Better. Still too close.
Did you change the hysteresis? Because that doesn't seem to work as well.
Yeah, that's it.
Yeah, it's
But
I did, yes, I changed all the settings to what has worked in the past. Yeah, I don't remember, what did you say you did?
this one, where should
Yeah.
I be in this?
It
Top right?
used to be bottom right yesterday.
It depends how close you are.
Right.
So
Correct.
you gotta kind of judge his size and.
There he is.
Yeah.
He's in. He's in. He's in. Keep him right there. Don't move him.
All
Move him
right.
backwards a little bit. If you could just move him in the water.
That part's important most.
He was a pretty blue for a second.
Maybe it did. Okay, at least the manual is working.
Starting to catch up. I'm not gonna be
Too close into the cameras where he is right now.
Yeah, one more of those, because it was, you know, in the right volume briefly.
I don't think it got its. Did it get the whole thing? Do you think? It didn't look like two separate shapes.
That was close.
Yeah. Close. Should be right in there somewhere. There he is. There he is. Coming back in.
A little
Connie, what do you
closer-
think?
A little bit closer to us.
Not coming in.
A little lower.
Yep. Oh, you just had his. Sorry.
Generally imported with parameters.
Oh.
Cameras aren't really going up. Oh.
I know there's a Tomopterus. Can you just note Tomopterus?
Okay, I think I have one more shot of this.
Okay. Up a little.
The lights are 30%.
Ooh, that might be. There he is. Almost. A little.
A little bit lower.
Oh.
A little closer to us. What? Yes.
Down just a little. There he is. A little too close.
All right. Thank you very much.
You're good with this?
Yeah.
Nicely done. See if he goes back one more time.
Cool. Thank you very much.
Okay. All right. Thank you guys. Take a breath. Shake your arms out. Work your shoulders. Thank you guys. That is not easy.
Yes, it's
And then if we get a full out, I don't know if we are. We might already be. Okay, thanks. That'll work. Perfect. You can keep them in a smidge. Yeah. All right. Down and forward, please. Can we drop the light on the science camera again? Why is this so bright?
The iris.
Okay, if we can close the iris, that would be great. Close some.
Could you close the iris on the
Science camera. Yeah, it's too bright.
Oh,
vamos seguindo aqui a nossa
All right.
descida.
Now that might be a little too far. Give us maybe one bump up more bright. Russ, you good with this? See you
It's
in
not
one.
here, but yes.
I like this. Okay, good. Cyclothony. Hi. Marcos has joined us. Look at that. There you go.
Cyclothony.
Marcos, there's your fish.
Yes.
You're very excited about it.
Thank you, guys.
Wake up.
Oh, sorry. Marcos is freshly woken up. We might need to give him a minute.
It's already begun.
It's okay. You're allowed to be.
I heard you saying my
Well,
name.
I
in
thought you were
vain.
back there. I didn't turn
In
my.
vain it was.
No, it was not.
It was.
No, it was not.
Except
Not that
it
time.
summoned you. So
It summoned me. I was kind of listening. I was trying to make notes for Cheryl so that she could read my horrid handwriting.
Feel free. I was just
correct
making
my
sure the light
typos.
was good because you and I have about the same desire in this light color.
Jelly?
Top right.
Is it or is it a kitty?
It's
cat?
it's a medusa, about 1 centimeter big. I don't know yet. I'm just might be.
I was gonna.
Uh,
Is it a
I don't
squid,
know.
Or are we coming up on a?
A sinker. We've had a couple sinkers today.
We
That.
called it everything we could.
Yeah,
so
please
far.
do.
fish. Yeah.
Oh, it was a fish.
It's a fish. Yeah, I think it's a fish.
Sarah Vomer? Is that one that hangs up like that?
I don't think.
I don't know if it was.
Seem very-
They're long and silver is what I remember.
Yeah.
Okay, we can keep going. Thanks, you guys.
Actinopterygii. It's about as far as
Curiosity.
Okay.
I'll wait till you stand next to me. The letters more good.
Marcos es uno de los dos investigadores de la Universidad de San Paulo que está a bordo. Son brasileros.
So in the copepods.
Marcos Dos Reis, que está haciendo doctorado en la Universidad de San Paulo, y el orientador de él, que es el profesor Marcelo Melo.
E a gente acabou de ver um peixinho agora, né? Mas não dá pra saber o que que é. Pode ser um cerrivômer, que é um tipo de enguia. Mas se moveu muito rápido da gente e não conseguimos identificar.
Middle? Yeah, please. Oh, Russ, you know this one. We've seen this a couple times already.
Oh yeah, it's the.
That
I think
angular
it's Chinophias. Is what it's been-
Siphonophore. It's one of the Siphonophores, yeah.
Or Clausophis. It's a Physonectus.
It's a Physonix?
Yeah.
Oh.
Oh,
I was-
isn't this spelled
We've
copper?
seen them a
it's
couple of times.
It's a Calicopper. Sorry.
Okay.
Oh, I think it's up purpose.
Sorry about that.
That's okay.
My
This is
brain's
one we
still
talked about
waking
looking
up.
like
I'm
a
only one
rocket the
coffee
other
into
day.
the morning.
No worries.
Lo que estamos viendo en pantalla es un tenóforo.
I think with it as broad as what it is, it's going to be Chinophias.
If we get some good video and some zoom like this, we can.
Can you spell it?
Are you talking about the
Ross?
Venom dashboard?
Can you spell it?
Chun is named after Chun, but Dougal's pronunciation of it is weird. Not what I would expect. He has a bunch of things he pronounces very differently from
C-A-G-U-N?
Chunophy.
That's great. That's great.
Yes, something like that.
Yeah, nothing shows up.
Do you know which way I need to go up or down?
Chun or Chun. Not showing up in
That's
the search.
great.
No.
It should.
That's great, yeah. We get this for a little bit. As long as he'll be agreeable, then we can move on.
See if I can find it.
Nicely done.
C-H-U-N-O-P-H.
Okay. We get one more. While he's hanging out.
Achuni, en vez de O-I.
Perfect.
Ah.
En el chat lo están diciendo.
Maybe it's different vowel there.
Sin O, sin O. Ahí.
Ah, okay.
Okay. We are good. Yeah. Thank you guys. Moving on.
Someone on the
Got it.
chat.
Yeah, got it. Yeah.
Chen instead of Chun?
Chummy.
If it's showing it's a Siphonophore, it seems right. Chun, yeah.
A little less on the iris would be great. Yep, please. Thanks. Middle? Thanks, Sonny. You're seeing it before I am. It'll be a Ketignath. Lovely Ketignath. We can keep going. Thank you.
They're just trying to look innocuous, so we leave them alone.
And we are. We have been collecting them in the nets, so that's been good.
They collect okay in nets. They're abundant out there.
Yep.
Oh,
So you
Sol
got-
Mandela,
Yep.
bipenteculata, or is it an angina?
I-
2 tentacles or 4?
I think angina has the 2.
It's 2?
Wow.
These are 2.
Sol Mandela, bipenteculata.
Sol
It's a
Mandela
narcomedusa?
bipenteculata.
It's
Bi-
another
oh,
one of these
bipenteculata.
species complexes where the defining feature is a 2-tentacle narcomedusae, and there's more morphologic variation
We
that
can zoom
Dougal
on
sees,
down,
but
if that's
none that
possible.
is in
Yeah,
the keys.
that's great, you guys.
Wow.
This is great. Almost looks like teeth.
What is the genus name?
Hmm?
What is the genus?
It's Sol Mandela bipenteculata. It's a narco.
Yep, but no.
It's actually
No,
nice,
this is great.
nice
Yep.
pictures, nice footage of it.
Yeah, I was gonna say this is great. Yeah, we're good.
Gina.
Cool, cool. I think we're good, you guys. You got some great footage. We're good.
Ooh, look at them.
Thanks. Heading down and forward, please. A little less light on the camera. Thanks again.
How much did the bulky ritual far
Mm-mm,
from
we're good.
CMS? Nothing?
It's just because it's a totally different platform. It didn't help that for a little while the CMS
Bottom right.
built it
Ah,
up a little bit.
the Ketignath.
That's obsolete. So
Yep,
part of the-
Ketignath.
Mais
Love you.
Ketognado agora
Keep
passando.
going. Thank you for doing your predatory
Esse é o
things
tipo
in the
de
midwater.
órgãos que a gente pega
The
bastante na
Ketignaths or
rede.
arrowworms
Então,
are
no
aggressive
hobby a gente está mais interessado em
carnivores,
registrar do
and
que
they
em
eat
coletar mesmo.
copepods extremely fast in terms of how
It's
quick they
not
can
like-
eat.
It's view,
These two
VUE.
Yeah, View 3. Actually, we're running Quasar, which is also based on View, but not quite.
Fish.
So,
This just feels clunky.
just funky.
But it might just be the inner.
I think this is a development tool, not a production tool. For, I know, but for not as integrated in.
Bless you.
They, like, it was kind of explicitly not what they set out to do.
Right.
I don't know.
And again.
I disagree. with
That's a
that.
waste of time.
It's like they weren't worrying about like integrating it with
Fish. We
Não,
try it.
peixinho ali.
No, it's Tomopteris.
No.
Ah,
It's the worm.
Tomopteris.
Yep, let's go for him.
Uma poliqueta.
Tomopteris. Yeah, he's gonna give you some challenge. Are we still collecting Tomopteris?
I suspect we are.
Probably yes, right?
We gotta try and give Karen something to do with her free time, because she seems lost.
Oh yeah, and this guy's a different color, I think.
this.
Yeah, these guys are great.
So this would be the third one of these if we get it.
Okay. We're
which
gonna
is
get some
kind of
good
the
video
magic
right
number
here.
for
You guys are doing
sequencing a
amazing
stream.
on this clip. Great. And then we're going to collect it as well.
Oh, it's, guess who shook up?
I know. I just felt I had asked
Essa é uma
the
espécie de
crowd.
poliqueta
But I was trying to
e
get team
é o
input.
terceiro que a gente vê e vai tentar coletá-lo novamente.
You know.
It's good. This is the new species.
This is the new one, right?
Yeah. See those white
Aqui a
bands
bordo
on the
tem
tail?
a professora,
Yes.
a doutora Karen Osborne,
Ooh,
que é
that's it.
Karen
That's
Osborne,
You can
do
count
Smithsonian,
those quickly
e ela é
flipping
especialista nesse
segments.
grupo.
There should be 23.
E veio correndo aqui super empolgado.
Ooh, there's something in its
Testing and there was simply no time.
Or maybe it's just our reflection. It just seems a little different in that spot. It's
Right.
a bit off.
Oh, you're right. Right there.
Can we get us? Oh yeah, look at that head.
You want to zoom on ahead if possible?
If we can zoom on the head, although I saw what I needed.
Yep.
The platform.
I would rather catch him than get a better view
You're
of
right.
the head.
So we're gonna go into number
Current
one.
species described from the
Nice. Love it. Oh, he's close.
Done already.
Don't lose him. Let's collect him.
Oh, he's gonna try
He's
out
gonna
to
get,
us.
yep, he's trying to get in that wash. Stay with him. I've got him.
Did he just
Yep.
lose his tail?
No,
Or
they
contract it?
contract them very quickly.
He's got it back out. He's going up off my screen. He's off my screen still. He's way off my screen. You've got him. I don't have him yet.
It's cumbersome.
Where is he?
On
He's
the
still
top,
above? Okay. You keep on him. I don't have him yet.
vai voltar agora. Passou.
They're extremely fast and maneuverable.
Yep.
Sorry. I apologize.
There he is in my screen, middle. Over to the right. He's caught in this wash though too. Yeah. Feel free to suction when available.
He's not gonna relax.
I would look to the right.
Oh, little buddy, I was gonna make you a butterfly.
Okay. I don't see him. Down, low. Oh, I don't see him either. Anybody got an eye on him? Yep, he escaped. He's a lucky individual for the moment. All right.
You see the next one.
Yep. Yep.
And I'm thinking that collecting little jellies like that, like Solmandella,
Yeah, and I feel like
even if we know what they are and we think they're the same species,
should
unless
collect.
somebody has sequenced them from off Brazil, which
Okay.
generally they haven't
Okay.
done, that we should probably collect them.
Right, I wasn't sure if we had already collected them, that's why I didn't
I don't
go with
think,
that
I
one.
haven't seen any Solmandella yet.
Come in. Okay, perfect.
So like, yeah,
Yeah.
even jellies that are not used in science
Yeah.
are new to that species.
Yeah.
Eu não tô vendo o chat.
Sounds good. On it.
That's because these platforms.
Marcos consegue dali, né?
Yeah.
Ver o chat.
Things we've collected so
That
far.
would be lovely.
That would be a useful thing. We can have Sam out with
A gente anda na troca agora, a professora Silvina acabou de sair da
Little
sala.
reference. And then these are all the old data sheets that we've already used for the ROV stuff over the past.
E nesse momento o ROV está a 870 metros de profundidade. A gente está continuando nossa jornada
1200.
até chegar em
we can get
1.200
there,
metros.
We will get this little tomopter.
But you also don't want all these, like, so, I'm not sure, like, I
Middle?
understand why. But like, as you measure all these things, I really don't want, right? With functionality.
It's gonna be a Siphonophore.
Unexpected. So.
Yeah, please. Oh,
Siphonophore ali no meio.
sorry. That whole, you used his name.
Space in between the two sides. Is that a thing, or is that just my crazy?
Well, it's a, I wouldn't define that. I always think of it as kind of like the animal.
Yeah, he's small.
Thing was kind of like
A little more if you can. Science team, any IDs on this?
I can show you pictures. That is a apolemia.
Apolemia?
I don't see the tentacle, but the rest
Well,
of
there's little somethings
They're
there.
small.
Like they look floppy, maybe.
Yeah, that'd be
It's how
fine.
angular it is. I don't think of them as that angular, but I also don't look at them that closely.
the problem is just more
Esse é mais um sifonóforo, uma polímia.
small Mars. But I don't see the tentacle.
Could also just be
Yeah, no, that's fine.
It could
No,
be
that was
very
okay.
inspired by how
Where it was good.
with that few nectar verification.
É esse organismo.
Yeah, but there's actually a nice picture in
That's great.
Midwater concept.
Any chance for a
Esse é
little
um
more
organismo
zoom?
colonial,
Or is that
né?
gonna
Então
throw you
isso é
guys off?
uma parte da colônia, pode ser um indivíduo muito
I
jovem,
guess not.
someplace else.
Thank you. That's
uma
perfect.
colônia muito jovem ainda que não está completamente
Oh, then it
formada.
goes too far. Yep. That was my bad. Sorry.
Of the house on the first area. So I've got it in my lecture notes.
Yep, I got you.
I can send it to you. It's
We can
not
just
a
get one more good look at this. This has been squiddled, right?
What
And the
kind
bar's
of
got a fair number that are tagged correctly as fiddle area. So that's an easy one.
Yeah,
where you're
that's
talking
great.
two different animals.
So we've only done this.
Okay. All right. Nice job, you guys. Yep, we're good. We can move on.
Even for
Thank you.
stuff that we know what it is, if we want to collect what they get from the screws. I added it to the reference library
Yeah.
that we should collect, so that's why I collected the heliceras. Because then we don't have to do anything in processing them, right?
Good.
We image them,
Exactly.
use tissue sample them, so like things like Sulfentella, you know, like representatives of everything,
We're
and I'll
good.
have Sam.
No, we should stop.
Oh, you did?
Yeah.
It's small.
I haven't seen it on the
but
list, but I
it's-
haven't looked at all the lists.
Yeah, I've got to get, I'm typing mine up now, so because
Oh, you don't have to.
well, I spell things so wrong. I'm just doing it so I can keep track
Okay,
of it
okay.
for
Sam
Cheryl.
will be very happy about this. So,
If you guys hear this, the one down there.
So, I mean, even with the,
Good luck, my friend.
these Larvaceans, even if they're
Team
not
members?
something
Exec,
That
perhaps?
is a Rhizaria.
This is a raised
I
area.
mean, my
It's a
assumption
colony.
with that Tenopterus was that it's the same one we have in
Yep.
California. There's 2 species in California. It's a different species here. So it's
Mhm.
like,
Yeah.
looks the same. It's not the
Well,
same.
even for Rhizaries, even if I thought I could put an ID on them, I mean. it would be
Okay.
something
If we get some video and zoom real quick, then we should be
Yeah.
good.
insurance in another
Well, it's,
ocean.
right, exactly. It's a
But
different
I just
location
feel
that has
it's
it.
kind of, what worries me is we're so bad with the kind of like news that I'm hopeful the document.
The camera is pretty good now that I printed one of the light traps. So now we have
Good.
decent background.
Okay.
We don't have decent lighting, so we can't zoom all the way in like a 65 millimeter, but you can go probably 4x.
Weird things happen.
Pretty decent.
I mean, it's
You have
better,
to
better
play
than
with
nothing.
those
I guess the question then becomes, what do you do in
Okay.
a situation like that if you can't photo
I
document?
think we're
so-
good. I think we're good, you guys. We can keep moving on.
I mean, I was able to with the
preserve
Thank you,
a
Tenopterus.
thank
specimen?
you.
To me, it would be why preserve specimens and try to rely on genetics. If we can't say what it is.
Well, you should be able to do
Well, what it
Depending on
means,
what it
what it means is you need to cut the trunk, the tail, and the taxonomic feature is often in the tail. So
Nope,
which
we're
side
good.
do you on a Larvacean?
Heating now.
Right, so you need both
It's in the
halves.
tails.
Yeah, let me figure out all that.
See, they need some sacrificial
No, I
blades
can't
like
even
that.
find any ROIs.
They have some, if they're big enough to cut that pretty
What
enough,
other
it's okay, just don't do it for
should I
6.
search?
Yeah.
Whatever you do, don't cut them
Well, I
for
mean,
6.
even on almost all the crustaceans, there's enough different
There's
blades
some
as they
legs,
get
right? They
bigger.
have 2 sides. Thank goodness for biomodel but some of the jellies, right? You're gonna destroy them with each other. No, but I can show you what we have set up over there. It's not ideal, but it's a hell of a lot better than everything else we have.
Well, my,
We're good.
you know,
Heating now.
my preference on
Yeah,
this,
that's
unless I
every
decide
time.
that it's a described species, like it's being developed.
House.
would be to put it away in Kevin's description. I think that's the first thing was the king and our. If it's a known species, then not.
If it's a known species, and we have another southern Atlantic, that's
DeepPIV.
useful, although they do let the Europeans do the
This comes from
Atlantic meridial
wherever
transect.
this is.
They don't teach you all
Yeah,
of this stuff,
the
but it's
shallow.
all toes.
Yeah,
Right.
the shallow stuff. I mean, is theoretically a ship.
Do they not
Dry
go
nut
very
stuff?
deep? Just the
Well,
way.
it's kind of. I'm not sure what they use for their station samples. I'd be very surprised
Where are those focus
if anybody
records?
had been interested in things like
They still just
is not put down.
Yeah.
But I've never gone looking for the data, so I don't know.
Well, I mean, on the groups that I work on,
Figure it out.
they've done some stuff, but not nearly as much as I really expected.
It's so dependent on having either an expert or a person with interest in any of the groups, right?
So I've been doing all the things they would have gotten in nets, right? Getting even the stuff we say is we know and stuff, and getting
Middle?
the sequence for
Browsing this
That's
will
the
hit your
way.
and give you
And
an
that-
idea.
that is-
Ooh.
that is one of the deliverables.
What is that?
We want this.
Siphonophore, probably.
Yeah,
No.
community that,
No.
isn't it? Shrimp?
A shrimp. Or you, or
If we
mice.
could, if we could zoom on this,
Now, if I could tell those things apart on video, I would be
the
happy.
crustacean.
Pode encontrar agora um camarão aqui.
Just with the string though.
We're calling
They're mostly mice. Yeah. I know, which is why I'm like, it's a shrimp-like thing.
With only maybe one long antenna instead of
But
two.
those at least are being done, like Heather Rack and Kristen is doing tons of.
Yeah, that's an antenna for
decapods,
sure.
So I'm not as worried about
Great.
them.
I always think.
Yeah,
of
he's got,
mice
with it,
as
sometimes they
got a
have
cute
too short
little
and too long.
helium
So
there.
yeah, that, you're absolutely correct there.
But a lot of them don't are well developed.
Yeah.
So
See if we can zoom in on him again when you
There's
get a
supposed to be
chance.
this, you know, exposed,
He's
frankly,
going
there's just not, I can't see them. I don't know what is meant to be, but like on video, I can't tell.
Yeah, the statuses can be really hard to see on some of them. And you know whether or not the
Parece que uma das antenas está quebrada, então a gente só vê uma delas.
I mean, at least the Phoenix original works really well. It's cute, but all
I
the
mean,
others-
it definitely amazes me the amount of things that Douglas synthesizes in the images because these I would still
I know
want to
some
put this
peeps.
under the scope, even though I reasonably view the tails, it certainly has got statuses.
That is
Although
an
that
impressive
structure
tail
of
that
the
it
tail
has.
tends to be more moist-like than
Yeah.
a decapod with a really long telson in it.
Marcella, you have squiddled this, yes?
Sorry, say it again?
You have squiddled this?
Yes.
Great.
You get a closer picture though. Take a capture now.
Ooh, look at that
Ooh,
back.
yes, that
Look
is a
at
mysid.
that.
A
That is
mysid.
cool. I think we should
So it's a mysid
collect
shrimp.
that.
If we can, that'd be great. He hasn't
Yeah.
been moving much,
He
so
hasn't
maybe
been
he's just
moving.
happy being
He'll
here.
probably wait till he touches the side of the D sampler. Or that
He's
laser.
sleeping.
Do you want me to turn? do you want me
Yes,
to
that'd be
turn
great if we
After
could
a
collect
hard day
them.
of eating
If we could, you want the DeepPIV off?
I could.
I think maybe.
Doing
Their eyes are pretty sensitive.
we doing detritus or we doing suction?
Suction.
We've already blinded them. No
It
worries.
is the first available jar, which
Something
seems to be
about
number
hybrids. You
one.
can't blind them.
Thank you.
You can blast them with ridiculous amounts
Então a gente vai
of
tentar
light.
coletar
They
esse
pop
aí.
back within a few minutes.
É um bicho bem diferente.
It's crazy.
Oh, if
Because we did not expect
Mesmo o
that, but it
pessoal que está aqui a
certainly made our
bordo não
lives
sabe
easier.
exatamente o que está pendurado nele.
I've got a couple of amphipods in my ground.
Ele vai tentar.
Great.
There's a little
Like
call number
separated
tab
out for
on
me.
it.
I
A
can-
princípio eu achei que fosse uma antena, mas e a outra tivesse quebrada, mas
I don't see.
aparentemente não.
I'll pull them out. I need to.
Got to the
They're like the
right.
little wee bean
She will
shaped
tire.
things. At least one's a male, which means they
Upright
must
lower.
be pretty small.
I've lost
as
them.
adults.
Yep.
Below.
There's a lot
Oh,
of really
here he is.
small
He's
ones.
lower now in the front.
And they all tend to be in the like pair a day, which all look the same, which is not so nice, but that's all right.
Oh, that's funny. It woke up.
Oh hey, look at that. It does have a little to live.
So I should have a key for the mysids and that crest.
Yeah,
Might-
that seems pretty distinctive.
Yeah, might be
He's
an
out of the
optoid
woods.
at this point for genes.
Esse provavelmente é um parasita que tá segurando nele, né, que tá preso nele. Agora a gente acabou de... it's gone. Ainda mais sincero.
I wouldn't spend a huge amount of time chasing this thing.
Yep, yep.
Yeah, it's gonna be fairly mobile.
The shrimp lost that long fin.
Because
I don't see him.
those were, I'm guessing they were antennae on.
Maybe?
Yeah.
Him without his antenna.
Yeah.
It was this morning, but you could
But there might be something coating the antennae.
Yeah, that's him.
Ele perdeu aquele apêndice, lá se for uma antena, um parasita, mas a gente ainda assim vai tentar coletar o indivíduo.
I just, if you do not want to do not enough time, then just let us know, because then we won't put you on the schedule, or we'll put you on the schedule for the time that's good. You can decide. I know you have a lot of lab work to do, so.
Oh, close, but no cigar.
Close one. Very close.
Ele passou bem perto ali da parte de sucção. Mas agora ele parou de se mexer um pouco, acho que vai facilitar a vida da gente.
Yeah.
Entrou coletado.
Good job.
Nicely done. Let's see
Você vê esse braço,
arrival
ele
into
chegou
canister
aqui
number
no
1.
canister.
Here he is.
Tá lá.
Nicely done.
Canister
You
fechado,
can index
primeiro
it,
indivíduo
that'd be great.
coletado.
Oh, he's going to the tip. Stand by a second. You got it. He's in. We're good. Thank you guys. That's
See if I
great.
can get a genus on that.
Yeah, and I texted a couple of peeps.
There's a mysid key in Boltowski.
Oh, we've got it. It's Physectocaris microphthalma. It's a Pandelidae.
Pandil?
Yep, my colleague wrote a paper on it.
Pandil, well then that means it's a real shrimp.
It's a shrimpy shrimp, right?
It's a shrimpy shrimp, yeah.
Yep.
Yeah, well, it's actually large shrimp, so I don't know why maybe we should call them something other than shrimps.
It's an oxymoron, isn't it?
It is a little small though to put into a salad. I've not seen one of these in the samples yet.
No, neither have we.
In the nets.
Not in the nets.
And I think probably because the nets haven't
You know
been
what
deep
that
enough.
is. Keep no.
Say it 10 times, not more.
You didn't
Okay,
have-
well, I'll say it's 17 more, I'm sure,
Yeah.
because he gets us every time. Every time. Cool, that was great.
Mais um ketognato aqui. Oh, right in front.
Right front, I think it was a fish, yeah? And he is gone.
Yeah.
I think he was a fish, my friend. I just don't know if it was
Yeah,
a
it's
cyclophony
there.
or something different.
Yeah.
Oh, in the middle?
In the middle.
Yep, yep, I see. Try for that. Yep. It's a good old cyclophony. Thank you. Oh, he's nice and close now. Cool.
É
There's
um
that
peixinho
Rhizarian.
ciclotone.
I don't think we need it. We can get the zoom on this Rhizarian right here.
Ciclotones são os peixes mais abundantes do planeta. Os
We've
vertebrados
already
mais
gotten a
abundantes do
couple of
planeta.
these, right? Russ, this is the one that he's collected, correct?
This is Rhizaria. We've been calling it the sparkly Rhizaria. for lack of a better word.
But I'm pretty sure
he would love one, but we got D-sampler open, they're both tied up.
we have one D-sampler taken, one available, and we have 3 containers left.
This will do best in a
D-sampler?
D-sampler.
Yeah, let's try it.
It's very fragile.
Yep,
Como a
agreed.
risária é muito
Okay,
frágil.
if you guys
Vai
can
tentar
put this in the
coletar ele.
D-sampler, that'd be great.
Só que esse é um organismo muito frágil, então não vai ser coletado por sucção. Vai ser coletado nesses...
So all the sparkly things you're seeing are actually glass rods
Yep.
that are secreted by the animal, so it's really...
Vai ser coletado nesse tipo de canister que é uma coleta mais fina.
But it should be 5. You could,
4 or 5?
Test if you need.
yeah, 4 or 5. Yeah.
Yeah.
Okay. You ready? Going for it. As soon as I see him go in there, I'll do it, all right?
Yeah.
And we'll zoom, let me know.
He's in front of it, just down. Out front again.
As risárias pertencem ao grupo chamado cromista, que antigamente era agrupado nos protozoários, que a gente não usa mais esse termo, um grupo que não é monofilético, não compartilha um ancestral em comum. Mas eles pertencem aos cromistas, que é um grupo bem abundante de oceânico.
In front again. I don't see him. Now he's back out. I have it. Oh, maybe. Yeah.
Okay, close.
Yep. Close.
Close.
Close now. Yeah, go. He's still in. Yeah.
No,
That is the slowest close. We need to work on that close if possible. Nicely done, gentlemen. Thank you.
Fechou,
We got
tá lá
him, we
dentro
got
agora.
him. Nice.
Eu só fico um pouco
thank
ansioso,
you.
que a tampa fechou bem devagar.
Thank you. Sweet. Let's go back to scanning mode and going deeper. We're almost at 900 meters of a potentially 1,200 meter depth today. We are in the water until...
Until we come up.
I do have the schedule. I will look at it right now.
I will make a separate, can I make a separate chat just for
I
the
think they
schedule?
adjusted it. Yesterday.
It's in general right now is where it's
I
been
know,
going.
just- I was gonna make another one, a new group for the schedule. 'cause
Yeah.
It's hard to find.
É mais um
It
camarão aqui.
is-
Another one of the shrimps.
Yeah, it's not the same one though.
Can I do a quick zoom now?
Yeah, if you could do a zoom, that'd
Of
be
course
great.
it's not. It's one's
Oh, he's
captured.
great.
É um camarão muito bonito, dá pra ver os apêndices se movendo ali.
We know this one. We've collected this in the Gulf. We being the scientific community, not me.
All right, Heather, I made a schedule channel in
Os
our
parapódios ali se mexendo.
WhatsApp.
Okay. And I'm gonna find it in a second. I was just taking a pic of the shrimp.
So
This is great, you guys. This imaging is great.
Does it have eyes? Does Karen want it?
It does have eyes. Okay. The schedule is
Small swimming legs or walking legs.
I don't have that schedule. I don't have the ship schedule. Oh wait, here it is.
Do you want this one?
Should we collect this one? I haven't heard about crustaceans. Let's just we only have 3 containers. Let's
Well, how much time have we got left?
that's what we were just trying to figure out. 3 hours? Ish? It says 1830 is ROV recovery.
Shrimp will be friendly with others, right? The problem is it's not a squid.
That's not a problem. I love all deep sea animals. He's great. I do.
Tá.
Oh,
a pouco ele-
Cystolabis.
Is that the full zoom now?
Assim?
Yeah, he's pretty
Kind of-
stable.
If you could, that'd be
Oh.
great.
Just focus.
Que bonitinho.
Here, this one? You think?
Yeah, looks fine. Put him in the other one.
Put him with the other crustacean? You don't think the other guy's gonna come out? Are you sure?
That's fine.
Put him draw in the water, the flown in water, he can't swim out there.
You don't think so?
Try sucking him up, but once he gets excited, he's gonna have a lot of velocity.
Yeah, he's gonna zoom away.
You kinda got one shot at it.
Right.
And then I think he's gonna be too hard to chase things.
Sunny's suggesting that we might
That's
want to
my
put him
ass.
in with the last mycet or the last
You know,
shrimp we
you
just
probably
got.
could.
ELL.
Should we try that?
E again?
LL.
Yeah.
Oh well.
So that was
Send
back
you-
to canister
send you a
1.
sample, they're pretty robust.
Yeah.
Right.
Let's go back to canister 1.
Oh, sorry. I was, okay. ASIS.
Can I see it?
ASPIS.
Are you guys collecting this?
Yeah. It's okay.
Yeah?
Yeah,
How do you feel about that?
I like it.
Okay. We're
the-
doing it.
Only one,
What is it?
I don't know.
Oh,
Bring it in.
It's a crustacean. We're very convinced
it's a
crustacean.
shrimp. I'll get you to shrimp.
Is it cystolapsis?
That's what I wrote down for my friend, and I just sent her pics just to see.
Yeah,
I
send
never
send
see
another
these things
picture.
alive.
Can we get an ID on it too?
Yeah, I did send a picture.
No,
All
I
right.
don't know.
Good.
That's just
And little guy in there is not
closest thing I
coming
see
out.
in the deep sea guide.
Okay. Second he feels this, he's gonna freak.
So you're looking nice and rested.
We're at 900 meters.
Ah, a gente tá vai,
Yeah.
decidindo se vai coletar isso aí ou não.
Wait, you have strong suction on?
People were giving
Earlier
out
today with the worms, we had 0 suction.
It's like good, like a
Yeah.
game commercial.
He's gonna feel it with those antennae though.
Mano.
And then as he gets
Vai
close, suck
tentar
it up like
colocar ele
a
agora
selfie
no
mode.
sucção.
It's also translated, right? So. You see the little translation thing, it's auto-translate.
Oh, he's trying to get away.
But she's been.
Sylvina might be able to do it as well. He's going off my screen. You guys still have him?
Yeah.
Okay.
Yeah, host and a Siphonophore.
He's going back here out of view. Here he comes back. Freak when he touches them. I think you get tired. Going out.
For replacing it yesterday.
On.
For
Oh. Close though, close. Right concept. Yeah. No. Dang it. He was going to the right.
Diz que ele aumenta, mas não para peixe, camarão e
essas coisas.
You're like, come on, go to your home. You don't think the full suction would help either, right? I don't think so. Off my screen. Coming back in.
Ó, agora a tentativa de coletar um animal com um suction sampler, mas é um camarão de águas profundas. E esses animais.
What do you think? Do we try the full to start?
Mais difícil de coletar.
It's as easy.
Eles sentem o fluxo.
He went over right. She's off screen for sure.
E imediatamente eles.
He's gone for them to swirl. Okay. All right, nice try, gentlemen. We're gonna try this again the next time.
Okay.
He will not get away. We got great video and the zoom was amazing. So we can definitely see what this guy is.
Are we gonna move the canister?
Yes, let's move the canister back to number 2, because that guy's empty. Yep. Perfect. Thank you, guys. Great. Forward and down.
Depths we doing Niskins at?
That's a great question. We have Niskins at 1200, 650, 2 at 50, and 2 at the surface, if that works out. We have a want column and a get column. So, see how that goes. Yeah. We're good. Connie, did you find that master schedule? I can't find the daily schedule. I found the ROV schedule we made.
Yeah, what is
right here.
Is that
another?
Is that a spreadsheet somewhere? Because just having that link somewhere
We'll
would
need up in a second.
really, I think.
It's in our shared folder. The wish list schedule. It's the last thing in our shared folder. And I haven't put the actual hours because every time it changes.
You mean this one or?
Yes, I mean this one. This is the one I was looking for.
So currently you've got 1900 on.
Okay.
That doesn't
Great.
tell you what time the vehicle's on the water.
Right, because it changes every time. This one says ROV on the deck at 1900, and then 2130 ROV off the deck.
All right, so I want
But it's
that
short.
spreadsheet.
Can you look at that? That's not a spreadsheet. That's the ship's.
That's the Google Doc.
Nope.
All right, can I get a link to that Google Doc?
That's the one I need to.
I'm gonna put it in our WhatsApp chat. so
Okay.
we can refer to it.
Don't do it. Don't do it.
Or we can prepare.
You know, yes. That would be
Just
great.
type in an email.
It's going in the wrong way for a sinker, so.
Don't want to be added to the change.
Yeah, I just wanted to.
It's back there.
moving.
Yeah. A fish? Can't tell from here.
Right now, it's only view this view. I'll make it.
Okay.
Oh, slick.
Yep, we're good. We're good. We're good. Yeah, I was gonna say. It was something really cool.
I don't know how to do it yet. Well,
I think the captain and Karen are the only ones that have it, or somebody has permission for that one.
okay, as long as it's just view it, yeah. Okay.
We're good.
Hooray. Thank you so much.
You should have a link to it anyway. That's
No, yeah, that's perfect.
We're good.
Hallelujah.
Thank you, Kakani.
Amazing. I'm so excited.
Me too. It's on every monitor everywhere, but I
know,
never
but
can
like-
find it when I need it. We could move forward and down a little bit more, or forward a little bit more, I guess. Thanks. Yeah. Middle?
Alright, Kakani, should I just rename DeepPIV
This guy?
files to have their timestamp in his name?
Might be a fish.
Right, so today's Tuesday, right?
True.
ROV's on deck, 1900.
Esse mouse aqui?
We have an hour, less than an hour.
Actually,
And then the second guy.
1900 to 6 o'clock or 7 o'clock or something.
Okay.
Sorry, my
we do
friend.
not have the right time zones. Sorry, 1900 hour time, which is 4 hours.
Pessoal, então aqui na sala a gente tem a dupla do Ió aqui, eu e o Marcos, e o Renato na gata da FUG.
If the canisters are full, I think, right?
Os 3 brasileiros a bordo aqui.
We're good.
I don't know if she was open to us earlier.
I need.
Okay.
Trying to go.
Thank you, Paul.
Eu
This
tô
is
tentando pensar
so wonderful.
That way. This
Yeah.
guy? Yeah.
Não.
Oceanologia.
Oceanografia. Mas aí, ó, também. Só que aí a galera às vezes erra. Escreve escrito assim. Daí o pessoal
He was like, nope, staying away from you.
7 p.m. and
Marcos.
how do we want to organize
Yeah, fish. That's it?
No, still up there.
Oh, this guy.
Yeah.
Got lost in the white dots.
Right.
Yeah. He's not agreeing. Yeah, I'm okay. We can keep
The
going.
idea was that I would be able to pull through till the end of tonight's time. Which would probably be done.
You mean the next time
You're right. I'm good.
through the whole thing? Well, why don't we take a nap?
You take him now.
I don't know.
See, that's how that works.
I don't know.
It's the same. It's like it's not really oncoming. I tried to sleep through much of this day, and look, this is where I am. But maybe I'll.
Do you have that schedule up easy?
what's that one up at center?
Yeah, it's in our WhatsApp now under the schedule thread.
Where you looking, Russ?
There's one up dead center. It's out.
Yeah, trying.
Oh, I see. I see you. Left, right?
Yeah.
Correct.
Slightly left of center. Yeah.
Yeah,
Out
exactly.
Fairly far.
Yep. That's been the thing. There's been some Sunny's been trying to get, but things are out there and moving away from us.
Well,
This
I'm
guy might
used to
not.
being out there.
E agora o Rob está a 970 metros de profundidade.
some reason it says it's 2024.
No, I'm trying to move forward, but I'm not doing.
Esse é bem próximo do limite entre a região mesopelágica e bate-pelágica, onde a luz solar ela já não existe mais.
Still out there, it's moved a little bit more toward the right.
Esse mar azul que vocês estão vendo, essa luz é provocada, só possível ver por
So
causa
I'm
da
a
iluminação
member, but I don't see a
do
schedule.
Sebastian, do Rob Sebastian.
We'll be pinned.
Swing right a little bit more.
30 days. Right there, Heather. You should be able to see it now.
You're awesome, thank you.
Something there?
Oh, right here?
Middle.
Sinker of some kind.
Looks like it's a big scythe, is it? Or
No, it looks like a sinker to me.
Yeah, it's another sinker with a little.
Yeah, there's a little medusa near him, just below him.
Esse é muco.
Yeah, I don't see any animals in this.
Yeah, I think that's what I was
Yeah,
saying.
it's like a mucus house, and we call them sinkers. There's
Yeah.
lots of words for this, but
Zoom in on the
sinker is
little
one of them.
medusa so we can put a
Yep.
an ID on
If
it.
we can zoom in on that small medusa that just went off screen. Back on screen. If we could get some zoom, we'll get this for Dougal. In terms of video and ID. Thank you.
So little halocreatid.
Halocreatid medusa. If it's possible to get the bell, that would be great. I don't know if that's gonna work with this little guy. He's moving around a lot.
Allotrephes probably. Is that the bigelow one?
Oh yeah, almost.
Ooh.
It was a little Siphonophore, wasn't it?
Yeah, I was gonna say a little Siphonophore beside it.
Família?
We get that.
Alitrefes.
Weirdly, all the video files
Não,
from
não,
the
não.
have creation time.
Quinidária, medusa.
Alicreatidia para baixo.
April
Yeah,
28th,
yep. I realized I was
2024.
like, we're too close.
And a time offset
Ali
about an
trefes
hour and a
aqui,
half
achei.
from the actual UTC time.
Traquimedusa.
Traquimedusa.
Maybe we can use
There
that.
he is. Okay.
But it was my intention to help Emily as well at some point today. One thing we want to talk about is logging.
Okay. I think we had gotten enough. You think we got enough, Russ?
Yep.
Okay.
Good.
Then we're good. We can move on. Forward and down, please.
Definitely take a break. And what do we need to do for like? What do we need to do?
I think we
Nope.
should work on it.
Still not here.
Just kind of, so we have baseline of what we expect from the gym setup and goals and some of the changes. 'Cause, I was immediately swapping lenses around and like, let's get this working for this kid. Because, you know, there's a few, we have some things we can try to set up a little bit. I haven't looked at the software, but the other thing is to, Karen's MO for calibration is a lot of light bars where
Fish.
it's images that are interesting and like we should build that into workflow in a consistent way. Not the way Karen does it, like she'll take whatever image is needed, but
Cyclotron?
she
I
takes
think it
some
was.
reference image and then without touching it, does a clear image so that she knows the last one is always calibrated, even if in between she's changed focus,
Coming up
distances and stuff.
about
And we can
midway,
also
Yeah, I see it.
to the
like
left,
use that
Nice eyes,
for
gentlemen.
other sizes.
kind of
Max.
vertical looking.
Yep, we got you.
Because for now,
Siphonophore, perhaps?
Oh, or we another tomorrow? No, it's a.
No, it's Siphonophore, I believe.
that's a pretty one.
Yeah, he's got great bell.
DeepPIV candidate.
Do you guys want something on this, you guys?
Bem bonito, parecendo. Bem grande também. Dá pra ver como
Okay.
completo.
Ah, dá pra ver
Yeah. Let's get some video.
Os nectóforos ali pulsando.
Zoom if possible. This guy's great.
So I think this is the one he's kind of been calling the white bars.
Okay.
Fisonect.
Yep.
PHI.
So it'd be Bisonect, Primar.
You use that to piece things back together.
PHI, S, O,
Funny,
N.
you know this animal now at this time.
Yeah, he's great.
Fisonectes, achei aqui.
PHI,
So bad to
Beautiful.
Fisonect.
He usually wants to do what? Zoom in on these tentacles and tail usually.
Família.
Yeah, if we can zoom in on that. Great. And then at the top of the bell would be the other option that he looks for. This is great video, you guys. Thank you. Yeah, up at the top of that would be great. Great. Thank you. All right, Kakani and Yost, we're ready if you guys are. DeepPIV, yeah? Yep. All right, DeepPIV.
E agora vai fazer o
He's pretty
escaneamento da
large.
espécie.
Little button to reset the zoom.
Where?
Just to the right.
Zoom reset? That means zoom to the max.
Zoom out.
Agora mais uma dessas cenas de filmes sobre ficção científica, né? Quando o bicho é escaneado, ele passa por esse feixe de laser. E aí parece que ele está em chamas, mas na verdade ele não sente nada, é só um feixe de laser para o escaneamento dele.
Wow. That was beautiful otherwise.
He's just hanging out in there now.
Dá para perceber como esse feixe não afeta nada ele.
Yep.
Ele nem percebe que está sendo escaneado.
He's just taking his time.
Oh, that's too bad.
I know. It's gorgeous, but the wrong way, right?
Yeah, it's out of the field of view.
Yep. So if we could head to the left some for that exact magic to happen again. On the outside still? Yeah. He's too far out still. He's gotta be here-ish. Ooh, sorry.
Alex can smell the scanning of Siphonophores.
Oh, this would be lovely. Now he's too low. Close and low now. Oh. Do it. He's almost done. Now he's out. You can go back the same way if that works. Nope. Okay. Want another pass, Katani?
Yes, please.
Yeah, of course.
But yeah, if the head is pointing towards us, that'll be better. either towards us or laterally or sideways.
Sideways could work.
We just can't take tail first.
Right. Sideways is okay. Head is okay. Butt first? No. Tail first? No.
Give him a call. Tell him.
Yeah, exactly.
Tell him to turn around.
If you could just pass that along to him. Yeah. It's definitely the head for the head is not anywhere near it. Tail first.
Where's it going?
Out, sadly.
Uh-huh.
RV bridge.
Go for RV.
Hey Zach, I'm going to try and bring the heading round 45 degrees, but I'll keep you out of the circle.
45 which direction?
To port. So I'm going to come to the north of you and then come round just because we're using quite a lot of thrust because of the current.
Okay, copy that.
Maybe. Oh, come back in.
All right. You're in a good spot.
Oh.
Close.
Come on, buddy.
Maybe you can bribe him.
I was gonna say, what would he?
If you
Oh,
sit still for a scan,
is that
he
in
won't
frame?
suck you up.
Okay, one more.
Come back.
Okay, a little bit higher.
Oh, maybe.
Okay.
Okay.
Ah. Yeah. Keep it coming. Keep it coming.
Always going to the right.
Keep it coming. We'll take it.
Yay! Nicely done.
Oh shoot.
And you got another one. You got a secondary.
We'll take it. Thank you.
All right.
Agora tá. Não, it's
Now he's just hanging out in there.
Yeah, but you see how that tail blocks the
Yeah, the head.
view.
Yeah.
So
Yeah.
thank you.
Cool. All right. Thanks, you guys. We will let him get away from us, and then we will move along.
Yeah, I feel like all of these settings, it would be nice to have like a return to zero button or set to zero button.
Deve ser isso aqui, Irena. Deve ser o Irena esse aqui.
That's fine. Very low.
Perfect.
Just need fewer meetings to make it all happen. And
Bottom left, middle. Okay.
There's a question on the chat. Are the DeepPIV iris scans shared in a database?
The DeepPIV models are actually openly shared. You could do a Google search for Sketchfab, DeepPIV, and Mbari, and you're going to see all of the models that we've already created. And of course, we don't have the models yet from this cruise.
Middle left.
A doutora Kakani está explicando aqui que todos os modelos já produzidos estão disponíveis no site do Mbari. Né? Qualquer um pode entrar lá no site, fazer uma busca e baixar os modelos.
Yeah, that'd be great.
Esses desse cruzeiro
This is
agora não estão
Larvacean, and he's
disponíveis, ainda vão
home.
ser processados, mas tem várias outras coisas
He's just bailed. He's
disponíveis
bailing as we
no
speak.
site.
Oh wait, is
Let's
it?
zoom in
That's
on the
fertile-
absolute fertilaria.
That's Fritillaria.
Yeah.
Yeah.
So see, oh, Karen's gone. You can see
What do you
that
want? Do you want the
fertilaria.
animal or the house?
Ideally both.
Do you want the animal? It will not serve, the likelihood of you getting it is too small.
Okay.
You're not, based on the surge
And the house
that
is
we're
starting to
dealing with,
disintegrate already.
Larvacean.
Okay, Kakani, would the house help you guys at this point?
Potentially, the house is staying in place, you can scan the house.
Sure, let's do a scan.
Let's try it.
please.
DeepPIV, scan of the house. Yeah, this guy looks small.
You get one good shot at it, and after that, it's gonna start to contract.
Gonna get gosh in the wash.
Yost.
Zoom in ali, ó. Sai, vai.
Esse é um uro cordado, não, Larvacean, que é o fritilária. Esses ureiros, na verdade, eles estão mais próximos da gente do que das águas vivas, apesar dessa aparência dele de água viva.
For some reason, when I was hitting the record button, everything went dead.
Ele acabou de sair da casa.
Later.
He's low. He's not in focus at all.
Nadando do lado era o bicho. O bicho nadando.
It's nice if I
Saiu da
find
casa.
one that's, to get it scanned while it's still in the structure, but this is a starter.
Coming in.
Oof, that is small.
Yeah.
Okay, I'm gonna zoom in a little bit more. It's gonna be fun. Okay.
So trying for that box.
You have to keep one out of the room, because every time I do, you get something good.
He
It
is.
does look like it's slowly deflating.
Yeah.
Yeah.
We might have. That's probably all
Yeah,
we're gonna
it's
get
a lot
because
smaller.
Yep.
it's deflated.
Right.
Okay.
Okay.
Thank you.
Thanks, you guys. Moving on.
Whoa.
I'm here if you want to switch.
Oh, and you
Too
too?
bright, too bright.
Look it, our replacements are here.
Yeah, too dark.
Yeah, that's great. All right, I am going to hand it over to Emily for the next couple of hours. Thanks, you guys. C-Log is also switching over as well. We've got 3 canisters left and no decouplers. So we have 3
Bom, a
canisters
gente vai trocar agora,
available.
vai entrar aqui o professor Renato Nagata.
And these are the
Fazer os
Niskins.
logs.
These are the
Eu vou ficar
wants.
acompanhando vocês pelo
If there's a decision to
outro
go up
microfone.
before we hit 1,200 meters, then get it as deep as possible.
Oh, we're not coming up, we're still.
No, we're still going down.
Wow, you guys got a lot.
Yeah, there's been a bunch.
Get to
So
the
headed to 1,200 if we
Yes,
can
you
make
have a
it.
boat.
We have
Yeah,
2
go
hours.
ahead. I just never
3 hours? We have 3 hours.
recycling it.
Yeah.
And then 650, 2 at 50, and 2 at the surface.
Okay.
If they can,
Yeah.
or just pass it
Yeah,
on
sure.
to Russ if they haven't
There's
done the
probably
surface.
some other ones that are in the recycle.
Sweet.
So we have to be on deck by what time?
I think it says 6. I believe it's 6 before.
Okay.
Yeah, give it a try. I don't know that that's bigger than what a lot of the bottles have been.
Boa tarde, pessoal. Professor Renato Nagata, do Instituto de Oceanografia da Universidade Federal do Rio Grande, FURG. Agora, começando aí a transmissão da descida. Já estamos a 989 metros, quase saindo da zona mesopelágica e entrando na zona bate-pelágica.
Yeah. It'll look a little low technology though compared to everything else.
Russ, I wanted to show you the basic coordinates. 3 years
That, Emily.
Don't apologize.
Talked very
No,
well.
don't apologize
Right,
at all.
let's keep
Because
heading
everybody's got
down,
different
and if we could
purposes
get a
for the
little
projects we're
forward
doing.
motion
So it
as we
doesn't matter.
search.
If you can't, then we can find somebody.
Oh, we can
Promise.
go in on that.
There's too many projects.
Is anyone
It's all good.
interested in the larvation?
Something interesting.
Russ,
Come in, right?
what are we?
Larvacean.
In
Larvacean.
center at the
What do we got here? What is it? I'd like to zoom.
No centro ali, uma coisa interessante.
the doliopsid?
It's that's two, right? Connected. Doliopsid train. Russ, are you looking for more individuals in a chain?
Oh, this is different.
No, yeah, it's different.
This is not.
How big is this?
Not.
It's
It's
got a really
actually-
big
it's not
lumen
bad.
on one side.
Bigger than I think.
You see that there?
Lindo animal.
I mean,
Tem-
we have all the choices. We have
This
no
is a tunicate. I have no idea
I
what
think
this one is.
You think it's a tunicate, not a doliolus?
So
The
Well,
people
it-
want
it-
bands are
to laser
doliolus.
scan
I
it.
mean, this looks very doliolid-like,
Right.
but this is not
Cool. Let's
sedusa.
hold
Right.
on it for a
Right,
little
it's
bit,
something
and then we'll
different.
go to DeepPIV.
It's similar to sedusa in the
Estamos
fact that
tentando
it's got this little process off
entender
here.
o que é.
But the gills, there's no gills obvious in sedusa.
Mhm.
Right.
This has got the gill division here,
Mhm.
and the doliolula has got doesn't have this big flared
yeah, this one's different.
instruction, and you can't see these round muscle bands. It's got two S-shaped muscles down the sides. This is quite likely new, like completely new.
Let's get it.
Do you hear that,
So we're-
folks? Maybe completely new.
Definitely gonna want to scan
Do all of
it,
it.
and yeah, so we'll just do the press. So we'll go first with iris.
Oh, okay.
Then
But
scan.
it's-
Agora dolio
full treatment.
bastante
Yeah.
diferente aqui, tá todo mundo intrigado com o que pode ser.
Okay.
Good luck, team. You got this.
Vamos
Thanks,
tentar
Heather.
pegar ele, né, e também fazer o escaneamento desse
Okay,
animal.
so we're gonna start
Maybe
with
his question is, do we
Iris.
want any
Iris is
more
opening.
close video
See on the chat.
scan?
This is a panoptic camera, kind of like a fly's
unusual.
eyeball. That's gonna get a bunch of different angles on the same
It-
shot to create a 3D
Where
image.
the I would almost put it under, yeah, call it a doliola because I don't think we have doliopsid in there. It's a different family technically
Hanging
from
out
the
just below
doliolid as a
DeepPIV.
family, but it's all part of the doliid which is the next level up.
Okay.
All right. So Jimbo, you have all of the. Yes, great. Full treatment.
Mhm.
Now this is very transparent, so it's unclear to
I
me.
would say chain because it looks like there's at least two.
Iris, but we'll at least try.
Got something else flying in too.
What is that?
Is that a little?
Too close to the vehicle. What is that?
That's something else because
Oh.
the
Ironically, there's
doliolid is
something
still
else
out
trying to
in
get
front of it.
charismatic.
That's hilarious.
Pick me salp or something.
It was funny. That was booping. Yeah.
Pretty good.
Was it
Three and no
Yeah, it's gone down the
samplers.
side.
3 buckets, okay. You can come up when they're full.
Okay.
It's like an image or something that doesn't exist.
We were considering, well, just need to figure out if we want to still go down to 1200 to get the bottle.
No, it's just meant to be the deepest.
There really isn't anybody.
Oh, okay.
The
Thought that
Cool.
was gonna
So
be
we'll
it.
start
fellow
going
in
back up after
Croatia
this.
who worked
There's a
on
new
it a little bit.
doliolid looking thing.
I mean, Larry was kind
So we're gonna
of
get Iris, then
the
DeepPIV.
person right
Yeah,
for a while.
Very few
And,
people work.
Garo.
I have a PhD student that is working with doliolids as well.
Speak now or
but
forever
on
hold
taxonomy,
your
yeah.
peace on
On the
that one.
taxonomy?
Yeah, he described one species last year,
Yeah.
a new species.
Well, then this might
Yeah.
be
Give me a
up their
shake.
alley.
Oh, I will,
Do
yeah.
the on-bottom thing
Have
on
to
event
be
on
for-
the big.
Oh,
side.
perfect. Okay.
Thanks.
Will do. Thanks.
I don't see the other camera view, so I don't know.
Okay. Kakani, do you see? It looks like it's starting to get in the.
I don't see the other camera view, the one that's on top of the camera, so I can't give any input.
Do you know what that camera's called?
Well, it's the one that's on below to the right of the iris feed for the pilots.
There it is.
Yep, yep, yep.
Oh, so yeah, the red dot going into the middle.
Try it again. I think it's just a little too far.
It showed up in the camera there for a second.
Very little. All right, let's move to DeepPIV, please.
Okay.
Okay,
So,
Yeah. What? Yeah.
All right, this is pretty small, right? So I'm gonna zoom in.
Okay, it's down. That's interesting. The Larvacean's still kicking around from
Okay,
before.
doing your lights.
That fertile area up there. Don't worry about
Could
it.
you
It's a little
swap with the
high.
DeepPIV?
But it's interesting. It's still close.
Still a little high. There, I think that's it.
okay, we're gonna guide the doyle through the laser sheaths.
A little
Wow.
low.
So close.
Right, more space on the right. Nice.
Beautiful.
Good.
Oh,
Oh, again.
not that one.
The first one though, that was so good.
Sorry. That again. I think it's a little bit too far to the left. Alright. Little fast. Just do that again. Oh, a little bit too high. A little too high. I mean, the directionality was interesting. Now let's do one more good one and then we'll try and collect it. So then you say you have a student that can work this up.
Yeah, he's doing like taxonomy and
Oh, oh. Ah. A little bit high, I think. Okay.
are we
Just a
going
little
to
bit
collect?
the left.
Are we going to try to collect
It's
this?
Too
out
far
of
to
the
the
area.
left.
I
Yeah.
gotcha.
So we're gonna collect it. We're gonna do sections here.
Yeah, good.
All right. A little fast. One more time. Oh, a little low. Oh, a little high. No.
It's too small a region.
One more attempt. One more attempt. I think it's in a good spot. Oh.
It's bobbing. Bobbing and weaving.
I think it's too close.
Hmm.
Close and too high. Too high. All right, now it's out in front of us.
Okay, I think
Yeah, let's try collecting.
Okay.
I think we've got at least one worthless scan.
Cool.
Where's it going?
Yeah. We are.
There. Okay, there
Yeah.
it is.
Me and
Still
my
in front of
student
us.
were struggling hard to find
All right,
someone
this will
to
be
have a
bottle
feedback
2.
about the doliolids.
Yeah.
And then we
Well,
wrote.
I know the other species.
Thanks
in
so much,
this
Sonny. That was great.
pretty well. We get them to go.
Diorama
and
does that.
expect good pictures of them.
Yeah. We wrote to Van Soest, Dr. Van Soest, and he may be like 80 years old. I don't know, but he answered us and still.
So can we zoom in on that animal again?
Yeah.
Because I think we had 2 individuals. We're now down to 1.
Yeah. You don't want to miss it.
Yeah.
And it's now looking much more like Doliolula.
Just keep down with the symbol.
Yeah. They want you to zoom in again if you can.
Sure.
But I'd never seen the one siphon open the way we had it in those first pictures. Now this looks exactly like Doliolula.
Esse animal que
You
está.
can see the F-shaped muscles on the side.
Esse animal que está sendo filmado agora é um doliolido. É um cordado do grupo dos doliulos, né? Então é um animal que é um cordado como nós somos, próximo dos vertebrados, mas ele ainda é um invertebrado. Ele não possui coluna vertebral. Eles possuem notocorda, mas não desenvolvem a coluna vertebral como outros vertebrados.
Oh, wow.
Wonderful.
Gorgeous.
Wow.
Yeah, come.
Great piloting.
No. Imagens
What do
bem
you guys think? You
detalhadas
still
do
wanna
bicho com a
collect?
câmera de alta resolução.
Yes, we should.
Yeah.
What you're going to need to do is it's going to be really fragile.
Oh.
So you're going to want to kind of like flick the pump off and on to keep the velocity from getting so much that when it goes into the basket, we just plaster it to the screen. So bursts of pump and then kind of let the velocity carry it a little farther, burst of pump until we get it down the tube.
Roger.
But it's okay to have full velocity until it gets into the tube. Once it goes down, that's when we need to start giving it bursts, unless you can ramp the power down on the sampler.
Okay. I'm gonna bring it towards the funnel just in there so I can.
Yeah, so it says it's approved.
So as we get this towards the vacuum, for those online asking questions about the laser systems, both IRIS and DeepPIV help us get a full 3D reconstruction of the animal.
Vamos tentar coletar esse doliolido agora.
Super important
Parece
for
ser uma
determining
espécie
if
que
it's something
ninguém
new, and
conhece, é
also
um grupo que é muito pouco
creating,
estudado por
a good
especialistas.
look at its morphology or
E
its
ele
body
vai,
plan.
né?
That is difficult to
Estamos fazendo
do
a
since these
coleta
animals are
com
so
o
fragile.
suction
If
sampler.
we take them out of the ocean, it's much more difficult to study those features.
Then you can hear that you're not talking over her. Yeah.
Okay.
Yeah, this is a doliolid for those just joining that we're trying to collect. This is a solitary one and originally had a second. They can form chains.
Did that go down?
Yeah.
Okay.
Okay, we're gonna look for.
Off, or at least start flicking it into second or two on, second or two off.
Roger. Moving ahead of
Drive,
them.
drive slowly forward.
Um animal pequeno, então é difícil fazer a manobra para chegar na posição em que ele está e tentar coletar ele de uma forma cuidadosa, porque ele é frágil. Então ele pode tanto ser destruído pelos propulsores do ROV e também dependendo da força que o suction sampler estiver operando.
Perfect. It looks like it's still in okay shape. Great job.
Jimbo, let's mark the bottom. Let's record bottom depth, or sorry, bottom of dive or whatever it
We can
is.
do that.
Great. And then we're gonna start ascending in our search. No, 1200 was our target, but we can just wait till the 650 bottle.
No, I think they said the deepest you go
Oh,
to.
deep
the
as
one.
possible? Oh,
Yeah.
okay, perfect. Then,
No,
yes,
you could
let's do
double
that.
check.
No, I mean, we
Mhm.
have the bottle, so may as well. If they don't want it, they can just toss it.
Yeah, we should definitely fire the bottles.
Okay, great. I'll mark where we're at. So we'll be doing
Just
at
kill your
987.
But were we gonna fire the bottles at our deepest point?
Yeah, we're gonna start ascending
Oh,
This is
now.
Yeah, so I think
This
we've
is
been
the
doing
entrance.
one, the last two dives, we've done one at the bottom, one
Mhm.
mid, and then 250, two at surface.
Yeah, 2 at 52
Yeah.
at surface.
I think she wanted to go 1200.
Yeah, Karen said since we have so many filled, she'd rather we start ascending.
Yeah.
Yes. So we're gonna fire a Niskin bottle at 987. A Niskin bottle is just a water bottle. We're opening up to collect the specific water here at this depth.
Coming to the main camera view
Perfect.
yet?
Thanks, Sunny.
See it? Coming up around the sample.
What are we looking at?
I think it's a Freya.
One.
Freya
Is it
dig.
one?
Yeah.
Siphonophore.
Ok, foi feita a coleta de água com a garrafa Niskin, né, que
Oh, we can
vai trazer
turn
uma
iris
amostra
off
de
too.
água que vai ser estudada em relação ao DNA ambiental, ou seja, tudo que tiver DNA dentro daquela garrafa vai ser estudado.
All right, let's start a search forwards and upwards. So as we search, I saw some people mentioning Fathomverse. If you'd like to join this dive even further, the data and all the images that you're seeing here are going straight into the game for annotation. So you can be an at-home annotator for this specific expedition. So check out Fathomverse, free on the Google Play Store and the App Store.
A Emily do Monterey Bay Aquarium está mencionando Fathomverse, que é um jogo.
Sinker, I think.
É um aplicativo que pode ser baixado e vocês podem ajudar, né, colaborar, colocando tags, anotações sobre os animais que aparecerem. Então todas as imagens do mergulho vão estar disponíveis nesse jogo e qualquer pessoa pode ajudar a anotar que tipo de animais estão aparecendo nas câmeras que registram esses organismos.
Is that just a house down low?
Yeah, a little house.
Right lower corner.
Oh, what do we got here? Jelly.
Up.
Ooh,
There we go.
hello.
Nice.
Oh.
Something new.
Something interesting.
Where is Dougal?
Let's check it out.
Agora uma medusa interessante apareceu.
Go up or down?
Up. Oh, there it is. Wow. Gorgeous. Atola genus or something?
Atola.
We saw them yesterday.
Atola, yeah. So it's in the.
This is really
yeah.
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 It's big.
Gigantia.
Yeah.
Olha só.
We're calling out Dougal if you hear us. Tell us what this is. That is stunning.
Yeah.
Oh my
Look
God.
at that amazing
Oh yeah. It looks like a mooncake, the top.
So each of those white kind of things you see around the outside are embryos.
Oh.
Right beside the dark brown.
She's
and before the
pregnant?
ring.
Fraggers. That looks like George's
Oh.
Atola. Good luck.
George's?
Muito bonito aqui, uma tola.
George Matsumoto, if you're listening.
Something similar to it. I know, it's oddly fat.
Uma Siphonophore do grupo Coronada.
Hey,
So this Atola jelly has embryos in its bell. You can see it's slightly reddish.
Só tá comentando que
That makes it kind of invisible in the deep
ela
sea.
tem larvas dentro da umbrella. Eu não tô enxergando. Nessa posição não dá pra ver.
We could get rid of something.
Pretty
Well,
big.
we could get rid of the protist.
Wanders, you asleep?
500 meters. 500 meters is, well, at least according to
Yeah, go ahead.
the Monterey Bay observations, is where we see a lot of H1.
Wow, look at those moving kind of inner tentacles underneath. Hopefully it gives us a good view in a second.
We choked the suction.
He's got to sleep sometime. Wow, is that something in its mouth?
That is its mouth.
That is its mouth. Okay, great. Glad I have you
It
here
looks a
to
little
answer
beaten
me.
up.
What?
What?
Wow.
What?
Olha o detalhe.
I need to take it
Olha
now.
essa imagem.
That's
Its
beautiful.
little tentacles really tangled in its other.
Yeah.
Those, right? The really long filamentous ones.
Yeah. My hair looked just like that when I woke up.
I would indeed drop the Shadowgraph if you think this will fit.
We could do
The which one is
Esse
that?
animal parece bem grande.
The oh, okay, in the
How
riser.
big is it?
Sorry.
Well, I guess we're
Pulsação
just
dela.
gonna leave it behind.
No. I think we're gonna the desampler.
Where do we get
Is it
rid
gonna
of?
fit?
Well, I mean,
Yeah.
we've seen when we've got the D
How's our
sample, we
lasers on
see
there?
it routinely.
Probably 30 centimeters.
What?
30
The
centimeters.
It's not like it's.
Yeah.
Oh, we are
It's not
ditching.
particularly unique. We're ditching the proteins.
We could do.
If it will fit, I don't know.
Yeah, can we do,
How do you
a
You
quick
guys
just-
scanned it, yeah?
No, I'd like to do just an iris
dense.
measurement.
You're okay.
It'll be
Not for
too
iris.
dense for DKV, but for iris, I think it'd be great.
Wow.
So, Sunny,
Oh
Yeah.
my
I can
gosh.
get my lights deployed
Which one?
For iris.
on iris.
You size it first before you dump these.
So when
Oh, you will. You'll
Sorry.
already. But do you get
I don't know where that came from.
Yeah, we get measurements from iris right away, right? Yeah. And they just had the green sizing lasers out too.
Or something else to put it in and be done and go,
call.
oh,
Good
wait a
call.
minute.
There it is. So to the right a little? Yeah.
Okay, so
A little bit,
we're using
it's a little far.
iris for this because with opaque animals you can't use DeepPIV. It needs to scan through it. So we're just using our multi-sensor camera in iris.
A little bit closer.
Suppose we try this auto depth limit and depth range limits.
A little bit closer.
Yeah, he almost looks blue.
That's on our end.
What does it mean then? Is
Just
it okay?
the depth range.
Incredible.
Hey Manu, how incredible do you think it is?
I think this one first.
One thing that would be worth doing if you ask the pilots after the scans are done, just switching the light off for 10 seconds.
Oh, to see
Yeah.
bioluminescence.
Most of them have.
We don't want to lose this.
Yeah. Not. I guess we would want
It's
to
a little
capture
too
it.
close. It's a little too close.
One more zoom? Can I do a little more zooms on it?
Yeah.
Kiss it with iris.
Wow. That's really excellent. Seeing all those different colors is really good. That means we're getting a lot of data. We're looking at about 30 centimeters across
All
the
right, I
bell.
think we have enough data.
Estamos
So we
tentando
should
fazer.
collect this.
Estamos tentando decidir se vai
Okay,
coletar ou
so
não.
we're gonna try and put this in a D sampler.
It's fine, we'll do it here.
Okay, great. Glad to have you on board. All right, so we're gonna try and get it in the D2, the outboard D sampler.
Ah, vamos tentar coletar sim.
Oh, but really quick, before we do that, could we do you mind if we turn the lights off just for
I think
a few
as I
seconds
said,
on it?
maybe
Maybe not, in case we lose it.
If we catch it, should we be able to do that in the lab?
yeah, I can. Just they get beat up.
I just don't want to.
Exactly. Billy, you could,
Sonny's confident. We got it.
Turn almost all of them.
Yeah.
I'll just do it like we're doing for DEPIV,
Yeah.
right?
Very short.
All right, take a quick look. We're gonna see it in the dark. Oh my
Agora
gosh.
apagar as
Looks
luzes
like a
para
tulip
ter uma
bulb.
outra visualização.
So on, all right, ready when you're ready.
Yeah, go for it.
Com luzes e sem luz. E aí agora com
Yeah. That's it.
para fazer
All right.
escaneamento.
Lights back up.
Escaneamento com equipamento.
All
DeepPIV.
right, and now we're gonna go for the outdoor descent.
That's cool. That's cool.
Nice find.
There was still
You know, do some more zooms and stuff first before we try to.
I think we're
find
set
what
there.
Might be a little big for what we.
Okay, so you said it was about 30 centimeters on the
That's
bell?
yeah, it's tentacle to tentacle. It's pretty big. I don't know that it's going to go in there really nicely. We can try that.
Os 30 centímetros mais ou menos são só da umbrella, né? Que é essa
Yeah,
região
that
do
is what we're
guarda-chuva.
doing.
You can see if you
Any
if you sit
objections
down a
from the
little bit,
crowd?
you should be able to see it right over them.
Yeah, we'll do that.
Oh my gosh, that's wonderful. That's pretty
I
big.
mean,
Yeah.
Uau.
it's a big boy.
Olha.
Anyone object
To what?
to putting it
It
in?
might.
Oh, well,
Maybe.
maybe.
We're just sizing it.
I mean,
to leave it for this.
I mean, for the ID work, what tissue do they really need?
Yeah, did we
Like,
get
Dougal
tentacles?
needs tentacles.
Yeah.
We get
Like-
tentacles.
Okay.
That's the main part of it. You probably lose
Yeah,
some
let's open it up.
So a number. The right one, right?
Yeah, right
So
side,
the right
D2.
one.
The D's the only,
That is 5.
Yep.
it's big enough, it's
Yeah,
right
D2.
diameter just to go into the D.
Tô vendo aqui perguntas da Aline Ramalho. Um abraço para você, Aline.
It's gonna be the biggest animal.
Wow.
A cor dela é real, sim. Animais de profundidade tendem a ter essa cor vermelha.
I think we should target the tentacle.
The yep, no side camera looking now.
I feel like we got a lot of nice fish. We got some
Opa, perto do D-Sampler na garrafa, olha ali. Olha, será que entra?
got tentacles
não.
in there, so
Ah,
we
os
wanted
tentáculos.
to
Three
grab
is close.
those.
Quase.
I just don't want it to just drag them out though.
We don't want to do tentacles.
It'd be nice to get the animal. It's big enough, it's hard to lose. It's just a question of springing it into the hole.
Ooh. Yeah.
Get ready, get ready. Almost.
Almost.
No, breathe. Breathe.
170 close stuck in there.
Julia,
Yeah.
could you bring
One more
the
time.
ramen?
Bring the ram in.
Yeah, this way.
Nice view.
Wow, nice view.
Coming up here.
Oh.
Wow. Gorgeous. Oh my god. Oh. So this is a jellyfish for those asking. It's an Atolla genus.
Lily is here.
Yes, hi Lily. Hi my sister Shannon who's watching. Shout out.
They zoom out on the system.
Zoom out on what?
The size or
It's zoomed out. Up and pan left.
yeah, palm left. I don't know where it is.
Agora ela sumiu.
Still on us?
That's it. Yeah.
No, there it is.
Oh, there's.
There it is.
Found ya.
Ok.
What's going on with this?
I don't know.
Put it in one of the biscuits.
Not real.
Turn it on.
It's not just sit.
What
There's
else?
a couple different feeds.
Yeah, it's a couple feeds.
Push into a fiber.
Okay. Good thing we have so many. Okay. That's what it was doing while we lost it. It was disabling our cameras.
Oh, come. What is that?
Oh God.
Slow death.
Oh.
Oh.
Many things.
Oh.
Let's just close it for now. Or I was just gonna say worst case scenario. Yes, is this the calibration that it's doing?
Jimbo, could you be able to?
Look
Would you be able to
beat it in
the,
this.
What date? Yes,
Step
what date?
and switch
Is
his,
the calibration that
bring
we
him
want?
so you can see.
70, the recent one.
Yeah, is this the
Recent
recent?
third one.
Is this the recent one?
Yeah, that's the recent one.
Oh, back.
Yeah. Yeah. Have you done one of these
No, no,
calibrations?
no.
Okay, yeah, you don't want to. No, we're good, but I'm more worried about the great discrepancy between the recording things. When we record
É
events,
muito difícil fazer coleta. Além das correntes naturais, né, as correntes marinhas, tem o próprio locomoção dos propulsores que gera bastante turbulência ao redor do ROV.
is that changing when we're like when we
Opa,
record these
opa.
runs? You can see it.
Oh.
That's it.
That was close. Okay.
That was cool. That was cool.
Okay. All right. Well, there's no Riseria, and
Amazing,
there's
guys.
the Atolla now. Okay. Uh-oh.
Amazing.
The feed's glitching over
Almost
here.
had to tuck it in with the top. It's going out. Tap it a little.
Mais animal lindo, fantástico para ser estudado aqui pelos pesquisadores.
Put that.
The bell's intact, that's good.
You know, put that sample down to 0.
Some tentacles may be intact. A large number of tentacles
So
are
that was
intact.
number
There's a lot of
5.
tentacles. We're
Put
going
5
to
to
take some
0.
DNA off the tentacles, probably on the edge of the D sampler when it comes up.
Yeah.
Before we open
There's
it.
no. Put it to 0.
Shears them off.
So everything's cool?
Okay, we're having a ROV pilot changeout.
Are we okay, Jimbo? with
Yeah.
the,
No, we've just, I think we've lost the PI.
The what?
Okay.
Então, serão feitos estudos de biologia molecular, né, para entender coisas sobre a diversidade genética de vários animais e também estudos de isótopos estáveis para entender o papel ecológico desses organismos nos ecossistemas.
What? What'd I do?
Iris
I
on
want to
Atolla?
find the chat. We don't need to dwell on it now, but at some point I'm going to dig
Why
up,
is this
dig up the
here?
live stream and see if we
That
can
was a
capture.
very weird moment.
I'm confused.
It's okay. We can move on.
Okay.
You saw the 0 at that.
Yes.
If
I'm still swaying. We've been watching, yeah. We let you guys.
I've been trying to give you a
We wanted to
break.
come take over.
You know.
Uh,
They kind of go like, come on, Steve.
And then stuff started crashing. I was like, I'm not doing it.
Hi, Mom. Sorry. She's mad that I said hi to Shannon. I'm in trouble. Aaron O'Donnell, hello.
Okay, so coming up.
All right. Hey, pilots. What's that?
Smell it.
Something.
It's like one of those things you smell that helps you wake up a bit.
What?
But it reminds me of my dad. My dad smells like that.
I have a little peppermint stick
So
in
I
my
do it
pocket.
for that.
Nice.
I have these when I was a kid.
Okay, so we are heading up. Searching forward and up.
Agora vamos voltar.
Haley and Massimo.
Estamos a 960 metros de profundidade. Voltando e vamos fazer um novo mergulho às 9h30. Programa.
Yeah, yeah, you can have it.
I still don't understand why the doves are huge.
All right, eyes peeled, everybody. Tell me when to pull over.
And it also didn't seem correct. Like, when the depth map would be good. So from back to front of that jellyfish was definitely more than 40 meters. Open the file and see what's.
Well, the humidity values of all of our housings still good.
Oh, I thought you were. I thought you were announcing that they were not good.
We're done.
All right, what's everybody manifesting we find?
Oh, that's gonna work today, yes.
Let's hear it. Manu, what about you?
Do I wanna see it?
Yeah, what do you want to see? Produce. The vegetable of animal categories, aka it doesn't have a category.
To make sure.
Connie?
Acabamos de
What
trocar
would you like to see?
a tripulação que está fazendo a
Giant
pilotagem do drone.
We saw them between 600 and 700 meters.
Okay, let's head up there.
The houses are very, very fragile. It looks like you can't see the inner house.
Okay.
They're very transparent. That was earlier today, so it's
You know in
possible
that
that they've been
working-
filtering a lot of stuff and we might have things that can scatter.
Os pilotos fazem turnos de 3 horas e os mergulhos podem
Cool,
durar até
okay.
12 horas.
Let's book it.
600 to 700 meters.
Emily, I wanna see
Joelle
it.
and Lisa are back, or Sarah, you guys are back. Ooh, that would happen. It's been 2 hours. It goes by fast. Really? Really cool stuff. Yeah.
Let's try and cover a bit of ground. We only have till 5:30. Is that right?
To do so
Or
many scans.
6?
Right. 7.
What? I think we have till 6 to get back on board, right?
I think it was 7:30 is what it
Oh,
shows.
7:30.
Yeah.
Oh, okay, that's better.
There is a new chat thread called Schedule, and it's a link to the Google Doc Karen uses to create our schedule. So you can literally look at the doc.
Nice.
Is that the one you want?
Yeah,
7
yeah, that's
p.m.
cool. Thank you.
Oh, perfect. 7 p.m. RVM.
Okay, we got it. No, we just wanted to see
celebrate.
it.
Do you want to see it all the time?
No.
Oh, Russ has brought us chocolate. Doesn't look like it. Yost, what are you manifesting?
Thank you, Rose. Thank you, Kakao.
Did you Medusa from Yost? Weird cephalopod. I'd like to see one of the ultra dark fish. I'm not getting to that.
I just got one. Thank you very much.
The chocolate's being passed around the room. So while that's happening, my name is Emily Clark. I am from the MBARI, the Monterey Bay Aquarium Research Institute. I work for the Bioinspiration Lab, which is the team that put IRIS and DeepPIV on the ROV for this expedition. And I do a lot of work on Fathomverse, a community science app that allows you to annotate real footage exactly like the images we're seeing now. In fact, this expedition is going straight into the game as we speak. So if you'd like to help us annotate this footage, download Fathomverse and check it out. Yes, anglerfish, maybe dragonfish. We saw a gulp reel last night. Ooh, deep staria, that'd be fun.
Yeah, you can help.
A little protein, not the sugar. Much better than the sugar. There's one more piece of chocolate. Who wants it? Noon? No?
Is anyone looking on the screens? Or just at the chocolate?
Chocolate is important. My menu.
I haven't added them yet, but I've got three more. Three more coming. Yes, they're made. I just haven't uploaded. I went to sleep instead. Oh yeah.
You should tell them about Fathomverse. I did.
But it's not out yet.
Everybody in the chat
So you can get them excited.
Get them excited. Ooh, Cali Love, yep. We're from Central California on my team, but we have folks from all over the world on this expedition. We've got some from Hoku University in Japan. We've got a bunch of Brazilian scientists on board. University of Rhode Island, Harvard, Bigelow Laboratories.
See what happens.
SD stands for super dark.
The yellow?
Only super dark chocolate with milk.
Can we zoom in on that yellow dot there?
There's this computer just sitting in the electronics lab. Stay away. You stay away.
It's pretty tiny, I think.
It hasn't finished yet.
That's okay. We can keep searching.
We know where you live. Here.
Yeah.
Look at my room. In this room.
Accurate.
Disclose to
All right,
team members
Russ.
which.
I will finally show this to you.
Yeah, start showing me again. We always get something good when you start doing this.
So this is inside of our inlet channel. And then we do see a pre-filter.
Yeah, the pre-filter's
Mm,
really
that's
close.
fair.
That's what I'm saying. It's really not stopping, but it's probably there retained more to provide some structural
Okay,
integrity.
freaking 900 meters.
The
It's a
design
big house. We
requires
can
some,
keep going.
scaffolding across that opening.
Yeah, we probably already dispersed it.
How big is this animal? This not the animal, the house. Okay. Wow.
So is that a series of tubes on top of the tube? So it's kind of like there's a layer, you've got the big lumen, and then you've got the tube going up across the top of it.
Yeah, so if I zoom out here, we've got an outer sheet.
The rest area, I think.
Inside. These
Smaller
are
filament.
individual channels.
What? I didn't say it.
We're going. It's right there. It's right here.
Okay, yeah.
That's the same one we've been seeing, right? This is a single-celled organism.
Bigger chamber.
Whether we see
And they're all feeding into another lumen that then connects to you.
Oh, that's beautiful.
Manu, what are those filaments made of again?
They're silicaceous.
So this is silica. So this is basically like a glass single-celled animal.
Does it show where the little hairs are connecting it onto the house?
Beautiful. So fine.
Agora estamos vendo um protozoário, uma célula, um animal unicelular, mas uma célula gigante que pode ser vista a olho nu. É como se fosse uma ameba que tem extensões, projeções da sua membrana plasmática que são feitas de estruturas de sílica. Então ele tem uma armadura de sílica com orifícios por onde a membrana plasmática da célula se expande.
It shows up in some of the drawings occasionally, but it's very subtle.
E ele usa essas estruturas que parecem com tentáculos para capturar alimento, né? Partículas da neve marinha que estão caindo, né? Que são aderidas.
Oh, really? This is super. I wonder.
Because there has to be there has to be a focal point, right? That connects, that maintains the animal stability.
What do you think, Manu?
Oh, I think it has peptides on it.
Oh, great. Let's collect it.
Look at that, up on the top.
It's got freeloaders on it. Oh, that's its food, or they're taking a ride?
You're showing there to be a lot of
Farther away on the
slack
tentacles.
space. As opposed to a kind of a distinct, like,
É possível ver, né, o alimento que é capturado e que é transportado para a célula.
That is
no
so
centro, ali que vocês estão
beautiful.
vendo.
Dawn did it. He made the analogy that it's kind of like a tube, like a piston tube, only instead of having a ram, you've got a sinusoidal movement, but that it's a seam.
That's fathom verse.
Pushing a parcel
Thanks for
of
asking.
water as opposed to
All right.
simply
Do we
trying to create flow
grab it?
in the area.
All right. Let's do the vacuum sampler into bottle 3. Oh, weird.
Vamos tentar fazer a coleta com o suction sampler.
This is quite small.
O professor Manu Prakash de Stanford solicitou, ele que é especialista em protozoários marinhos.
We have the other
Yeah.
So we use this in the giant larvacean tail chambers to measure fluid flow, to estimate
Yeah.
and so do you want the lasers? I don't see.
Yeah, too small. But Manu will scan it with all kinds of instruments once it's on board.
No, the laser's been helping us. Actually, it shoots across right here,
Mhm.
and it gives an extra depth.
Oh,
Quem está narrando em
nice.
português é
Well, if you'd like to bring it
o
up as a
professor
guide,
Renato
I'm sure
Nagato,
it'd be fine.
Universidade Federal do Rio Grande.
Full sun right now, just over.
Yeah, I still think of that as a little bit more, it still has kind of a tail. Not huge,
Ooh.
but there is
Just
a
about.
tail.
It's on.
Yeah, it's just on the edge.
Gets to be,
There it
all the
goes.
tails, the cross section is you have the tube.
I don't know.
And then you
Well,
have
it
the
might be
fin.
flopping around.
All right. It's
Renato, there's people in the chat asking who's been translating. If you want to introduce yourself.
Yeah, I answered them. Thank you.
Oh, okay.
Yeah.
There it goes.
Founded on
All
Dorsal
right,
and
check
ventral
the. Oh, there it goes.
Yeah.
All right.
Oh, foi.
Yeah, it's every time it sheds
And
some
then the,
of the longer
Parece que conseguimos coletar. Eu tava olhando no
All right, so we have a 4M and 3. Great job, you guys.
cellular.
It's easy peasy.
Não são tantas garrafas, então esses animais que vão ser coletados
Alright,
têm
and
que
we
ser
will
selecionados.
keep searching.
Apenas coisas bastante interessantes, desconhecidas, espécies que podem ser novas, que são coletadas. São 8 garrafas, 8 possibilidades de coleta.
Yeah, let's try and really book it up to like 700. We'll just close our eyes, see if we make it.
On larvaceans, take a look at different cell layers. A on the tail and also have like the arrangement of so we looked at the muscle layers and we also looked at because one of the things we're wondering is like what's the function for the tail morphology. Because we're seeing differences in morphology between Bacterius magnetti, which is the biggest. I couldn't even. That was the biggest backbone.
Yeah, I'm sorry.
Stygios is smaller.
Yeah. Respondendo a pergunta do chat, o professor Marcos, da Universidade de São Paulo, saiu, ele trocou o turno comigo, o professor Renato Nagata, da Universidade Federal do Rio Grande.
That was at
A
about
FURG.
890.
That good speed or faster?
That's good.
Well, the Bathyphoria's tail is unique in that it's bluntly cut, and most of the others have actually either
6,000 miles.
tapered or double tapered.
Gonna be there in 2 weeks.
Small, more central panel that goes in depending on the species, both in the fritillarids and in the poiquiparids.
Yeah, and then you can see that too in their tail regions, right? There's definitely far more sinusoidal right before that, whereas for Bathyphorius, it's
Peixinho ciclotone, provavelmente.
Karen was saying she recommended a spherical Tomopteris tank.
I think they tried it.
Oh, really?
Yeah.
She said they live pretty well in captivity.
They seem to really like it though.
Longest part of their tail is
Mm.
I was told, yes, did we get some good scans of Ziparius last year?
We've got several batches of them. We've got some in, what was it, October? Right before Halloween.
Yep.
We got some in Super Bay, in Monterey Bay. We also got a bunch off San Diego 2021.
What's that centering going down?
Is that
Just going off
sinking
screen now.
house?
No centro.
Oh, it's all right.
Passou ali.
That's we gotta get. There's gonna be a lot of up top.
So that you can see the layers there where they mostly
Yes.
off.
Yeah, and Kakani was saying they were seeing a lot of large larvaceans. What did you say? 700 to 400?
By a lot, it's 2.
Okay.
600 to 700 meters.
Sure.
We're optimistic.
It's what's been eating the scattering layer
Yeah.
area.
Yeah. It's so
That's what
crazy
it
that
was,
layered
structure
scattering layer.
250. Secret hideout for
What's your guys'
megamouth
sampler
sharks.
situation? So all the D's are full.
Agora nós
there's
estamos a 800
contention
metros de
whether
profundidade
we have
subindo.
2 and 4 open or just 4.
3.
Estamos
Well, didn't we just fill 3
acompanhando o
with the
movimento
4?
de migração vertical diária que é realizado pelo zooplâncton,
Yes. Sorry.
que também está subindo. Na medida que vai escurecendo, o zooplâncton marinho acende a superfície para se alimentar, aproveitando a escuridão da noite,
We did. You're right. Because that's a question.
que os ajuda a não serem predados por outros animais.
First and 2 is in back there. It's deceiving us.
Okay. Nice.
Não é possível ver agora aí vocês que estão no chat, mas
Okay.
4 available, including the one that doesn't close. Is that it or 3?
We have 2 available. 2 and 4.
Okay.
And both
So
these samplers are
including the
full.
one that doesn't close, or not?
Wasn't that one 6? That one is full, according to this.
No, but the very last position, there's no blanks, there's no way to have them all, not sampled.
Okay.
The last bucket remains unrotatable.
Okay.
So that drives
But we
what
can
you
still
will or
put
will not
something
put in.
in.
You can put something in, but there's always a chance you can get out if it's really mobile.
Not sure how we ended up with
It's such a long swim.
one in between, but
Between you swim out.
So 4:30.
This was 9:08.
Oh yeah.
Sparkling stick ball. Yeah, no, that's, tentatively, we're going to call it Pastigurina, but it's tentative because we don't actually see many of the finger-like injections that are inside the house. So the idea is it's
Squid,
possible
squid,
it's
squid.
It's also possible that it could be a finger
That'd be great.
silica-like shell. So we're gonna wait for this guy to come up. We've had a few come up, but a little bit damaged, so we can't see the shell. But they all form these silica layers. If it's a Feodarian, many a times the silica has
Tone of disappointment.
and they would
last
all
night they kept
be
fooling me. They were like
the
curled up, or they were swimming like a
category.
fish, or it was a different color. And I got fooled like 6 times.
Oh, I see. I see.
Ooh. Hello.
Pequena água viva.
And a little fish.
Again. Where's Dougal? Oh, it seems to be.
And we have found a jelly, so we are gonna
That looks
continue
like the
this.
kind
Yeah, it looks like the
yesterday,
jellyfish
right?
we caught yesterday.
What's that called?
It's
Renata?
we don't know. It
think we have one in the box
looks to be the same that Dougal.
I think we've seen this before.
Yeah. It's a tiny one we got collect yesterday.
Let's just get a little footage and then we'll keep going.
Oh, we see if he wants it, and then we'll fold.
Yes, I think holding on.
Yes, it is a
for the
rhizarian, and
research.
then within that smaller group
We
foraminiferant.
need some images. At least some.
This is a medusa.
Uma medusa coronada aqui novamente, para ver as pontinhas brancas que são gônadas. Tecido reprodutivo dela. E nós coletamos esse animal ontem. É a mesma espécie. Não sabemos o que é, pode ser uma espécie nova.
Sure. Renato, is this a scyphozoan?
Yeah, it's also a coronate Scyphozoan.
Okay.
We
can mark it as a coronate
We collect
scyphozoan.
in the Tucker Trawl and also in the
I don't think we collected
in
this
the
one yet,
DSM.
I think we did.
We collect, yeah.
We got some good footage, so we can Oh,
Go for it.
collect it, you think?
Unless there's something more important we're trying to get.
Yeah, it's important because we don't know what it
We
is.
don't know
Yeah,
what
yeah,
it is.
we don't know what it is. Yeah.
Collected one, so
Please,
we
please.
don't have a genetic sample. Or
Yeah,
Oh, okay, I thought we had collected one.
We're paying for
All
it
right.
then. Yeah. or-
Could we do vacuum sample of this? Of the jelly?
So you can just stop here. We'd have to go down a little bit since we have
And this job.
some out right now.
Yeah. Get a
Or I can pay and make us real tight and then pay out. bring Delta to
The difference,
Yeah, usually. Further, or is that good? That's good.
Vamos tentar coletar esse animal com suction sampler, uma água-viva menor, então pode ser coletada por esse amostrador que deve ter mais ou menos uns 15 centímetros de diâmetro. E parece ser uma água-viva desconhecida. A gente não, o professor Duga Lindsay estava discutindo comigo ontem. Pode ser uma espécie nova para ciência. Opa! Coletamos.
Smooth as can be.
Excellent.
Let's look at the
Thank you, guys.
jar.
Okay. So, Chief engineer.
It's in.
Oh, he's there.
There it
Right
is.
there.
Nice.
Well done.
All right, so I'm gonna stop, having you come up. And get situated. And we'll change that to, you want it on 2 then?
Yeah, rotate.
Take to 2.
Okay, so that is the last sampler now.
Mhm.
It has to be
2.
something that may come up.
Okay.
We're going to jar 2. You want us back
Yes.
on 2?
Yeah.
Jar 2. Yeah, I see that. Yeah, there's a little
There is
something
something in
too,
there?
We've seen it before.
Yeah, I can see it.
It's not marked,
We
but
collect
Right there.
yeah, I see it too. Okay, well, with that, we may as well just come up if that's already full.
I know, but
all right.
the other one looks more like it.
We're not gonna look for
Well,
larvacean.
Did you see what we've
The
got?
chief
the other
engineer was
pieces?
just
We
in
got
here.
like-
Well,
Something like that.
Okay.
so we're not gonna come up.
Okay.
You're gonna pay up?
He wants us to pay out. He's going up to look at the outhaulers. I told him to come on clowns, and then we're gonna pay out.
Apparently George.
He was hoping 10 to 20 meters.
What are we? Oh, it's a calcophren.
Oh,
Beautiful.
Looks like it might be coniferous.
She's a relative.
Surely we're not gonna be looking at for larvaceans.
Oh wow.
It sounds like with
Good
the
question.
engineers need us
Oh,
to
nice,
pay out
nice.
first, and then we can see.
Quite big at this
Yeah,
point.
what is this?
Coniferous multidentata.
It's genus Coniferous.
Well,
I'm
like
looking
much farther because
at
we can't
the
collect
shape
things, so we
of
can
the
go up to
somatosis
larvacean
now to
depth.
tell species.
Okay, can I get something sort of more generally? That's a siphonophore?
It is a
Yeah,
siphonophore.
yeah,
Yes.
Thank
This
you.
is a
I couldn't make it
siphonophore.
simple.
Ó, que lindo.
I
Siphonophore
can't
agora.
see the somatocyst lying perpendicular to the long axis of the animal. So I'd say it's Coniferous moseri.
Dan.
Chuni from Charles Kuhn, his name, Chuni.
Here it is.
Yes. The body.
Moserai. Got it?
Cuniferous. Moseri from Fanny Moser.
Moserai. Family
Two people's names. One is
Clausophidae. You got it? Okay. É sifonóforo, outro quinidário que é próximo das águas-vivas, mas ele tem esse formatinho de torpedo aí. Então, 2 organismos, 2 indivíduos, né, que pulsam ali como água-vivas, mas é muito interessante como é o formato deles, né? Bem, eles têm uma natação bastante eficiente.
So this is
e
a-
isso
oh.
a gente pode ver porque ele está desviando bastante aqui da câmera, bem interessante esse animal que é colonial, né? Então a parte brilhante ali, mais branca, é a parte que ele usa para se alimentar e para se reproduzir, né? Então são vários organismos, cada pontinho brilhante é um organismo diferente, né? Que é como se fosse um pólipo de água-viva que vive junto e que faz a captura de alimento ali no final da colônia. Do gênero Coniferous.
Yeah, I
A
told
lot
them
of the
to come
so-called
over
rocket
clowns, so we
siphonophores you see with arrow leaves.
Nobody's interested. No one is interested.
Yeah, they call them rocket siphonophores.
Um,
I mean, we can't
O
collect
professor
it.
Dougal estava comentando que o nome popular dele é Siphonophore
But are
foguete.
we,
Ele realmente tem esse
We
formato.
just waiting on Dan to
É hidrodinâmico.
Okay.
Opa!
do something with him.
So in the meantime, we can look at that Siphonophore over there. This is, I think, a Fisonect.
I've got an ID on the one in the foreground now.
We're zooming in. Yeah. Teeny and tiny.
I mean, we don't usually turn them until 500, so.
Another.
Dolio. Going past.
Coleopsis.
Oh, yeah.
So just get close to the camera.
Did you catch a Doliopsis yet?
What's that? A larvation.
It was really weird. Colony of.
Oh, yeah.
And the one siphon was completely distorted open. Like it may be feeding or spitting, and by the time we got done imaging and bouncing it around, one thing, single individual, and it was back to the more egg-shaped or spherical body, more consistent with Dolio. So
Yeah.
Oh, look, there it is.
Following and he saw the
Oh,
Atorella.
Dougal, there it is.
here we go, Dougal.
Amazing, different one.
Sorry, what's that?
You wanted to look at this
Yes,
Siphonophore?
please. It looks like a Bugmania. Fisonect Siphonophore. Bugmania, Bargmania.
Agora
Getting a little bit.
Siphonophore Fisonect.
Beautiful. So that top part is all of its swimming bells pulsing.
Vocês podem ver
And then
ali
it's got
pulsando, né,
tentacles
os
and the little red
lectóforos.
part is its
Looks like
stomach.
a newly described Bugmania species.
Provavelmente do gênero Bargamia.
Yes, loud and clear.
All right, excellent.
Okay.
Let me know when you're ready. Are we just paying out haulers are currently off?
Yeah, we need them on.
Need them on? Okay.
No. Only genus. We're not sure on the species. Okay.
Haulers are enabled.
Bargamia. We cannot identify the species.
The natural.
This one.
No, it's one of the newly described ones. The most recent.
Okay, we're doing an engineering test and we're gonna let out some feed.
I don't think so.
Oh, is that a
Elongata?
No,
Solstice?
it's not there yet.
Okay. I'll keep in genus.
Helascera bigelowi.
Yeah. Another jellyfish. Opa. Voltando para o português.
Yeah, I hadn't seen the rest of the
Ó,
dome yet. I was thinking
lindo. Outra água viva.
Look at the, here we go.
Hmm.
Around the mouth, whether the shape of the
Alicera.
gonad containing portion of the radial canals is
10 meters, but
circular
we'll keep going.
or more straight.
I know it's okay. I mean, yeah.
What are those two
Bigelowi.
bubbles hanging?
Alicera, bigelowi.
Bubbles?
Yeah, there's like two round little bubbles in its bell.
Can you see?
white little dots. It's
Okay.
the apical end of the bell.
Oh, really?
Oh, that's just
That's all what I
reflecting.
want.
It's
Oh.
the light's reflecting off the jelly.
Agora uma água-viva traquimedusa, que é um grupo que não possui pólipos no ciclo de vida, apenas águas-vivas. Alicera bigelowi. Tem vários canais radiais ali na umbrella, né? 8 canais e tentáculos de 2 tipos diferentes. São 4
Yeah, so we're
tentáculos
gonna
longos e outros
go
tentáculos
up
curtos.
to about 600 and look for larvaceans to scan. And then after that, head to the surface.
Somebody was looking at that already.
Sorry?
You said somebody was telling you about
And if there happens to be an anglerfish on the way,
Oh, yeah.
So
Yeah, he was following
we'll
us
chase
live
it.
and
Oh, yeah.
he saw the image and
Everything.
could not recognize the species.
Ah.
Yeah.
It's in my Japanese film guide.
Yeah.
It's new, right?
Cool. We got like.
Tell them we have one in the fridge.
We got yesterday another
Yeah, another one
specimen.
today.
Wow,
So it's
that's
good.
cool. That's cool.
man, we'll do also our next Niskin bottle at 650.
much was the first one? Like 800?
The version is skin. It was like 987.
That's stuck and not published. Like 15 years ago, with life cycle and can be the same, but
that's real.
Yeah. But it's not sure about it.
Best part is you don't have to wait.
Do it right now.
Sinker.
That's another rocket, another Coniferous.
It is?
Yeah.
Oh.
Yeah. The bells are often at an angle like that. If we zoom in, we can look again at the somatocyst and see if it's perpendicular
Is
or
it Siphonophore?
along the same axis.
Calcoprin, yeah.
All the same, kind of the same level.
Mm-hmm. Good. Quick
Are you
look.
gonna continue or?
Yeah, we'll just get a little bit of a longer shot and then we can keep moving.
Oh.
Shedding a bit.
Yeah, I think also Coniferous moseri. I can't see the somatocyst lying transversely in the anterior nectopole.
No.
It's like a paper airplane.
The Coniferous multidentata, they have
Yeah.
different one.
Yeah, it's
different
cross-shaped.
notches.
Cross-shaped. This one looks to be alone. Okay, move on. Thank you.
All right, thank you.
Siphonophore do gênero Coniferous, provavelmente Coniferous mosari. Tem que se dar um zoom nas estruturas internas para se identificar a espécie. Estava discutindo isso com o professor Dougal Lindsay, do Japão.
Mas
parece ser
we're at
Coniferous mosari.
about 720 meters, and once we're up to about 500 meters, we'll start intersecting the deep scattering layer. That is a thick layer of animals, a lot of fish and shrimps and jellies that hang out at between like 200 to 500 meters during the day, and then at night they do a vertical migration towards the surface.
One of the hell of creators, dragons.
Medusa, probably one of the
Rob from Australia is saying, hello, Dougal.
Hey, Rob.
Rod?
Yeah,
It's Rod.
yeah.
Do you know him?
Yes, yeah.
Where is he from?
As in Rob Beeman, probably. It says debrief.
DeepPIV, yeah, DeepPIV, yeah.
Welcome back to the top.
$6.50 something.
Mm-hmm. Yeah, for our next bottle.
Oh, what is that?
Phalaenopsis, maybe?
Sure. What's that?
So far, center going down. Some type of jelly. Yep, right on the edge of the screen now. Forward.
Little fish, too.
Oh, some Mandela.
Bonus.
Two tentacles, maybe.
Hmm.
Oh.
Something that said its tentacles eaten off by a hybrid amphipod.
Oh, it's been grazed on.
I think it's that solmissus that we caught with the
tentacles,
nematocysts on the
solmissus.
outside. Yeah.
Oh.
The solmissus
This is a
pretending to be
solmissus jelly.
Solmissus. It's like I
It's
wish
had
I
a
was
hard day.
salmonella. I didn't want to be born as a solmissus.
It's unhappy.
Narcum and juicy. Narcum and juicy. Solmissus. SOLMISSUS.
Okay, or maybe the one we cut the tentacles yesterday.
Yeah,
from
the one
lab.
with the
yeah.
nematocysts scattered on the bell.
Okay. Thanks.
And the high hemispherical dome. It's a thing over here, isn't it? I've never seen them before, but
Like
yeah,
with this
all over
shape?
the place.
The-
Yeah,
the bell shape?
the nemat- always the same morphology.
Mm-hmm.
Maybe it's a Brazilian shape.
Yeah, a little more head.
That means
Tall head.
I said we'd see some
Little fish.
I think good. Good.
Yep, sure enough.
Good.
Yep, that's good.
We were doing just about the right amount of water in our house.
That's good. So no water.
Not much.
What does that mean?
40% humidity is actually kind of high.
Oh.
I think that it's not gone up. I think
Okay.
that desiccant we put in there is maybe not quite fresh.
Okay.
Got some hacks here.
Thank you.
I'm not
Thank you,
over the
guys.
housing
Good.
myself.
It's not. Oh my god.
40% humidity is a nice low number compared to what we were observing a few days ago.
All right.
But 40% of the volume was humidity.
That was a jellyfish. It had a lot of its tentacles grazed on by amphipod, so it only had a couple long ones, but normally they would be more uniform.
Have you been picking tentacles off solmissus again, Renato? You know how we sample the tentacles of the solmissus for DNA analysis? You haven't been doing it before we catch them, have you?
I collect the tentacles for drone.
Got there.
Yeah, for DNA. Yeah, I'm just saying this solmissus was missing his tentacles. Have you been swimming?
So,
Yeah, he's escaped.
Did you go swimming first? before you
Agina
went
or
there?
Solmandella, which is it?
Oh, this one. Solmandella, I'd say.
Oh, again.
What's up with you, toothpick?
It's only got one pointing forward. It's like, yeah, it's a bit of a drag. Hold on, but
Three tentacles, yeah.
That's a lazy
no,
posture.
No, it's not Agina.
You don't think so? You think it's
It could be Solmandagina, but we need to zoom to take a look, I think. There he is, it's still there. It could have also caught another jelly, because it eats jellies.
This eats jellies?
Yeah, so it could be a piece of it. It could have a tentacle from another jelly hanging on it, or that tentacle
that's
could
gross.
be embedded in the
Aegina?
jelly.
Oh.
It's
That'll be perfect. Okay.
what genus? Aegina?
Some of us have to eat.
The thing.
The shape of the bell and the medusa scattered on the
okay,
ace
okay,
umbrella
okay.
is saying it's not. I think it's a Solmandagina.
Solmandagina.
One.
But that one of those tentacles is branching off way up on the bell.
Yeah, it looks
It's
weird.
it is a mutant, yeah. It must be a mutant.
Narcomedusa.
A mutant?
Narcomedusa.
Mutant jelly.
And there's nothing
Just
on
like
the other
me.
side. Oh
What do
my
we got? We got
goodness.
8 laplets
Shortly
total?
before world
What
domination.
on earth?
He just likes to have his legs crossed. Is that so bad?
8 gastric pouches.
Yeah, we're gonna eat
3 tentacles.
And it looks way. The stomach pouches.
Yeah.
It looks bigger than the Solmandella's
Oh,
were.
Rasho.
That is
Ooh, cute little
Uau,
daisy shape in its
uma
bell.
espécie que nós não conhecemos aqui, do grupo narcomedusa, né? Que tem esses
And
tentáculos aí
Doliopsis coming in front.
para cima, né? Típica postura dessas medusas. Estamos observando as estruturas ali bem na margem, bem na borda ali da umbrella, brilhando branquinhos. Agora a posição dos tentáculos também intrigante. A posição da inserção dos tentáculos.
All right, do we have enough footage here?
We'll call it Sol Mandela.
Sol
And I
Mandela?
think, Sol Mandela. Which is, the species name is Bitentaculata. This is Tritentaculata.
Wow, Tritentaculata. She's special.
And it's one reason why when you try and describe new species, you should have more than one animal to make sure that the morphology is something it usually has, rather than having been trumped and grown back an extra arm or something.
Wasn't that the thing with like someone had a raccoon holotype, but it was actually just a mutant that had been brought to an island?
Yeah. Something like that.
There's a species of Larvacean that's called Cinerestella, something. It's basically the body's flipped, and it's the opposite direction.
Yeah.
And it's like one specimen.
That's crazy.
Collected once.
And it's just
And they
reversed.
made new species because everything's reversed. And
Mirror.
it's probably simply mutant.
Wow. Okay,
Just
that's
like
a wild
people have
mutation.
got their, their whole anatomy.
Thank you very much.
Yes, yeah.
But how is it a mutant, not actually a new species?
Ah,
to be able to
requires that you are
Yeah.
reproductively viable. And
Yeah, you have
pass
to
on
find
that.
another one.
No, you can't just tell them like, can you confirm that you're reproductively viable?
Well, because we haven't observed more of them, that's a pretty good indication that it's not still perpetuated.
Do you know, Catherine O'Hara had all of her organs on the opposite side. Rest in peace, Queen.
Good.
Yeah.
Good. Thank
That's
you.
a nice piece of trivia. Catherine O'Hara.
Yeah.
Yeah?
All of her organs are on the opposite sides.
Awesome.
Isn't that crazy?
I don't know who that is.
So Sol Mandela
She's
is
a
only
comedian, actor.
Don't know what
She
that
died last
jelly
year. It's very sad.
Bitentaculata is the only
No.
species in Sol
It's
Mandela.
amazing.
Right? Sol Mandela genus.
It's the only currently recognized
Best in show.
species. But there's more than one.
Schitt's Creek.
But
You can't tell them apart.
Home Alone. Oh,
Larvacean
let's take out
house.
that
Yeah,
larvation.
crypt. Okay, yeah. By genetics, you can tell them apart.
Moira. Yes, Moira Rose. Amazing.
With the gonads and the
I'm
part of maybe.
A house or a jelly? It's just a house, right?
I'd say it's a house.
House.
Yeah.
Keep
I'm
going.
surprised with the deep.
Just a house?
Yeah,
We're finding Sol Mandela because
All right, we'll keep.
in the part of Brazil we stood there like epiplanktonic.
We're not sure.
Yeah.
Warm waters, but here
I'm gonna
it
say
seems
house.
that we
Actually,
see
you can just
more
put
like
house
deep-
there.
They're
Sol
from the
Mandela.
Arctic, the Antarctic,
Oh, what's
There you
this?
go.
Mediterranean,
Site-
shallow coastal waters in the tropics.
Oh, and something green.
Count.
What? Well, how do we choose? I go big. I choose big.
Oh, shiny,
This
shiny.
one.
The one on the left.
What's the on the right though?
What is that? What is that?
The one on the left.
The one.
Yeah,
Come on.
By
The one on the right's the Siphonophore.
That's the Siphonophore.
Always.
Yeah.
Always.
The one on the left
Let's
was
scan it.
the dinosaur.
Oh, I didn't see the ctenophore.
It was a sedipid, yeah. It was an undescribed
Okay.
ctenophore. The one on the right, this looks like an apolemia probably.
Whoa.
So we need
And its feeding posture.
Looks like a hungry one.
Wow.
Ready
So
for a meal.
it really shows how they exploit the area around them when they're fishing.
Go for a swim.
Siphonophore agora, na sua posição de alimentação.
He senses us.
Pull in all those tentacles.
Com
Wow.
todos os tentáculos dentílios
That is a big
estendidos.
Siphonophore.
Animal bem grande.
The fuzzy look makes me think it's an apolemia.
Isso é muito interessante, como que ele consegue, né, abrir esses tentáculos e
Wow.
ocupar uma área enorme,
Oh, is that
né?
all
Então
red?
consegue coletar muitas partículas, muitos animais desse
That's
tentáculo.
very cool.
Parecido com o espinhel.
All
né?
those
Com várias,
red bits are its full stomachs, right?
Várias linhas.
Stomachs
E
and gonads and all sorts of different zooids.
essas partes avermelhadas são os estômagos, a boca, o estômago, né? Então todos esses tentáculos coletam partículas e direcionam essas partículas para os estômagos, né?
Yeah, focus on the front end.
Front end.
Fogos de artifício aqui, gente.
It is exactly a fishing net. That is true.
Focus.
How would you be in a krill or something?
Probably
Swimming
Find out,
in-
find out. It was
I mean, you're really well lined up for a scan. We might as well do it.
And probably apolemia. With DeepPIV?
Provavelmente uma apolêmia. Essa pontinha amarela
what's that little
são
thing that's
é o
next to some?
flutuador, né? É o
That's the float.
flutuador que
That
parece
tiny little
That's
amber
one
thing?
zooid that runs the
como se
entire
fosse
length of
o
the animal.
flutuador da
Holy.
caravela, né?
It's a float full of carbon
Uma estrutura,
monoxide,
zoide, né?
monoxide,
Que
which would
possui
kill
gás.
most
em
animals.
cima.
Wow.
That's for flotation, and then that zooid goes all the way to the end of the animal, and all the other zooids are attached to it. It's a polyp colony,
That's
but
crazy.
rather than being attached to rocks or branches or whatever.
Try and scan it,
It's
sit
attached
still.
to
It
itself,
starts to swim.
basically.
Yeah.
first scan.
Yeah.
All right, yeah.
Let's get it really.
Yeah.
We gotta really
So when we
sneak
start
up on
hitting it with
it.
the laser, I'd like to
Vamos tentar escanear esse animal aqui.
That's amazing.
um,
We're trying to really creep up on it so we don't disturb its feeding posture. So we're turning on our DeepPIV instrument. This is a laser sheet. We're going to record it as it passes through. It's basically going to CT scan this animal. Nice and slow.
Escaneamento agora do animal com o equipamento IRIS.
RV lights.
Opa.
Oh yes.
Agora sim.
Yes.
Keep going forward. It's a good speed.
Wow. Really good.
I don't know if my encouraging words help. It's going great.
Really good.
Looks good to me.
Oh, shifting just
Uau.
outside.
Olha, bem no meio, bem no centro.
Wow.
Escaneamento perfeito. Olha, de animal
Oh.
muito longo.
Holy.
Science camera.
Yes.
Off screen. Oh yes.
Holy.
Definitely apollemia, yeah.
Oh my god.
Well, hell no.
Oh my god.
I'm just gonna keep going. Yep, sorry.
Awesome! First pass. Right, now
That was incredible. Not backwards.
Yeah, no,
It still looks good.
Do it again.
Still in frame.
Yeah, nice.
It's just off.
Yeah, now next.
Moving a
Wow, look at
bit.
that.
Tornado.
Galactic wormhole we're going through over here.
Nossa, que
Oh
lindo
my
esse
god. Incredible.
Okay, was that fun or what? Let's do it
That was
again.
great. That was good stuff.
But let's do it one more time.
Oh, come on, Yost.
We
Most
could
of it
just
was
watch the
just
recording,
off
but doing
the
this
left
again
of the
would be more
imaging
fun.
Yeah.
Wanna do it again?
Yeah,
He's a
let's
Jedi,
one more
is what the chat
pass.
is saying.
One more pass, then we'll cut him off.
Do you want the lights on?
And by him, I mean Yost, not the Siphonophore.
Scan that blood Siphonophore will.
So as that passes by, the camera's recording the laser, so we're getting frame by frame slice images along the entire body of the animal. I hope you were recording that, Yost. Don't. That's not funny.
Good practice. All right, now for real.
Right, go slow.
Oh my god, it's beautiful.
Lights.
Escaneamento
Wow.
com Siphonophore de apolêmia.
Side view as well.
Então, assim que o animal passa pelo laser, são feitas várias fotografias
It's twofold.
que vão ser
It's in a
montadas
food
num
coma. I can't
modelo
move.
tridimensional desse animal.
This is Dr. Yeast running the laser. Thanks for
À medida
recognizing
que o animal
him.
passa pelo laser, ele fica brilhante, né? E aí são tiradas fotografias. É o IRIS, desenvolvido pelo Laboratório de Bioinspiração, Monterey Bay Aquarium.
All right. I think we got it. Right, Yost?
In here? One more.
Kakani's not here. Well, okay, one more. Okay, one more.
Just want to make sure.
Kakani's sleeping.
She'll appreciate it.
But we'll make her proud. We'll do one more pass.
I'm just going to zoom in a little bit so we can get a bit more detail on the neck to
Yeah, what time does it start? I think so. I think we're just waiting for this animal to go
well. Lined up good.
Woo.
Yeah.
Beautiful. That'll be a really good
Very
detail
nice.
on the nectophores.
Keep pushing forward. Yeah.
Now let's take a biopsy
Okay.
sample.
I'm willing to call it here. This was great.
Amazing.
Aha. You don't want to stop doing it.
Once you get a taste.
All right.
All right.
Wow.
Thank you guys so much. That was amazing.
Good.
And it's still in its posture.
I know. What?
That's
We need
just.
to collect it, right? We can just like let it be. And
We can let
actually
it be.
Very kind visitors and just
I think so.
go around it.
Yeah. Go away from it.
Amazing.
Switch on.
Oh, great. Okay. So we're almost at 650 for the next bottle, and then 250 is two surface.
Bottle
At 650.
at 650. Okay.
Yeah, so we're almost there, and then 2 at 50, 2 at the surface.
Okay.
But we don't really have samplers
All right.
left.
We're 7 time for death.
7. Yeah.
7?
But
2 hours. Okay.
yeah, I know Yost wants a couple larvacean scans, but otherwise we can also come up early.
If we come up early, do we lengthen the second dive or how's that work?
I'm not sure. Karen
Oh,
just
just
said
done.
the main object was just to fill with
Good
samples.
job. We're stopping at 6.
650.
Russ, the stereo cam, 8 hours is the max.
Okay.
Stereo cam, we're not sure. Somewhere between 6 and 8 probably.
What size?
Well, I guess it means we aren't looking for something to kill if we've only got one sampler left. We're just
mellow for you.
We're just like, you know, the take only pictures, leave only bubbles kind of thing, right?
Yes, exactly.
Can you still be motivated, Russ?
He's losing his booch.
This is not.
All right, let's continue the mosey up. A little bit of forward as well as vertical velocity, so particles are coming at us obliquely and we have time to stop.
Let's just scan the entire water column.
If you see anything that you think is a worthy target, go ahead and slow down and let me make the call after instead of the other way around.
Me either. I'm looking right now. I gotta check out that scan because we can still go back, right?
Go back where?
Do that Siphonophore.
No, I think it's a little late now.
We've got Sarah in the seat.
I mean, I'm all about the scanning, so. You gonna do more scanning?
Ooh, shiny.
Keep going.
Rosaria. Bizaria.
We got one of those, didn't we already?
Yeah.
plus the one we threw away. So there's still one in the bottles?
Known as 4M, right?
Right. Number 2 we still have? Good.
Number 2.
Good. Because we had one in the D sampler and we didn't retain it.
Keep tugging that.
So we've got 2 hours, 650 meters.
Temos ainda mais 2 horas de mergulho, agora a 660 metros de profundidade, subindo, acompanhando o movimento de migração vertical diária dos zooplankton.
Go after this. I think it's just a house.
House.
Let's see if anybody's home, given that I can't take them.
There is someone.
It looks like the body's long. Is it a fertilarid?
Yeah.
Maybe. Well,
Yep.
Yeah. Cool.
Fertilarid house.
Agora um larváceo, provavelmente do gênero Fritillaria, dentro da casa. Nessa casa tem gente. Ele está movendo ali a cauda, né? Vocês podem ver com esse movimento de cauda ele traz água para dentro da casa, onde essa água é filtrada e as partículas são retidas.
Oh, nice.
Greenish.
Yeah, it does look greenish.
Imagens bem
That probably means it's like lime or pistachio flavored.
Fingers crossed for pistachio.
Well, you know, there are some plankton I've tried, I won't admit what, but I have not tried a tunicate. At least not a Larvacean.
Nice animal. You might actually be able to see to get it in this section.
We got one suction left. We could put it in. I don't think it'll swim out. It is one of the giant fritillarids. It's not bailing from the house. Let's try and suck it up, given that it's sticking with that piece of house. It gives you a better target than if the animal's swimming loose on its own. This will be the same deal. Once it goes down the tube, we need to try and make sure we don't keep it full blast until it's against the screen.
Probably does, yeah.
Yeah.
Maybe not this species, but the genus.
Yes, but usually the researchers that give the popular names.
Has it bailed? That is the animal right there. It's left the house behind. This is probably with the tail about 3 centimeters total length.
Lasers?
Change the term.
They have the laser sheet?
Yes.
Yep.
Turns on.
Yeah.
The laser sheet can actually help
Things are
them
just
to-
better with the laser.
That's what Sarah
Can't
was saying.
tell.
It's nice to have it added. depth
Down.
perception.
Down there? Yep. He's somewhere up there.
It's time to shift.
That it? Is that it there?
Squid.
I think so.
Nighttime.
Looks like
Where's
it's
it?
moving. The right way.
Let
Yeah.
me see. Schedule. Not just a schedule.
We gave it a good try. They're incredibly difficult to get, especially when they bail on the house. That's why when it was holding on to it for a while, I was hopeful.
Trocando aqui, voltando para a transmissão apenas em inglês.
Yes, we have a Niskin to fire now at this depth while we're stationary.
Well, it's halfway.
I thought we said 650.
We want it halfway between.
Oh, she told me 650.
What was our deepest?
Was almost 900, wasn't it?
So then the middle one.
450? Okay, never mind then. 450.
Thank you,
Renato.
Renato, champion of champions.
Looks like 987 was what we tripped at, so that would make it at 500.
Even finder of lost mountains.
In terms of round numbers.
We only have one bucket left.
We have one bucket left.
If it doesn't interfere with dinner time.
So Australian, it's like robbed from Australia. It's a heavy
We could, yeah, we've got.
Oh, I bet it's robbed.
You keep giving me the pull shift. It's not as fun.
Trade with you on the first shift. If you want.
Oh, okay. I'll do that for next one. It's depending on how long it takes me.
Schedule master.
Okay. Because like I just feel I'm under pressure to make sure we make it on deck and limited in what I can stop.
I know, and I told
But I've been trying my best to make
yank
it. To yank me? Oh, this sounds really bad.
Karen, are you trying to?
Almost sounds lynchish, you know?
No, I told them they have to stop us, otherwise we'll just go forever.
Yeah.
Because we like looking at things. So,
Well, I have not been that late yet on a pull target.
no, it was, you've been just fine.
Yeah, but the shift is
Okay.
5:30 again.
Oh, okay.
Dougal, on the other hand.
Coincide with dinner, I guess.
Yeah, no, we can
Is
either
that the
take our
biggest one?
time and look at stuff until dinner is
That's
over.
all the big one.
That actually sounds like a good plan because
think it's a
when
good
we
plan,
get
'cause
going
it's
too
like
fast,
5:15 or
yeah,
something.
when we get going too fast on the way up, we don't get any good data anyway,
Yeah.
or not much,
We have an hour and 15 minutes before.
and
That's it.
that would take us to 6:30. So we just get time to grab dinner. That was a sparkly rice area.
Rice area.
6:30. So then,
Yeah.
shift comes on.
Formerly known as 4-am rise area, right? We need to give it a different name. I think formerly known as rise area is too
Okay.
long to type out every time. You gotta help me make
tarde
a
a todos
better
que
list
estão
of
nos acompanhando do
Rosarians in
Brasil.
Squid, please.
Nós fomos a 1000 metros
So
de
for
profundidade,
those who
agora
are
estamos
listening,
voltando.
this is what
them are there?
sleep-deprived
Come on.
scientists sound like at midway
We need
into a
name for this
cruise.
particular one.
Professor Renato Nagata, aqui da Universidade de Rio Grande,
Yeah.
com vocês.
I think that he's taking everything off. Dougal, are you taking everything off for the second dive?
Now we're
Oh,
getting
the equipment, you
risky.
mean? Was that a proposal, Karen? I mean, this control room's pretty hot already, but.
I know.
Yes, I am taking everything off for the next time, Karen.
I mean, you never know.
I'm rooming with Dougal. I wouldn't say that twice.
Shadowgraph for the second dive.
Oh, I understood that. Yes.
Precision of language.
Yes.
Sorry.
Hopefully I'll have the team help me with removing it, but yes, it's won't be deployed. Yes.
Right. Okay. I'm gonna stop there.
You can like feel the blushing in the room.
Warm in here.
E vamos ter novo mergulho, né? O ROV vai ser retirado da água, algum checklist das coisas do ROV, né, vai ser feito, e 9h30 ele vai ser novamente colocado na água.
One of the first dives. And there's no label in them. Is it on somebody's list or still in the place? If it's in a list, because
Os animais que foram coletados vão ser armazenados e acondicionados para estudos em laboratório.
I think we're coming up a little. No, it's a nice pace. Could probably go. The trade-off, of course, is we cover more ground when you go faster, but then it makes it harder for you to be able to stop without dusting stuff. Oh, there. Fish, is it?
No.
Yeah.
it's a fish. The way it's swimming is a fish.
Peixinho,
It's a
mas
cyclophone, to be more
o gênero Ciclotone, uma das espécies de peixes mais abundantes dos oceanos.
Are there a lot of those in the ocean?
There are an absolute abundance
Infelizmente,
of these.
peixes e crustáceos são muito difíceis de se coletarem, né? São animais que fogem
Isn't it
mais
supposed to
rápido.
be the largest fish biomass on the planet is in this family?
The largest vertebrate biomass on
Vertebrate
the planet,
biomass,
probably.
yeah.
They like triple the estimate.
Although humans
Então
are
é
doing pretty good lately.
dos tipos de peixe, né,
Does
do
anybody know
gênero
what they taste
Ciclotone,
like?
e provavelmente pode ter uma das maiores biomassas
Oh wow,
de
it's a
vertebrados
very
no planeta.
dark species, Pallador, Pseudopallida, Parapallida. I don't know much about Cyclothone
You mean
from
like
these
dark
parts.
like an evil, or?
It looks like it's thinking bad thoughts.
Yeah.
That kind of
It's
dark.
had
It is
happier days.
Probably preparing to ram the ROV
Yeah.
as we speak, hatching a nefarious plan
Yeah.
to take over the suction sampler.
Nice.
Saving all its energy for the attack.
Yeah, keep an eye on it in case it tries to free our
He
samples.
seems very stunned right now.
He'll start swimming
There's
down,
the light.
hoping to take the ROV with him, like in Finding Nemo. These things eat copepods and ostracods that are roughly the same diameter as its throat. They go for the prey items that are the biggest things they can possibly fit into their mouth and down their throat. So every time it eats, it's almost gagging as it swallows its prey, trying to fill its stomach.
Esse animal come ostrácodes
And then we'll
copépodes
just
tão
float
grandes
there,
quanto o diâmetro da sua garganta.
digesting. Yeah,
Ele
it's
come
kind of
até
like me now,
encher
doing
o seu
this, like
estômago.
eating this great food and
E às
then
vezes
like
ele
coming down and
pega
sitting in a
animais
chair
maiores
for hours on
e
end
não consegue engolir e vomita
in
esses
a
animais.
warm room.
Can see all the little light organs,
There
the
he
little
goes.
shiny dots down the side.
I realized he was being watched.
Yep,
So
it's
the next
probably had
thing to
enough of
do
this.
is we need
Esses
to
peixes
zoom
fogem,
in
né?
on the
Crustáceos
gill
peixes já
rakers
percebem
when
a
it opens
aproximação
its
do
gill
ROV
opening.
não
Yeah, right.
deixam ser
It's very
vistos
hard
pelas
to
câmeras
ID these
produtivas.
things
Okay, we can move along.
from an external morphology point of view.
I'm sure our fish representatives would be very grateful.
Moving along.
Yeah, I mean, the things they have in
Moving
these
on,
keys for taxonomists to work out what things are, look at the teeth inside the mouth. It's like, how am I going to see that on a video? Its mouth's closed.
Open wide.
That's another one of our missions, to try and find out how you
Yeah,
can
that's
tell
great.
these animals apart just by their external morphology without having to count vertebrae or do other things that taxonomists with samples can do. Because it's much better on the ecosystem if we can just observe with something as non-invasive as a camera without having to catch absolutely everything and work out what it is. And that's one of the
And
things we're
fish
trying to
swimming
do
down the
with this
bottom.
project of describing animals based on images and molecular information without having to bring them home with us. It's aspirational, to be sure, but it's the direction we should be heading, and so we're working on trying to be able to do that.
Então, dos objetivos desse
All the
cruzeiro científico é a
organisms we
descrição de novas espécies.
catch and catalog. We're trying to provide DNA barcodes so that people can use
It's like
DNA
it's a
sequences to
rhizaria,
identify them in the
probably.
future.
E
That way, if they catch
não só coletas
this is a
tradicionais,
protist,
Yeah.
mas também amostragens não invasivas, né? Com imagens de alta qualidade, escaneamentos do corpo desses organismos. E que possam permitir a descrição detalhada da morfologia desses animais de uma forma não invasiva, ou seja, sem coletar e matar esses organismos.
Can we zoom in on that one, or did we miss it?
Isn't a cholidarian a type of food?
Lost it, yeah.
No, did we get a zoom
They
in?
keep changing it. They keep changing it.
We did. There he is down there.
When I was young, it was just
We didn't
right
see
there.
the
It
spine.
was just radiolarians and forams.
If it's an inflated cell,
Now forearms.
that is the one state we never see
I
them
thought there was a difference in the show, or they're
So if you
figuring
ever
out
see an inflated cell with a
sequence-wise.
the physiological state,
Sister, like
because that's how they are
polycystines and,
regulating buoyancy. When we catch them, the membrane just contracts.
Existem dispositivos agora desenvolvidos até por esse grupo de pesquisa que coletam os animais e retiram amostras de tecido e liberam os animais depois, né? Então, não necessariamente é preciso fazer a amostragem mais invasiva, né, que acaba resultando na morte do animal.
Oh, am I supposed to be squiddling?
I think it might be from 5:30 though.
It's going to end up being a kitty nap.
I can come, 10 minutes.
Yeah, kitty nap.
Ketognatos ali, que são vermes marinhos predadores.
Medusa off to the left. I need to process animals from 3 dives ago as well.
Yep.
But
Upward
Alguma coisa passou aqui.
I'll stick this out probably.
Oh, because you're...
That's a small jelly.
Ó, uma pequena água-viva ali.
Yeah.
Arctobedema?
Pode ser
Just kind of fly through it, just so we get a little bit better
Arctopodema,
view.
que é
Don't worry about
tipo
trying
de
to
água-viva
get
traquimedusa, tem tábuas bem longas e outros
no, it's not an arco.
Getting so much bioluminescence from that.
It is
outro show de imagens agora.
That was Arctopedema. It's a repellonematid tracheomedusa.
I don't think we got that one.
Okay.
Traquimedusa, Opalonematide, vocês puderam ver aquele brilho, que é o efeito da luz que incide sobre o animal e reflete a interferência. É o mesmo efeito físico quando se ilumina um CD ou uma gota de óleo, né? A iridescência, furta-cor, furta-cor.
That is probably a fish.
It looks like a fish.
Or a Keating so we're going to skip it.
It's a fish. It's a cyclothone.
Okay.
Peixe ali ao fundo, ciclotone.
You can see the very black stomach region on the whitish grey body, and angled down a little bit head, a little bit more than horizontal. And the way they swim, they're such weak swimmers. They just do a little bit of a shimmy and like, oh man, that wore me out already.
Larvacean house.
I better swallow something.
I better swallow something the same width as my throat.
To calm me down.
Yeah, get it.
Down here, I think it's another. It just contracted.
Is that a Bathysroid down there?
We're going.
Alguma coisa ali no centro, bem abaixo ali, outra.
It's a lobate Siphonophore.
Ctenóforo.
It's looking like a Bathysroid. Smelling like a Bathysroid. Oh yes, it is a Bathysroid.
Hey.
Does it taste like a Bathysroid? Sounds like one. It's the tentacle bulbs are at the top of the stomach there, which is Bathysroid specific.
Ctenóforo lobato, esses lóbulos que ele usa para natação, né?
Rap dancey kind of a swim feel to
Parecido com a natação de uma medusa, de uma água-viva, mas não é uma água-viva, é um ctenóforo. Outro filo animal. Primeiro filo de animais que surgiu na Terra são esses organismos aí, os ctenóforos.
Big wide oracles makes me think
Animais que têm pentes ciliados.
If it turns around enough, we can get it from the side and see how long the comb plate, the comb rows are at the top of the body.
É possível ver, né, esses pentes ciliados com essas linhas.
Very nice. Here we go. They look pretty short.
É possível ver a iridescência também, né, aquele mesmo efeito que eu comentei agora há pouco, da luz incidindo e refletindo de volta aqui para a lente da câmera.
At least that's what we can call it now until we find out there's all these cryptic species. Well, yeah, you never know. Although we're close to the type locality, like Atlantic Ocean at least.
We got...
It looks a little too long in the infundibular canal to be Bathysroid fosteri.
We got this away for genetics last night. Or or was it a thalassicalus?
O professor Dougal Lindsay comenta que é um ctenóforo que pode ter várias espécies desse mesmo gênero, que são espécies difíceis de se reconhecer pela morfologia, né? Ou seja, espécies crípticas. Muitas espécies com a mesma morfologia, apenas com estudos genéticos que elas podem ser diferenciadas.
Coming back down, very good.
Here.
Yeah, so at least it's in the same ocean, right?
Well, I think we're making it dizzy.
That's good.
Do you want to wait for it to recover, or you've seen enough?
I think I've seen enough. It's good video. We got some good shots.
Yeah, that's a small jelly. We just fly through it.
Possibly an Arctopedema to our left there.
My favorite Siphonophore.
Arctopedema.
I would say so. Arctopedema.
Pulled in its tentacles.
Or is it a small Arctopedema?
Oh man.
So cool.
Wow, that's so cool.
Nice one.
Does it get
Oh, that's Soroi just went through the lighting on the other side.
Se for som inside now, and it's impossible to get inside the neck before. It's too long.
Taffy with a Y.
Right.
Yeah.
That was really cool. I think I have a video.
Look like a sparkly.
I thought I saw a copepod with two egg sacs hanging off its tail.
Really crazy how these animals can do this. Because it optimizes.
I think it would just be so much more fun if we worked common names out for all of them instead of have to learn the Latin one.
You can make them up.
Well, I do that.
Oh, okay.
There is technically the Larvaceans are not referred to as sea tadpoles, but I think it's fitting. I have a little sway in some of these terms in the group. I actually love the term copulata, which is the order.
For larvation.
Yeah, it gets rid of the argument about whether they should be called appendicularians, which is confusing because it's the group name and it is a genus name and it is a plant. Copulata translates into side or, which is the tails rotated 90 degrees from what it should be if you were a fish.
Yeah. What else that?
So in the debate between larvation and appendicularian, you're at with the copulata.
Yeah, we could replace it with
Another
copulata.
Rosarian, I think.
You know, copulata. Yeah.
Copulata. You gotta come up with the rest of the words.
To the right,
We have to
Myctophid.
rewrite, cyclophony.
Myctophid on the right.
Myctophid, there he goes.
Alright, stop it on the fish. Keep going.
Yeah.
Oh, there's a Siphonophore bottom center, just off left.
Oh, it's big.
Maybe
Don't
a
see it.
Siphonophore,
Oh, down there.
maybe
Yep.
a string.
Yep.
Maybe it's
Take the sun in there.
Okay.
Let's go take a
Ooh,
look
way
and
out
see
there.
what he is, yeah. At that kind of a size, it's probably an Apolemia.
there it is.
Looks pretty furry again.
Still processing the last one, you guys.
Oh yeah, look how long it is.
Siphonophore.
Oh, look at it. Look, it's right up the top of the screen there, the other end of it.
De Fisonect, né? Uma longa colônia, animais coloniais podem ter dezenas de
Is
metros
the
de
whole animal
comprimento.
there or just the stem?
Tá no Guinness. O
I'm just
maior
seeing
animal,
Oh, there it is.
o animal mais
Up at the
comprido
top,
do
no, it's
mundo é
not
um
quite enough
Siphonophore.
contrast there. It almost looks like just a long string
A
of
really long string.
Colidarian.
No.
Is it? I'm not seeing any
Yeah, I'm not
tentacles.
seeing it doesn't have the head on it.
Is this
So,
that 3 meter long
Nectosome.
radiolarian that I've heard about?
So what do you reckon? The bright white thing in the middle?
We
or
definitely need to zoom in to resolve this debate about what it
what
is.
it is?
Yeah, come up to that end if there is an end.
Yeah,
see what's on it.
right now it's just a string. Yeah, I don't know what it was previously.
Yeah, right now it's just.
Opa, parecia um Siphonophore, mas não é não.
It was pretty big, whatever it was.
Apenas cordão, provavelmente cordão de muco deixado, um
As
pedaço de
far as
tentáculo
these
muito
tiny
longo,
things living in the midwater
deixado por
go.
algum animal.
Just
Oh!
detritus now.
Is that a rosarian at the tip?
Imagine.
Yeah, let's see if we can get a.
Oh, stranger things have happened.
Silica filament.
We should check the other end as well. No, this looks to be mung. Because they are known to produce long strings of mucus that hang down below them.
We'll call this other.
Yeah, let's
Yeah,
just call it
just
other.
yeah, stringy thing.
Stringy thing.
It's definitely something that titles.
Okay.
Yeah,
Could be
I
an
wonder
old
what it
core
was.
of a
Ah.
Siphonophore column, the last part to break down.
There's so much marine snow or particles that we don't know its origin, like how it was originally produced and how it got to look that way. There's a whole field of science just waiting there for some aspiring grad student to come and talk to a professor who's interested
Small
in marine
Siphonophore
snow.
colony and
Uma série de partículas que afundam, né? Essas partículas de pedaços de
Is that a
do
bug
corpo
mania or
de
a
animais que
fly
são
snake?
liberados.
small fish.
No, the one that was at the top of the
E que ninguém sabe a
the
origem,
top
né?
of the
São
screen, funny
Oh,
bag.
yeah, go up to make
Nice.
Dougal happy. He wants to see what type of small
That's a
Siphonophore it is.
yeah.
E que todos
I'll
eles
even
compõem,
label
né, a
it for
chamada
you.
neve marinha que afunda nos oceanos e que
Cyclothane.
transporta o carbono produzido na superfície para o fundo dos oceanos. E que colabora com o sequestro de carbono, o armazenamento desse carbono da atmosfera no fundo dos oceanos, né? Então, esse processo definitivamente colabora para amenizar o impacto das emissões de gases, né, do efeito estufa, do carbono para a atmosfera.
Find some copepods for
Yeah,
us.
stuff gets to be when stuff gets to be too abundant to annotate it. We stop paying attention to it.
Yeah.
I mean, the crustacean component of the plankton is so well assessed with nets. We don't get the fine spatial resolution on it, but you know, it's like dragging a net through a forest and capturing people in armor. They tend to survive really well. Whereas the jellies, it's that same net, you know, with soft, trying to collect
It looks like
balloons
we got a
would probably
krill.
not be very effective.
Krill in the top left, which
Yeah.
means we're pretty shallow, right? Where are we now? 606. Oh, that's a deep krill.
606.
I'm gonna run up to dinner, but John or Russ, if you want, I'm gonna set you a plate, or I can come down and relieve you after.
Yeah, you could set a plate. We're gonna probably get out of here right at 12:30.
All right, two
Whatever.
plates.
I eat anything.
Anything?
What's for dinner?
Oh, a big scythe?
Oh.
No, it's a tomopterid,
It's a
isn't it?
scythe, it's a bug mania. I think.
Oh!
Um Siphonophore gigante ali.
Oh, no, it might be a Lichnagalma.
Lichnagalma.
Oh!
That's easy for you to say.
Look how large those tentilia are. It's shedding them. We need to zoom in on one of those little bright dots that are hanging around the back of it to see whether there are 8 little tentacles looking like an octopus hanging out the back of one of those tentilla. Well, that's a nice shot.
Is it a type of Siphonophore or not?
It's a type of Siphonophore, yes.
Yeah.
And I think the way those tentilla are floating rather than hanging down suggests, yeah, it is. It's Lichnagalma. Look at that.
Lichnagalma, segundo o professor Dougal Lindsay.
L-Y-C-H.
É sifenóforo, animal colonial,
Let me
né,
pull it
que
up
aparece
on my
bastante
amazing
nessas
lazy
regiões.
loading taxonomy
E
function in
que
Squid
funciona
here.
como são vários organismos de diferentes morfologias, né? Tem uns que
Here.
parecem águas-vivas.
Spoke of the swimming part.
L-Y-C-H-N-A-G-A-L-M-A
Você pode ver pulsando ali,
Utricularia.
né? Eles estão pulsando. Então, várias, como se fossem
Yeah.
várias águas-vivas
There's only one
juntas,
currently described.
pulsando e conduzindo o animal. Né? Então, uma natação coordenada
There's a question.
que faz com que esse organismo se comporte como esse
Now, when
organismo
these
colonial se
come up
comporte
on
como
the
um
ship,
único
the
animal só,
first thing
embora
that
sejam
happens
vários
when you
animais
try to
juntos na colônia.
preserve them is every one of the individuals in this colony tends to break apart,
Oh, again,
and you're
let's
left
go,
with
let's
just
go,
pieces.
zoom
You have
in,
no
zoom
way of
in.
knowing how many colonies you had.
And focus. What are
Yeah,
those
well,
two
except on this,
big
see that little
zoids
white
down
part
there at
sticking out
base
at the
of
end
this
is the
medusa?
float. There's only one of them on the colony here. So if you dig through the sample
Oh, yeah,
and
you
can
can really see
recognize the
those
floats, you
tentilla.
can tell how many colonies we got.
Esse é um quinidário do mesmo grupo das caravelas portuguesas, né? Então a caravela tem aquele flutuador, né, que é o pneumatófono, e esse aqui também tem, ali na pontinha, uma ponta branca. É um flutuador, né? Um organismo que é cheio de gases e que mantém a coluna do sifonóforo posicionada de forma direta.
Oh yes, yeah, that would be great. The scan would be good.
Oh, we are scanning? Okay.
Yes.
Below that.
See if we can resolve the tentilla on this. That would be like pretty hard to do, but.
E vamos tentar escanear esse organismo.
Joel, zoom out. I gotta see where it is according to. Never mind. I've got it here. I just need you to zoom out and down to where I'm at.
Alrighty, let's slice and dice.
Nice and stationary like that.
Dougal, we have one canister left. If you wanted parts of this, we could certainly put it through, and you're going to get some of it. You might not have all of it left in the sample.
And it's the last one, so we won't be.
It is the last one, exactly.
Yeah, well, I guess that's.
So let's scan and then try and get some bits.
Scan and catch. Okay, I'm in for it. I had to sleep.
And we are at.
You can aim for a pretty slow approach because it's already like washing around a little bit. Probably want to make sure it doesn't start rotating more. Rotating is bad for the scan.
I think he was listening and he's like, nope, I don't want to be scanned. I'm going to rotate.
Like, how you thinking?
Too high.
Just the way those tentilla are floating up above the.
I was doing a
tentacles
pirouette
like that
for it.
is very
It's not-
distinctive for
not
the animal.
really.
It's not really. You don't even need to see the shape of it.
Always wanted to be a ballerina.
Just didn't have the right shoes.
Yes, aspirational.
Respondendo ao chat,
Not a
temos
big job market for
narradores
it.
que
It's like
falam
wanna
espanhol,
be a
mas
ballerina.
agora eles não é o turno deles. Eles já, elas na verdade, né? Professora Silvina Bota e Julieta Ceburrar, que vão vir em breve para narrar em espanhol, sim.
I need to annotate that we're DeepPIVing this, don't I?
Spanish speaker on team right now. Okay. Here we go.
Wonder if we're still getting this in the stereo cam. We probably are.
Who's working that?
Not in the room. We
We're-
have to,
we're- yeah,
we have
we're
to
not
call
o-
somebody.
over there.
Does he speak Spanish?
Oh, not Spanish. No.
No. Portuguese?
Just Portuguese. That I'm aware of.
Yeah.
Maina. So that
When you think about a surprising number of scientists are polylingual or multilingual, how you define it.
Maybe a surprising number aren't. As well.
I can say, can I please have a beer in a lot of different languages?
We are shocked.
Only a scientific application.
Mm.
You learn a lot from the locals.
Здравствуйте, меня зовут Dougal.
We can get that bottom bit to next pass.
Should be good on the way back through. Ooh, that looked like, was
was
that
pretty
framed?
good. Was that in the frame?
Yeah, it was in the frame. It was a tad speedy, but
Mais um escaneamento sendo feito aqui com
You get what you get.
o iris,
Tell me you wanna know the scan.
As my children learn.
que serve para descrever a estrutura morfológica desses animais tridimensionalmente, né? Através de várias fotos que são realizadas ao momento que o animal passa pelo feixe de laser. Vários frames são tirados e depois um aplicativo no computador vai fazer a montagem tridimensional do corpo desse organismo.
You need another laser sheet that shines forward to define where the back of that area is, so you know when you're off frame.
No, I want to shine it into an off-axis parabola so it becomes like just
yeah.
a straight beam.
Yeah, yeah.
Usually you do the opposite to focus with a parabola. Because we can point the laser up and do this, and then we just have like one more or less straight thing. We don't ever want to put a large port on.
Is that like sticking the flashlight under your jaw?
Not quite.
I've got a feeling I've seen you do that before too, which is a scary thing.
That's what you always do when you tell the ghost stories of the campfire.
Can we try one more pass?
Yeah.
We always say one more.
Yeah, yeah. I've noticed.
In mountain biking, you never say one more because that's where you crash. So you always say two more, but then you stop one short.
That's very smart.
Oh,
That
yeah.
was fast though. That was fast.
Yeah.
Mais uma passadinha para mais
Faster to
descanamento.
slow out.
Ah,
You've just got one hydraulic setting, right?
Zoom in on you.
On your thrusters.
Well, we have the gains that we can.
You're zooming in?
Yeah.
I've zoomed in.
What they did in Ambari on some of the vehicles is they engineered like a
Yeah.
hydraulic bypass. So when you got into this kind of mode, you'd switch to the bypass so you couldn't put too much force
50 frames a second.
in any movement. It basically buffered
Boosting that to
your
120,
motion.
or is it too much?
And it actually works pretty well when you're into this kind of delicate mode.
And honestly, with the speed
And it's not that hard of a feature. You just need, an actuator to switch the hydraulics off.
just out there.
Agora, pessoal, a Julieta Seburrar, da Universidade Federal de Rio Grande, se junta a nós para transmissão e em espanhol também.
Hola, buenas tardes. Buenas tardes, buenas noches. Bueno, estoy intentando actualizarme para ver qué es lo que estábamos haciendo. Ahora veo un sifonóforo en pantalla. Lo estamos intentando escanear nuevamente para poder tener una resolución completa y una imagen tridimensional.
Is that framed?
Y después, junto con el iris, vamos a poder sacar diferentes imágenes a lo largo de este organismo para poder reconstruir tipo una tomografía, como lo que parecería, como lo que sería en versión así humana.
This is one big reason why we don't open that big black house again.
Vimos varias cosas hoy a lo largo del día, muchas medusas, muchos larváceos, no sé quién nos está acompañando ahí desde más temprano. Y bueno, creo que nos quedan 2 o 3 horas más de rock sumergido y bueno, después lo vamos a levantar y de noche vamos a hacer otra inmersión. Así que vamos a tener fauna submarina por un buen rato.
About to get that one right there.
We can only torture the pilots for like so many minutes at a time before they are allowed to wait.
Ross está acá haciendo algunos chistes. Es bastante difícil que el láser pueda pasar por todo el organismo.
Yeah, let's suck it
Hay que
up.
tener mucho control,
Once we get
mucho control del láser, porque obviamente hay corrientes, obviamente el organismo también se mueve. Estos sifonóforos son organismos coloniales. Son organismos coloniales y hay una parte de estos pólipos tienen la función de locomoción. En cuanto a los otros, tienen una función alimenticia.
Psychological warfare.
Creo que en vez del láser, vamos a intentar pasar a la succión. Es bastante difícil intentar medirlos o capturarlos con el láser porque tienen que quedarse bien quietitos. A ver si tenemos un poco más de suerte con el succionador.
Bet you there's like 25 different species of raccoons. Yeah, right.
I think our best chance of finding new species, given that we're all sleep deprived, time deprived, etc. on these kinds of cruises, is you sequence them, you take a voucher specimen and then you go back to it and look for the morphological differences afterwards. No worries.
Uy, parece que sí, sí. Tuvimos muchas capturas hoy.
Okay, wash the canister.
Así que me imagino que una vez que suba el robo, vino. Medio desconstruido, pero vino.
Okay, it has to stay like that. Do not rotate it.
Así que vamos a tener bastante trabajo de noche.
Two.
Thank you.
Sampled in SS2.
We are.
Part this. So we are at 600 meters almost, on our way up.
Our buckets are all full?
Our buckets are full. My heart is full.
I was going to say the same thing.
Todos van a estar muy contentos después cuando suban las muestras.
Your stomach, it is
Tenemos
dinner time.
representantes de todos los organismos para todos los investigadores que están arriba del Falkor 2.
Swing left, we've got a Siphonophore. Not that we haven't had our fill of them so far this time.
There's a lot of Siphonophores, really.
They are.
Sí.
Very few ctenophores. It's an Apolemia.
Otro sifonóforo gigantesco.
This is really common, this species, around here.
Now the Apolemia typically fragment more easily, right, than the other genera?
Yeah.
Well, the other large genera.
Distributed chlamydia directly attached to the stem. Oh no, I think bunches there.
Como podemos ver, los diferentes individuos que componen este organismo colonial. La parte superior son los que están encargados del movimiento. Y a bolinha vermelha.
Right down at the bottom? Or there?
Cada uno de estos individuos que forman la parte que está encargada de la locomoción se llaman nectóforos, y como vemos ahí en pantalla, esa bolita roja es un
So this is not random in its organization. There's a repeating set of types of polyps that just continue
que
on and on.
auxilian en la alimentación.
Bom, então vocês podem ver aqui, voltando para o português, o final da colônia, né, que é o Siphonophore. É um estolão de onde brotam vários indivíduos, né? Esses indivíduos têm funções diferentes dentro da colônia. Tem indivíduos que são especializados na função de captura de alimento, que são esses que possuem tentáculos, né? Então esse tentáculo, ele leva o alimento para a boca, que se chama gastrozooide, esse tipo de zooide que tem a função de alimentação. E ali já agora na ponta da colônia, brilhante é o pneumatóforo, que é o zooide responsável pela flutuação da colônia. Atrás deles, esses transparentes, são 5 ali, 5 nectóforos, que são como águas vivas que ficam juntas, né? E que fazem a locomoção, a propulsão da colônia.
Bueno, acá,
Okay.
acá como nos contaba Renato, este organismo colonial está formado
Another
tanto
one
por los
lived to fight another
individuos que hacen la captura del alimento, que son todos aquellos que tienen tentáculos y están distribuidos a lo largo de la colita del animal colonial y llevan o transportan el alimento hacia los gastrozoides. Un montón de palabras difíciles que estuve aprendiendo desde que llegué acá. Que son los encargados de hacer la digestión. Y en la punta, como habíamos visto, los neumatóforos que están encargados, eran unos 5 o 6 encargados del movimiento. Que tenemos acá en pantalla, un larvacio tal vez.
Yeah.
Alguien va a estar feliz,
I
tal
think
vez
I've read
Ross.
that with my kids. Isn't it? No, it's a hungry caterpillar, sorry.
Ah, sí, es un
It's been a few
larvacio.
years. It's an oik, I think.
It's got those bumpy
yeah.
things on the end.
But the
Como
trunk,
ya
the
vimos
trunk
otros días,
is
los
nice and
larvacios
bulbous. Really nice. You can zoom in on this part.
Son cordados, igual que nosotros, son parientes
No, this-
lejanos.
I don't see any bumpy parts.
Pueden
This
ver
is pretty
ahí el individuo,
symmetrical.
que es como un pequeño gusanito en el
This is
medio
probably like
de
a
esa bola.
flora proper.
Esa bola es un mucus secretado por el organismo que ayuda a filtrar el alimento del cual él se alimenta, y una vez que esta casita que ellos forman está sucia y completamente llena de detritos, la descartan y comienzan a secretar una nueva.
So for those who are online, to place this in perspective,
Estamos
we're
vendo ali
making the pilots try to
larváceo,
hold position
cordado,
of the
que
vehicle
produz
bigger
essa,
than your van,
essa bola de muco, né? Ali no centro,
looking at
ele tem
something the
formato de um
size of a
girino.
marble. And we're expecting them to be able to maneuver this with kind of centimeter scale resolution.
In a current as well.
In a turbulent.
Well bouncing on one leg with one hand tied behind their back and an eye covered.
It's pretty bugs, right?
E esse larváceo, ele produz essas casas. Essas casas, elas ficam cheias de muco, né? Porque o animal transporta
So you can see the
água
animal, the white kind of
e
spherical end to the upper left, and you could see something iridescent, which is the beating tail in there, and then the rest of the structure are the food concentrating filters that are then attached to the animal's mouth by a feeding tube.
o professor Russ Hopcroft está descrevendo, né, o processo de filtração que esse animal realiza. A água entra na parte anterior e as partículas são retidas.
Oh, no, he scaled it out. Yeah.
E as partículas retidas são ingeridas pelo animal, né? São aderidas no muco e engolidas pelo animal.
Here we go.
You're ejected. Pilot ejected.
Thankfully, they're prepared for that.
Yeah.
Oh, look that chair!
Like
My goodness,
trying to
we
get
are
in
going
our-
to the beach now?
Yeah, trying to get our
Although
sunny
the
side.
expectation is in
Although we
deep
haven't seen
water
the
that they
sun
don't
for a
turn the
Tenemos
hoses over that
acá a
frequently.
Manu,
And they
que
are a little bit more
vino con una
energetic and
nueva ropa,
costly-
una camisa
costly
súper
to make.
hawaiana, súper floreada.
Down
Solo
there.
alegría.
It's not too late. Probably missed it. Let's just keep going.
You want to? Not yet. I go eat now?
Okay.
No, I can
Welcome
go
back.
and then.
Thank you. How was dinner?
I just
Oh,
realized.
what's for dinner?
And then yeah, I think the black shiny is definitely a Cinderella. It is a foran. It is not digitata.
Classified Siphonophore. Have a coffee in there.
Aubergine.
Stick it.
Bunch of good stuff in the salad bar.
Here.
We're never gonna start.
All right. I worry about that every time I go to sea. Small jelly off to the right. I'm not sure what it was. I'm assuming like Arctopodema. Larvacean house to the top, keep going.
Bueno, ahora es horario de... estamos viendo una pequeña medusa en pantalla. Es horario de la cena número 1. Los voy a dejar acá con Renato y después de la cena volvemos con la transmisión en español.
Right over here. It's just a sinking house. You can keep going. So, last night, when I told you to find something amazing, we did. So I'm going to ask you the same thing.
Oh, okay.
All right.
We're going to try.
We find something amazing all the time. It's like a scavenger hunt every day. It's like playing a video game where you're searching the room to find the treasure.
Yeah. Pokemon Go. Yes, it's kind of hard to get a community composition because your eyes get adjusted to like looking for Siphonophores and that fuzzy long thing, and then it's like, oh no, we should be looking at more spherical things because we've been doing lots of Siphonophores lately. I mean,
Yeah, you do develop a search image. Like, what is that little thing? It was a small jelly. It's okay.
Okay. Thought for a minute it might be a Tuscarora, but it was not.
So how do I know exactly the medusa? I see one or the other medusa.
If they're swimming, it's because there's a Physoneck there.
There are 2 Physoneck. You gotta choose, dude.
2 Siphonóforos agora aparecendo.
Yeah, let's go for that one.
2 Siphonóforos do grupo Physoneck.
This is pretty lightly built compared to many of the others. Are we not getting something like Rudjacovia here, or it's just not common?
I think I've seen one that I would sign off to say it is so far.
It's like in my latitude, they're the most common, right? Okay. And at Ambari, it's Nanomia that's the most common, right?
Yeah.
Straight through when we're done. Oh, we're not done. Never mind. I thought we lost. Well, we can be done if you'd like. You got enough on that, Dougal? Not quite.
Mark.
Oh, yeah.
Look there. Something behind us. Go up a little bit. Just go up and forward.
Okay, thanks.
Okay.
Should be able to grab it. You have to check if the neck before is
Good call, though. You never know. Things sneak up on us.
Right there.
Siphonóforo pequeno esse aí, né? Não é grande como outros que foram vistos até agora, mas precisamos dar uma olhada de perto para saber se realmente é uma espécie já conhecida ou se pode ser alguma coisa diferente, desconhecida que merece uma amostragem.
Of interest.
Moderate.
Agora ali na frente uma casa de um larváceo, né? Larváceo ou apendicular. A casa muitas vezes ela está vazia, né? Porque o animal, quando se alimenta e muitos detritos ficam aderidos à casa, às vezes eles entopem a casa e é o momento em que o larváceo sai da casa para produzir uma outra casa de muco. Então essas casas de muco que vão sendo abandonadas, elas afundam e compõem, são um grande componente da chamada neve marinha. E essas casas de muco são colonizadas também por outros animais, copépodes, outros crustáceos, e acabam se tornando o habitat deles, né? E acabam colaborando muito para a coagulação de partículas, né? Então elas vão agregando partículas e vai se tornando cada vez mais pesada e afundando com mais velocidade. Então é um processo que colabora muito, né, para o afundamento para a aceleração do processo de afundamento de partículas. Em alguns lugares dos oceanos, onde as partículas são muito abundantes, esse processo pode ser bem rápido.
It's the first one of the first spheronectes we've seen this cruise?
Yes.
Siphonophore acima aqui, ó. Agora subiu.
So you can see the curvature, the curl on the tail right now. It's not pumping actively. The house is still pretty inflated though, so it must have been swimming recently.
Então, dentro dessa casa de muco, há uma trama de cordões de muco que são secretados, e o animal secreta esse muco e depois engole.
Yeah, we haven't not seen a lot of animal load on the houses.
Yeah.
That said, there's a small copepod right here in that lower corner.
All right.
It's just you aren't paying attention to the details. If it's not, it doesn't look like a Siphonophore.
Então, como eu comentei, essas casas abandonadas são colonizadas por vários animais, né? Então, o pontinho branco ali é um copépode que está ali, né? E se alimenta do muco e de partículas que grudam.
Copepod say, "Thank you very much for collecting my dinner." It's chomping away on those particles.
Okay, that's probably about as good as we're going to get. Well, it's not moving, so it's not. But you did a wonderful job of coming in without disturbing the house.
Oh, the scythe's still there.
Your scythe's still there.
It's
You
still
want to
there.
zoom
Yes,
in on
yes,
the
yes,
scythe
yes,
while you
please.
still got everything set up?
Yep, yes, please.
There we go. That's an easy. Just after you started to lean on the thrusters, right?
There it is. It looks like a spheronectes. You need a shot from the side. It's coming in hot.
And there's the house. He bailed out of the house above.
Oh.
So we still are after the Siphonophore.
That's our scythe. Alrighty. Photobombed by a Solmissus or something there. Wow, when it pumps on that bell, the top looks almost pointy. It's such a rounded nectaphore swimming bell. That's kind of cool.
I don't have to speak Portuguese, but I need like a pause break before.
Oh, sorry. Tomopterid.
Good.
That is array 1, right?
Oh, I take it back. It's not a spheronectes. It's a single-belled polystriate lensia, maybe?
Well, it's entirely possible. I substituted
it's a
when you weren't
polystriate
looking.
lensia. Oh, wow.
Yeah. I was just listening.
Yeah, it's a Calicophora and Siphonophore. Man, that's one crisp zoom. That's
Still need closer, Dougal?
I need to go closer, like way up, even if we lose it.
Agora um sifonóforo do gênero Spheronect, né? Esse aí já não é tão comprido como outros, mas também é um animal colonial menor, né? Um único propulsor apenas, né? Esse aí não possui flutuadores. Apenas o propulsor ali no topo da colônia. Um único indivíduo que faz a propulsão e que carrega vários outros animais, né, que se parecem com pólipos de quinidários que ficam ali na parte posterior da colônia.
Definitely multi-ridged. Thank you. That was not a Spheronectes, it was a what? We need to make a...
Na verdade não era um Spheronectes, né? Provavelmente é do gênero Lancia. Grupo calicóforo dentro dos sifonóforos.
Calicophron, yeah, a praying calicophron there.
Another house, we don't need to stop on that tortured you with enough of those for a few moments. And we have...
Whoa. Oh, what was that? Was that a cell?
I looked away for a second, I missed it. To where? To the right?
Kind of past behind us.
Yes.
Oh, okay.
Keep coming up.
Oh, yes. Siphonophore.
Okay, thank you. Moving on.
Yeah, see when I do it. So the Selps are the other major class, for lack of a better word, in the Tunicata, which is where the Larvaceans also sit. They are considered protochordates, the precursor of us. Their claim to fame is the buccal cavity, the extraction of food in the buccal cavity, which is now what we call our mouth. And gill slits, which appear in fish, and we have lost over time, except in sea hunt. Never mind. Yeah, Sequest, okay. Right, where they had the man with the lungs, and man from Atlantis. So these simple animals are part of the trajectory on the way to the fish and all other animals with vertebrates and backbone.
Share a common ancestor.
Everything shares a common ancestor. Some level.
Not really a higher organism, we are just...
So on the chat, depending on the part of science you go into, you don't need great math skills, but you need good logical skills. There's quite a variation in how good you need to be with actual higher order math. Yeah.
Either a Siph or a Squid. It looks Siph or Selfie. It might be a Doliolus.
It could be a Doliopsid colony. That's my prediction. So we can throw away the giant jellyfish for this. Right, Dougal? That's the same Selpa aspera we just mutilated a few minutes ago. It looks like it's got a tear in it.
Which one do you want? The middle one.
Yes, the middle one.
Yeah.
Oh, it does, yeah.
The other one's got a bite out.
Isso é uma salpa que está à esquerda, né? Um animal bem encordado que é próximo dos vertebrados, né? Na árvore evolutiva dos animais. E as salpas têm um papel muito importante também nos oceanos, que é o papel de acelerar o afundamento de partículas, né? Uma vez que elas fazem muitas pelotas fecais, e o cocô delas, que são as pelotas fecais, transporta energia para o fundo dos oceanos.
Yeah, if we can frame this up, brighten it up just a little bit. Yeah, I'm falling off the legs. This is one of the groups of the tunicates that we really have very poor appreciation of the life cycle. This family does something different than the other Theliaceans, which is one of the 3 classes inside the phyla. And we don't quite know, we've never seen the individual that produces these chains, or every individual can produce a chain. We just don't know.
Então a gente está vendo aqui um tipo de talháceo, né? Então são vários organismos que formam um cordão. Que nós estamos vendo, parece que 7 indivíduos. Esses indivíduos, eles são produzidos por um outro indivíduo que se chama ozoide, mas segundo o professor Russell Hopcroft, esses ozoides dessa espécie não são conhecidos.
Ignore the man behind the curtain. Beautiful, beautiful.
Did Russ just say go the man behind?
Just crisp up the focus.
Sure, that's what he said.
We're fair ways out, so that's part of the issue right now. A lot of water and particles in between.
Any copepods or anything attached to
that's
them?
very pretty.
Any jellyfish polyps in the mouth?
Absolutely wonderful.
No, but Emily put it like for me. So I'm okay. Thank you.
That's a nice
Thank
one.
you.
That's just so weird being attached to each other by a stolon like that, and they're not pumping at all.
They don't. They don't pump strongly. I think it's entirely ciliary motion that keeps the flow going through them.
So they just kind of hang out and float without.
Well,
do they reproduce at this stage?
Let's see if we can get a little closer and crisper, a little bit more detail.
Esses animais, eles não pulsam com musculatura.
If you collect these with plankton nets, the problem is the tunic is really, really sticky, and all sorts of stuff gets stuck to them, and invariably they get rhizaria stuck to them, or vice versa. And I spent a long time trying to decide if this is a feeding relationship or it's just the fact that they're both sticky as heck on the outside. And they're mutilated enough that all you can recognize is the digestive tract in the animal, right? Is that badly damaged?
Yeah.
So I've known since the time I got to Alaska that we had a tunicate that was unidentifiable. And it's only when we got some ROV time and started doing shallow deep water nets, fine mesh, good weather, that we started bringing them up intact.
You can see their brains.
Brains?
So the digestive tract is that, in this image, it's the yellow that you're seeing. Okay. I think that's probably as good as we're going to do. They're going to get dusted in a second.
Esse é um animal que coleta partículas através da ciliação da faringe, né? Então ele bate os cílios e transporta água. E essa água que contém partículas é filtrada. E assim o animal coleta uma série de partículas em superfícies adesivas.
This is too big to close your eyes, my friend.
I can't do it. You can't make me do it.
Close your eyes.
You might do it if money comes down to relieve me for dinner. Otherwise I'll be yelling stuff out.
Small jelly off to the left. Keep going, and a nice house off to the right corner.
Oh my goodness. There it is. Oh, is that the Aquarium? Oh.
What? Something exciting going on back there?
You would not want to be a copepod or a krill trying to swim through that. That is phenomenal.
Yeah. Curtain of death.
Repellent nematode.
Is that a small? Okay.
Jelly?
Yeah, it's a little
Yep,
repellent
and
nematode.
another jelly I
That's a
can't see moving.
That's Helicreus, I think.
It might be. I thought I saw a bit of a pointy end. It's That's right. So the tunicates, as well as this mucus and gill slits we talked about, have not a precursor of the backbone, the notochord, and that is what runs down the length of the tail and provides the rigidity for the swimming. And in the salps and the doliolids, it's greatly reduced and doesn't provide the same structural value.
I mean, there's steps.
I took a few steps just now.
Just getting rid of the noise, right? Oh, is that a
Lobe
thalassocalyx there?
of some type.
It looks like a thalassocalyx.
Ctenóforo lobado agora.
Smells like a thalassocalyx.
Parece ser
Okay, this
Felacocalus.
is probably our last target before we're going to have to start the recovery process and the bottle that we need to do at 500.
Temos apenas uma garrafa ainda para coletar animais, então talvez esse seja o último alvo que vai ser coletado antes do Rob voltar.
Oh, that's a long one.
They have been photobombing the last couple of things we've stopped on.
I know. Maybe it's time to focus on them.
If you look closely, on the end of these arms are little cameras, and they're doing selfies.
What?
I think Russ is
Yeah, so it's a selfie of it with the Siphonophore. Yeah.
I was wondering.
Not yet.
Do you want dinner?
I think a plate's been put away for us. I will be gone. Oh, there's the rigided, the branching.
I'm leaving now.
Canal.
You can see me leaving my seat right now.
Yeah, I can see.
Thalassocalyx, I believe.
Is that a ciliary band of some kind?
It's got a ring muscle that closes the medusa-shaped
No, but
body.
the ciliary band follows that entire topology.
So that's the cone plate, and then the meridianal canal is a flat tube with cilia in it. Like a tube that takes the gastrovascular
It still
fluid after
still amazes me. I mean, we get the young ones of these. Yeah.
Esse é um ctenóforo que tem uma natação diferente dos outros ctenóforos, né? É um ctenóforo que pulsa como uma medusa. Então ele tem uma musculatura que contrai e realiza esse movimento ondulatório, né, como uma medusa, ao contrário das outras espécies do grupo.
Okay, everybody, this is Risaria time.
Yeah.
Agora o pessoal dos protozoários apareceu. Mas todas as garrafas já estão cheias, aliás, tem apenas uma. Né?
One of the sparkly Risaria we've been.
Here we go. I got a common name.
Sorry,
for
there was a selfie moment. The sparkly Risaria that we've all been seeing. We just imaged a sample that came in from a dive last night. So it's definitely a foram. I think most likely it's
the
Hastigirella.
canals, the branch canals. I don't know if that's where the gonads. To me, that looks kind of like gonadal tissue. But Dougal will know the real answer. I'm speculating.
Are you going to scan this one? Que imagem linda desse animal. Pentes ciliados dele batendo, né, gerando esse efeito da iridescência. Parece que é o chamado furtacor, né? É um...
Alright, so if you look at the iris feed, you guys are going to get to see something really cool.
Is that meaning you want to iris it?
No.
No, they're going to show us. They're going to pipe the output from some of the scans today, so people can see what we've been doing, all our hard work and suffering.
I guess I'll do it.
Okay, we're done with this animal? We don't have any place to put it, so my
No.
suggestion would be, while they're playing with switching that feed, there we go. Is that being sent out, or is that just us local?
Well, it's on the Everts, but that's up to Alex.
I can show that up. Yeah. One sec.
Yeah, that's pretty wild.
Yeah, I wouldn't want to be a fish swimming through that, let me tell you.
I've always thought them as really comparable to like long line or even gillnet fisheries.
On steroids.
At a micro scale, yeah. Microfiche.
Alright. So I can put that up.
Yeah, so I missed the scan, but I heard about it. You guys scanned with DeepPIV one really big Siphonophore.
Yeah.
And Yoast quickly took that data and converted it into this.
Do we want to stay on this, Russ? Or
Hmm?
we want to stay on this?
No, we can go. Let's try and get up to that depth for the Niskins before we start the pull.
Okay.
Because I think we're almost out of time. I think I was told we needed to start pulling
And
it.
then we'll spend a
If not now.
How much are we firing?
Are we showing this in the stream?
We're firing one bottle at 500.
Yeah, so if you,
our shares with everyone.
We should only have one bottle tripped.
Are we showing
Yeah.
the scan in the streaming?
Only one, number one has been tripped.
Agora vocês estão vendo
Yeah.
resultado do escaneamento desse do iris, né? Um sifonóforo. Vocês podem ver toda a trama de tentáculos que ele produz. Essas colônias podem ter dezenas de metros e ocupam uma grande área, né? Permitindo com que o animal capture partículas e animais, zooplâncton principalmente, né? É um animal carnívoro e que otimiza muito, né, o processo de encontro e captura de organismos através dessa trama de tentáculos que pode ser vista. Um lindo modelo tridimensional de um sifonóforo.
I can't believe I was there
It's still a work in progress.
almost 30 years ago.
Seria bem parecido com uma linha de espinhel, né, com milhares de outros anzóis, né? Então é um animal que utiliza essa estratégia, né, de captura de alimento, e ele tem essa posição estendida dos tentáculos. Quando ele está nadando, ele retrai todos esses tentáculos para o centro da colônia e assim diminui o arrasto causado pela natação, né? Então dessa forma aí, como ele está numa posição de pesca, né, como podemos chamar.
Sorry, I was reading a comment. Someone said, "Why do I want to go eat some shredded wheat?" Because of the marble.
Probably a house.
Oh, a house.
Oh,
Oh, slow down.
Well, we're gonna be able to sense this one.
Oh, hurts so bad.
Hurts so bad. Can't do any. Oh, this one has.
I really
speed
wish I had
instead
my
of force
Shadowgraphic
line.
system along to be getting me some of the data on this. I have one that does about 250 liters a second.
How does it? Is it just open?
It is in shallow water. I can do in full autonomous undulation to about 150 meters. So you play your wire out, and you just babysit it, literally.
Okay.
We've deployed it to 1,000 meters with a reconfiguration, but at that point, yeah. No, I've told him he's going up to fire a bottle, so I'm not going to stop. In that mode, we have to pay wire in and out. We have to slow it down significantly, which reduces the volume filter. But it allows you to get distributions of various protists through the water column, as well as the metazoans.
Oh, yeah, that's really
Well, it's
small.
about 50 micron pixel resolution, so larger protists, it does fine on. I showed you some images from it, so it's good for all the larger stuff. You get up to 0.5 millimeter and larger, you can tell what they are.
Yeah.
But it's kind of like doing deep CTDs. The deeper you start to work, the more time it takes, the less your colleagues appreciate the time you're taking doing your science.
Agora estamos a 500 metros de profundidade. Estamos subindo E vamos entrar na chamada camada de dispersão acústica, né? Uma camada onde se concentram organismos e que pode ser observado através de equipamentos de acústica. E nessa camada esses animais eles acabaram de realizar a migração vertical e estão ali próximos já de uma região que começa a ter influência, né, de maior concentração de partículas, onde eles vão se alimentar. Nessa região da camada de dispersão acústica, a maior densidade de organismos, né, é observado, uma vez que os sinais acústicos que são emitidos pelos navios, né, se dispersam. E ali muitas vezes se concentram organismos zooplâncton e também peixes que se alimentam desses organismos zooplâncton, né? Então, uma área de agregação, e que é uma área sobre a qual se direciona muitas vezes a atividade pesqueira, né?
Just the material being sloughed off them constantly.
And breaking up into smaller.
Yeah, I mean, we always think marine snow that like Larvaceans and tunicates are major producers, but okay, let's trip that bottle.
O professor Hus Hopcroft está explicando, né, que existe uma série de animais que produzem partículas, né, pelotas fecais, pedaços de corpos desses animais, e produzem muco, né, substâncias mucosas que vão agregando partículas, e essas partículas vão adquirindo maior peso, né, maior biomassa, e afundam com maior velocidade. Então esse processo de coagulação da matéria orgânica é um processo contínuo, né, de agregação e afundamento dessa matéria.
Okay, so it's this one in. Okay. Number 3, just hold 0 out on that. Okay, fire. Let's move in. Go in.
Yep.
Mook bags don't exist.
Okay, almost done.
All right. Well, as they say, up we go.
That was bottle 2?
That was bottle 2.
Thank you.
And the sky is the limit, so to speak.
Agora acabamos de fazer a coleta de água através da garrafa de Ninsky. E essa água vai ser analisada também para se descrever parâmetros, né, biogeoquímicos da coluna d'água.
You know, people on the chat drop off. I start telling jokes. Yeah.
What's our label for miscellaneous?
Hmm?
Do we have a label that things that is not obvious?
Coded taxon.
Yeah, where is that? Oh, all the way. Okay, good. Guess this is one way of labeling all the rhizarians.
Okay, so I think
Not
at
enough
this point
resolution.
you are supposed to be on deck at 7-ish?
Yeah.
Oh,
7:30?
Sinking
Supposed to be on deck
threads.
hour?
7.
Okay, well then, I guess you need to go into a maximum uprate.
Maximum uprate. We have more Niskin bottles to fire too?
We do. We need to stop at 50 meters.
Yeah.
And do 2, and then the last 2 will be just subsurface. There was another Coelodendra and another. I can't tell if that was a house or Thalassachilus. We just touched it. So up as fast as you can go. Make haste.
Então agora subindo aí um pouquinho, uma velocidade mais alta para recolher o ROV para um novo mergulho depois mais ou menos lá por 9:30 da noite.
All the jokes, yeah. My children say I have Muppet humor.
That's the best kind of humor. Waka waka waka.
That's a good description.
Or dad humor is the other term that seems to become associated with it.
That one's more common.
Yeah.
Muppet humor is.
Yeah, well, I mean, the Muppets have kind of been off the air for a while, more or less, or out of visibility. Yeah, they had a special.
It was good.
It was, because they were talking about it turning into a series.
Oh, Calicóforo.
Siphonophore. Bison or not bison
Calicóforo.
Yeah.
Siphonophore, Calicóforo apareceu ali.
It looks like a rocket.
It's fast.
Never actually, the Fisonect I understand. The Fis has to do with, the nect is, Fiso means like long, right? I think it comes from, not philo. Philo would be leaf. Fiso, I think, has to do with long, like, or breathing. And nect is, I'll have to actually look at the breakdown. Fiso, I haven't, Calicóforo, I haven't actually tried to do the breakdown on.
What is it then?
Well, foreign is to bear, a four is to bear something. And then an is to be the bearer of. But Cali, I'm not sure what Cali is. Hey, I've got this amazing technology here called my phone.
You can even talk to it now.
I could?
Yeah, I mean, that's what the kids do. They don't type.
They don't type?
Yeah.
They can type unbelievably fast. I don't understand it.
Anymore. They can just talk.
Multiple potential origins, including beautiful Norman Frenches.
What's the software you use to analyze the
Bridge RV.
Same root as in Calypso, which can mean
Bridge RV.
Yeah, I'm on Bridge channel.
so to bear beauty would be Calicóforo, something along that line, or the
more.
Well, they are kind of ornate.
Display
and
not
glass-like.
not needing that program.
Yeah,
stuff.
compared to the
more friendly.
Bridge RV.
Yeah, we're on Bridge channel, Sunny, no ROV.
Bridge RV.
Oh, that's
Talking to Jan, we can do a calculation to show just from a photon flux, what does that look like? And it should also be a little bit asymmetric.
You can also see the ropes that are on the line. So you get like a couple lines.
Oh, medusa.
Oh.
Yep.
Oh, it's gone.
I'm trying not to look because I look at one star and I can't.
What are we labeling unknown medusas as?
One thing I can tell you, you can just oh shoot, you have to get whatever's the highest part of that.
Okay, where would that be?
Liberia.
Yeah, medusa.
Maybe medusa isn't a group.
Oh, normally we announce the end. Well, yeah, we started pulling. We are out of time. So now we're going up too fast to be able to identify stuff easily and accurately.
Okay, another rhizaria on the left. So you know, the funny thing about everything we have collected, for it to be Hestigerella, it does not have the bubble capsules. So we're missing all the cytoplasmic. So seems like on the surface, when people collect it, which is where most of the data is the morphology is very different.
That was some type
it has
of
a buoyant lipid layer.
Siphonophore.
And I just image the one that we have, and it's totally missing all of the lipids. So it's totally missing an entire capsule layer around it. And this is like the best, healthiest ones that we've caught.
Is that from the net?
No. This is from this is yesterday's dive. Huh? Oh, I mean, the test is pretty small, tiny, but the cell is that big. I mean, defined cell, it's the spine. And then I was able to use that vibration trick to measure the stiffness. And from the data, we'll get some amount of elasticity.
So those are calcite, the calcite.
That's calcite. Yeah. No. Even the spines are calcite. Yeah. They have a very cool. It's using the moment of inertia trick. So the cross section is a triangle. So the spine, you know, classic. We do the I-beam in mechanics. The I-beam is the strongest of you. So it's using that trick to make a Y-beam. The cross-section of all the things that are dangling around is actually a Y. It makes it stiffer. Yeah.
Testing 1, 2, 3.
Gotcha.
But it's very clear it sheds certain kind and maintains another kind. I think we collected one today. Yeah, we collected one in a bottle. Yeah. I think we collected a Hestigerella. Huh?
I haven't seen one on the screen properly.
No, you have seen it. The shimmery. Yeah. The big shimmery.
No, they're paying it at a steady speed.
First couple of days, but you can't see the test.
To it the last like 2 minutes. You're not.
Oh. What is that?
That's what I'm wondering.
You know, it had a big red head.
Inside? This is outside?
This is too fast.
I'm trying to notice that little noise on the out hauler of the docking pad. Will you stop on the winch for a sec?
Okay, I'll stop.
Alright, start hauling in again.
Okay.
I do try and look up periodic
We're at 200 meters. Do you guys want to take over?
Roger.
Okay, Commander, stop. Okay, you can take command.
Go, command.
Okay, let's just start coming back up.
Okay, continue.
Yeah. Yeah, continue up.
Roger, come off. Yes, we have it.
You have it? Okay.
Yeah. Thank you.
Okay, I'm gonna have some
go ahead.
canisters to look at, and I wanna grab something. So if you wanna just make sure we get our bottles, 2 at 50, 2 at the surface.
You could probably all stop for now.
All right, folks. As you can hear from the change in chatter here, we are headed back up to the surface for recovery.
They're just gonna stop their move right now. Yeah. I'll get them.
So we'll be doing recovery. I'm going to leave cameras rolling, and we are
Looking calm outside there right now, right?
currently planning to get back in sooner rather than later, but I don't have an exact time for you. I believe this evening still is what we're planning for, but plans change all the time, so don't hold me to that. I don't have an exact time for you, but hopefully we'll see you there, and thanks for seeing us now. Enjoy.